happy
Yeah.
that you are able to hook up finally.
Mhm.
So, how are things in California with the ongoing pandemic, sir?
It's very good. It's very good. I'm in San Diego, so it is little better. But of course, now it is increasing here also.
All right.
So yeah, so situation is bad everywhere, but I think India is better.
Okay, sir. Okay, sir. So one thing, Dr. Debashish, do you think Dr. Anand Srivastava can share his screen to see if it's working? You want to try
Yeah,
that?
we have to make him a
Let me
co-host. Suresh sir, you can make him.
push.
Uh,
Okay, so you allow it because I'm trying to.
Yeah,
Yes,
you
sir.
are the
Yes, I
co-host.
gave the
Sir.
control.
Oh,
Uh,
so I'm the host. Okay,
Mr. Gopi, if you can make me the co-host
okay.
as well. Thank you.
Yes, yes.
Yes, sir.
Yes, I can now share. Can you see my screen?
yes, sir.
Yeah, very much, sir.
Yeah.
Can you try to put it in the slideshow mode? Let's see. So that's working.
Can you see now?
Yeah,
Yes,
very much.
very, very clear. Very clear.
Okay, and let me go back. So you can see, can you hear this sound?
San Diego-based stem cell research institute
very
believes
much.
its discovery may have
Audible.
saved the life of a COVID patient.
Good.
Yeah.
So we are okay.
Debashish,
Stop share.
Yes.
you need to you will be involved in hitting the record. Okay, fine. I'll take care of the recording here. That's not an issue.
Record, you can hit. Once the meeting starts, only we can do it, right?
All right. All right. Dr. Anand Srivastava, we are recording this meeting. I hope it's okay with you.
Yeah, it's fine.
All right, sir. Thank you, sir. Thank you.
Nowadays, everything is recorded. That is good.
All right. Just for documentation, sir. And
Yeah.
Professor P.K. Suresh, sir, the event is now in YouTube Live.
Great.
Thank
So
you,
you
sir.
can see the icon in the left-hand side.
Okay, sir.
Professor Debashish, sir, you can record the meeting in the cloud. Okay.
Yes. Once the meeting starts, I'll definitely.
Yeah,
Thank you, Gopi.
that's the one.
I'll whenever required, I'll give you a call, sir. You may stay
Okay.
as a.
Fine, sir. I can help you.
Gopi,
Yes.
the attendance sheet can be collected. At
Yeah.
the end.
Yes, sir. That's possible.
So it's going to start at 7 o'clock. Means you're 8:30.
Yeah. But we wanted to start a little earlier because we are having the inaugural after this. Normally, we would have the inaugural before because of the time difference. We do it this way. So for the inaugural, we are having Dr. Kannan, Professor Krishnamurthy Kannan as the chief guest. He was involved in ubiquitin research and a biomarker for hematopoietic stem cells. And we are also having our industrial partner is
Nilanjana, madam.
Advanced Cells.
We can try your slides also. I'm sorry to interrupt.
No, you will show the slide, no?
I'll show the slide, then you can read. Not a problem.
Yeah,
That's
yeah.
how
You
it is
please,
possible.
yeah.
Fine.
Dr. Anand Srivastava, your talk will be around 45 minutes or how was it? You have to unmute Dr. Anand Srivastava. You have to unmute.
Debashish, if you can
Sorry.
make
It will be around 45.
Good.
Yeah.
And after that, we have some time for
45.
question and answers from the audience.
Sure.
Okay.
So you are which place in India right now?
We are in Vellore. Katpadi is the nearest railway station. We are about 135 kilometers away from Chennai. It's about 200 odd kilometers away from Bangalore. And we have the Christian Medical College here, a famous place.
Vellore is very famous. Yes.
Right. And we have a golden temple here, somewhat like the golden temple in Amritsar, is smaller. As far as the resource person concerned, we have a couple of other.
Professor Anand Premarajan, sir. Sorry. Are you able to hear me?
Yes, I can hear you very well.
No, Anand Premrajan. There is another Anand, sir.
Okay.
Sir, Debashish sir, I am here.
So, would you be kind enough to download the PPT and put it in the presentation mode? The reason why is I have to admit many people. They are coming in. So
Yeah, I will do that one, sir,
if
better,
my
because-
screen stays engaged for presentation, no, it would be difficult.
Debashish sir, admitting
sir.
I will do it, sir.
Okay, fine then. Fine.
I will
I'll
do that. Yes,
I'll put it.
So
Debashish,
you can
this
concentrate
Javis
only
here.
on one issue.
Yeah,
and-
Javis, please.
Yeah,
Make
it
me
is displaying
co-host.
on
I'll
my
do that.
Thank you.
Okay.
Good
Make
morning,
me
Anand
co-host.
sir.
Yeah, I have made you co-host as well.
Yes.
No, the admitting part, you don't worry. You just take care of the YouTube, Debashish. You take care of YouTube.
YouTube, it is going live, sir. Not a problem.
That is it. Admitting can be done, not an issue.
Anybody else wants a co-host status? Please kindly let me know immediately. Asha Dev, ma'am, I'm making a co-host. Manjuala, ma'am, I'm making a co-host. Okay, so fine. What I'm doing is now I'm going to share the introductory slide for Dr. Srivastav. Let me see if it works. Is it visible?
Yes.
It is. It is.
Oh, sorry.
Shall we get started, Dr. Anand Srivastava?
We have another 6 minutes. 8:15 is the time, right? Good.
Dr. Debashish,
Yes, ma'am.
it is not visible fully. Can you make it?
Ma'am, what you can do is you can open the PPT in your screen, separate file. You can read from that. It would be in the centered mode.
1 minute.
You have to do Alt Tab toggle.
You can. How to do that?
Is it possible for you to see another file? I mean, the file you could have downloaded.
Or I can read it here. No, it is not possible.
Okay, madam. What I'm doing is I'm not putting it in the presentation mode, right? So that it would be visible.
Yeah, at least I can see the whole thing.
Yes. I'm trying to.
Now it is okay.
Is Gopi there or gone?
Yes, sir. Tell me, sir.
Gopi, if possible, can you come back during the inaugural?
Yeah.
After that, we'll take care. Just
Yeah.
part of the inaugural. Yeah.
Yes, sir.
Thank you.
Okay.
Is it visible now, ma'am?
Yeah, now I can.
So, Dr. Srivastav, let me introduce. This is Dr. and Professor Nilanjana Das Mitra. She is working in mushrooms and she is a very eminent scientist here. She is recently is rated listed under top 2% of biological scientists in India by the Stanford University. That ranking. And she would be chairing this session. Along with her, we have Dr. Anand Premrajan. He will be moderating the session. Yes.
Yeah, two Anands. One is Dr. Anand Premrajan there.
So over to Nilanjana, madam. We will start within 3 minutes. It's 8:12 now.
Debashish?
Yes, sir.
I make a few statements, very brief statements, if you don't mind.
Please, I think if you have to make...
Shall I start now?
Yeah, please, you can begin now, I think.
Yes. A warm good morning to everyone. We're very happy to commence this two-day virtual international conference on stem cells. The title is Update on Stem Cells, Academic and Industrial Perspectives, Future Challenges. Dr. Debashish, you have hit the record button.
Yes, I just start.
Okay.
Record to cloud. I'm going for record to cloud, sir.
That's correct. Okay.
Yes.
Okay. A warm good morning, everyone. I'm Suresh. I'm the convener of this two-day virtual international conference on update on stem cells, academic and industrial perspectives, future challenges. Welcome to one and all. Just a brief remark about the event. We have scientists from the USA, Europe, and India participating actively and delivering lectures. As on date, we have over 300 registrants who are going to participate, and a couple of instructions for the registrants. In order to be eligible for the e-certificate, you need to attend at least 5 sessions, including the inaugural, and you need to fill out the feedback form, which will be circulated either today or tomorrow. Without much ado, I'll hand over the session.
Just a small announcement. Our chief guest has also joined. I have made him the co-host. Happy to have you, sir. Dr. Krishnamurthy Kannan.
Okay. Okay.
We are about to start the first session, sir. I think we can go ahead with the session. Professor Hilanjana, we can, I hope we can start.
Yes, yes.
Thank you so much for the welcoming. Yeah.
Thank
Good morning to all of you. Good morning, Dr. Anand Srivastav. It is my pleasure to introduce Dr. Anand Srivastav today. For the first session, he is the first speaker of the first session, and I will give brief introduction of Dr. Anand Srivastav, and he has been associated with leading universities and research institutions of USA in affiliation with University of California, San Diego Medical College, University of California, Irvine Medical College, Salk Research Institute, San Diego, then Barnum Institute for Medical Research, San Diego, then University of California, Los Angeles Medical College, USA. He has developed several research programs, and he has an extensive research experience in the field of stem cell, which is documented by several publications in many peer-reviewed scientific journals. Dr. Srivastav is the chairman and co-founder of California-based Global Institute of Stem Cell Therapy and Research, GeoSTR, headquartered in San Diego, California. The company was formed with the vision to provide stem cell-based therapy to help those who are suffering from degenerative and or genetic diseases like Parkinson's disease, Alzheimer's disease, autism, diabetes, heart disease, stroke, spinal cord injuries, paralysis, blood-related diseases, cancers, and burns. GeoSTR is a leader in developing most advanced stem cell-based technology, supported by leading scientists with the pioneering publications in the area of stem cell biology. Company's primary focus is to discover and develop a cure for human diseases with the state-of-the-art unique stem cell-based therapies and products. The regenerative medicine provides promise for treatments of diseases previously regarded as incurable. So with this brief introduction, I would like to request Dr. Anand Srivastav to start his speech.
Thank you very much, Professor Das. I know this is, and good morning, everybody, because this is good morning in India. And of course, I'm in San Diego, California right now. And can you hear me clearly?
Very clearly.
Yes.
Very clearly, sir. I would request all other people to switch off their video, only let Dr. Srivastav to have his video. And, that will help the YouTube video recording and getting that.
Wonderful. And thank you very much for particularly Professor Suresh and Professor Das. And I know it's so difficult during the time of COVID to organize such kind of conferences. And particularly when you are dealing from India to abroad, like international, the time zone and all these things. So it's so difficult to really manage and organize. So certainly needs a lot of courage and dedication. So I really feel very thankful to Professor Suresh and Debashish, you to managing all this complex algorithm for Zoom and making everybody available can see, recording all these things. And of course, I very humbly pay my regard to Professor Krishnamurthy, who is the chief guest at this time point for this meeting. And now I would like to go for my screen actually. And if you may remove your screen, Professor Das, then I can put my screen on.
Dr. Debashish, you can remove this.
Yeah, I have to do that. Yes, so please carry on.
Yes, very good. So I can now try to. Can you see my screen now?
Not yet, sir. Not yet.
So, screen. Still, I am seeing the screen of your screen. So, share. Stop share. Okay. Still, I see only your.
You may try sharing the whole screen, sir. We are able to see your browser.
Presentation, sir.
No, but I have to.
Your screen is shared. Just click on the presentation. PowerPoint.
Okay. Where is my?
Yeah.
Can you see?
Yes.
Yes.
Okay, great.
Yes.
Always that's what I was telling it. A lot of credit goes to the Debashish that managing this complex.
Sir, it's our CTS who is really supporting us very tremendously. Technical
Okay, very good.
support, sir. Yes, please continue, sir.
Thank you very much. So, as you know, I'm here at San Diego, and this is Global Institute of Stem Cell Therapy and Research, that is GeoStar. And Professor Das already told about all, in fact, too much about me and GeoStar. So our aim is to develop treatment or therapies related to stem cell science and degenerative diseases using regenerative medicine. And this was actually, we formed this institution in 2008, almost 12 years back. And it's very interesting story. In quick, actually, I started working on stem cell science at University of California, San Diego Medical School in 2000. And in 2000, this modern stem cell science was not very much known. Of course, bone marrow transplant and hematopoietic stem cell transplant, entire world, including US, was practicing and is practicing last 40 years almost. But this concept or this idea using the stem cell to manage different kind of diseases and degenerative diseases or repair the damaged organ were not very much in practice. So I started working at University of California, San Diego in 2000, like 20 years back. And before that, I was interestingly working on the gene therapy concept in because human genome program in 90s. And so entire concept of human genome program was that if you know the correct gene or gene sequence of the humans, you may change the defective gene with the correct gene. So you may treat almost each and every metabolic disease practically, means theoretically. Yes, it is very complex. And I'll come, it's very interesting that today we are using almost a gene therapy kind of thing to counter this COVID-19 virus. So I showed in 99 that yes, we can change pancreatic gene with correct gene and it can be expressed. But, and I also showed that we can deliver tissue targeted gene. So a lot of things I showed in 2002, 3, 4, but gene therapy is really very complex. And so I was continuously thinking what may be the easy way to manage this particular problem related to metabolic degenerative diseases. So that's what I started culturing the human embryonic stem cell. That time it was embryonic stem cell. Now we are using adult stem cell, particularly the mesenchymal stem cell. But that time everybody started working on human embryonic stem cell. Then California Institute of Regenerative Medicine was formed. We fight a lot, in fact, and we fought. And 2004, California first time accepted officially to work on stem cell science against federal government and funded $3 billion to work on the stem cell science. So that's the way I started working. And then I showed that damaged brain can be repaired in 2005-06. So today, if we see the real problem, when the COVID problem came in the picture, and if I remember correctly, when President Trump was visiting to India in February, in fact, nobody was thinking that COVID is such a big, or going to be huge problem. But when President Trump came back to US, and after that in March, it started the news and all these things that it's going to be a huge problem, and almost 77 million people infected right now. And in US, almost more than 300,000 people died. In India also, almost 150,000 people died with COVID. But now India is doing very well, in fact, managing the COVID in comparison to our country here, US, I can say, because the recovery rate is very good in India right now. So saying all these things, when it was flaring up and nobody was knowing how to manage it, I'm talking March of this year, nobody was thinking really what is going to happen. And of course, there was only one way people were discussing: to manage your immune system or develop the vaccine. Developing the vaccine was going to take a lot of time, and we knew it is going to take a lot of time. So being a scientist, and of course, I get my education in Banaras Hindu University, and the philosophy of Banaras also inside me, that, okay, you have to keep thinking out of the box. If you are going to face some challenge and there is no answer, then really you have to think how I can counter this problem. And fortunately, as I told that I was working on stem cell science, and I used particularly this mesenchymal stem cell a lot to manage the autoimmune diseases and other kind of immune related problems. So then I started thinking, is it possible to really utilize the character and ability of mesenchymal stem cell, which is well known to manage the immune system and imbalance of the immune system. This is also well known to suppress the inflammatory cytokines and the inflammatory gene actions. And well, these all are well documented. And then same time, very same time, I think in April this year, one wonderful paper came and from China, and of course, China, Russia, and several countries collaborated, Poland, Brazil, US. And they published a paper that in China, they managed a severe COVID patient, not one, but I think 6, 7 severe COVID infected patient using the mesenchymal stem cell. Then that was the light actually in the tunnel. And I was continuously thinking in that same direction. Then I immediately talked to US FDA that till we really have any logical way to counter this virus, why not we should try to use mesenchymal stem cell, which is quite safe because safety and efficacy of mesenchymal stem cell is also well documented in animals and human models. So I thought, and I wrote to US FDA that why not we should use mesenchymal stem cell to counter this cytokine storm created by this virus. And we know this particular virus, that is SARS or COVID-2 virus, COVID-19, is when it goes in the body, it creates very strong cytokine storm. And of course, then it's a lytic cycle, and then it kills the cells. It creates a lot of problem with the lungs, and the water fills in the lung, then oxygen exchange problem comes, and a very severe respiratory syndrome comes out. So let's see, entire idea which I wrote to FDA, and then finally FDA approves. And I'm going to say, deal one by one all these points. So you already talked about this. These are the institute where I worked, in fact. So we know that this COVID-19 virus, basically, so attach with its S protein. We know this is RNA virus. This is a double bilayer envelope, bilayer fat lipid layer, and then RNA, and then it has a membrane protein, S protein. Very well and very known S protein, is scary S protein of this virus is well known. Everybody talks about S protein. So what it does, it binds with the ACE2 receptor of the alveoli of the lung, and that is epithelial. And particularly this bind to most of the time the alveolar type 2 cells of the lung, and then insert its gene inside the cell, and then mRNA start making the particular protein, and then it's the lytic cycle. So it ruptures the cell and comes out and multiply. This is the way this virus works. So it is well known that mesenchymal stem cell, which is a kind of adult stem cell, and the beauty of this mesenchymal stem cell, it is available in the fat cell. You can take out from the fat cell. You can get it from cord blood, peripheral blood, bone marrow. So this is very ubiquitously present all over the body, in fact, and very easy to isolate. Then second biggest character of this mesenchymal stem cell, that you do not need to match it from one person to another person. That is the another beauty of this cell. So basically you can generate these cells in the lab without any problem, and you can take from one person, donor to host, and you don't need to worry about GVHD, graft versus host disease or rejection of these cells. So quite easy. And you don't need any complexity. You don't need to really manipulate these cells. And these cells are well documented for its character for immune modulation and immune privileged situations. So what we see that this mesenchymal stem cell can really suppress the cytokine storms. So you see what it does. Can you see this this side, the photograph?
Yes, yes.
Okay, so this is mesenchymal stem cell, and it suppresses all the bad cytokines like IL-6, IL-10, and then second, it increases the IL-10, which is good cytokine, and of course suppress TNF-alpha, IFN-kappa, IFN-gamma, and all these inflammatory gene and cytokines actions. So this is in fact, and these kind of the cytokine storms are well known when COVID-19 infect a person. So the whole idea was not to kill, because there was no medicine, nothing was available that particular time, and still, like vaccine just came 3-4 days back, and still people are undergoing for vaccination. But before that, the best idea was to somehow neutralize these inflammatory cytokines. So once these inflammatory cytokines are neutralized, then the effect of this virus is neutralized. So even the virus is inside the body, the effect is not there. And then second best part, so when we inject the mesenchymal stem cell in the human body, and if the virus is there, then first step, it goes and neutralizes all those inflammatory cytokines and gene actions. So those are neutralized. So person does not feel the effect of the cytokine storms. Then second, this particular mesenchymal stem cell has parakine activity and secretes those molecules which enhance the regeneration of the damaged portion of the lung and also manage the fibrosis of the lung. So we see once it is the person was treated using the mesenchymal stem cell, all the cytokines is decreased. So we could rescue the patient, and I'm going to tell our results. So these are the detailed results, which I think just the data, showing that how it works. The most important thing is this. Okay, so this is the CT scan. So if you see the virus infecting the lung here, so its score is quite high. So we transplanted here the stem cells, mesenchymal stem cell, and this is the score of the virus or virus infection or loading of the virus, titer of the virus in the lung. And if you see, after 1 week, the lung started clear, and the patient really got rescued from the COVID-19 infection. So this was so good to know that COVID-19 situation may be cleared or managed using the mesenchymal stem cell. So this was the idea, and then I wrote to the US FDA, and as you know that US FDA is very, very quick in that sense, and I really thanks to them that they immediately approved emergency compassionate use of the mesenchymal stem cell, so we can use this mesenchymal stem cell to treat the COVID patient. So we are one of the most big pioneer institution in US to get this approval from US FDA to treat the COVID patient using mesenchymal stem cells. So now I will come on very interesting. So this was the part of the mesenchymal stem cell using the for the treatment of the COVID-19. So of course we are under the process of treating several patients. We treated very patient who was under coma for almost 60 days because of this COVID infection and ARDS, that is respiratory distress syndrome. And he came out, and now we are under the process of treating several patients. So one was, the idea was to how to suppress or neutralize the COVID-19 triggered cytokine storm in the patient. And I would say that we quite successfully achieved that using the mesenchymal stem cell. But the point, again, a stem cell, it's complex. You have to manage the stem cell, and then it has to be in good quality and all these things. So bringing to the masses and treat the masses may not be very easy using the stem cells, because you need a particular kind of infrastructure and the trained clinician and doctors. So certainly this cannot and may not replace the vaccine. So till we were waiting for the vaccine, then this is, I can tell this is one of the most effective way to manage the COVID-19 infection. So now the vaccine is in the picture, and a lot of questions and anxiety and enthusiasm related to the vaccine, and this mRNA vaccine, and Pfizer, of course, Pfizer made it, and AstraZeneca made it. So it is very important for us to know, and I think viewers may like to know, how really this vaccine works, that is mRNA vaccine, and why it is important to store at minus 70 degrees centigrade or minus 20 degrees centigrade, which is also a big infrastructure problem for developing country, and even in the advanced country, because you have to develop really very strong infrastructure to store this vaccine in minus 70 degrees centigrade and transporting this vaccine for several places. So let's see, and I would like to show my viewers how really this vaccine works and what are the difference between this Pfizer vaccine and AstraZeneca vaccine. And this is very interesting, one thing, and I'm very encouraged, in fact, because indirectly, this both vaccine are working on the basis of a kind of gene therapy mechanism. So let's see how this mRNA vaccine works and what's the mechanism. So I'm just first going to talk about Pfizer and Moderna vaccine, and this I made immediate because the vaccine just came and the mechanism and all these things. So I just sketch, and I took all this information from available on the studies on the journals and YouTube and Google and all these things. So I just want to make clear this is not 100% my idea to show the mechanism. It is all available on the internet and all these things. Just I'm trying to summarize for the viewers. So this is the basically. So what they did, this is the virus. Okay, this is the S protein of virus. So they made a sequence for mRNA which is coding to S protein. Okay, and they surrounded this to lipid nanoparticle. And this, because mRNA, so mRNA we know very fragile, okay? It is so easily digested by mRNA, and mRNA enzymes are available means in the dust everywhere, okay? So it's very, very fragile. That's what they are storing this in the minus 70 and keeping away from all kind of problems. So to make it very stable. So this is mRNA. And so they made a lipid nanoparticle with this mRNA, so it can go to host cell. So and this mRNA, the beauty of this mRNA which they made, it is not entering in the nucleus of the cell. So when it is not going in the nucleus, it is not integrating in the genome of the cell. So that is the best part which they are telling, and that's a lot of curiosity also whether this is mRNA, and if it may integrate with the nucleus, and it can change the genetic structure and all these things of the cell and human being. So it is not like that. Okay, so this mRNA is going to this cell cytoplasm. Of course, this is ribosome. This transcribes the S protein. Then it express on the MSC surface receptor and make antigen here express, and then this T helper cell comes and bind with the T cell receptor with this and start making the cytokines. And this also express the B cell, macrophage, and dendritic cells. And then this triggers the B cell proliferation, and then it makes plasma cell, and then plasma cells makes the antibodies, which goes and bind to virus and neutralizes it. So this is the entire mechanism. Here the MSC, major histocompatibility complex H1, and that is also bind here and produce the cytotoxic cells, which creates the cytokines. And if the virus is going to attack here to this cell, COVID-19 after the vaccination, this cytotoxic T cell can kill this host cell as well, but not till the virus infect this. So this is the mechanism of particularly this mRNA vaccine, which Pfizer made. So here, what are you doing? Here you are inserting a foreign gene in the shape of new mRNA, that is a nucleic acid, to the host cell. So you can say this is a kind of gene therapy. And now they are telling that, yes, it is very safe. So the point is here that how the Human Genome Program started in 1990. And the idea was to sequence the human gene, and so you may change the defective gene with the correct gene. And if you know, recently, Professor Chris Murthy is, I think, interested in hematopoietic stem cells. So he may be knowing that they treated sickle cell disease, transferring the disease here. And this New England Medicine Journal just published that paper that they use the beta globin gene to transfect the cells, hematopoietic stem cell, and infuse to the sickle cell disease patient, and they rescue those patients. So now they are coming on the point that yes, you can do you can use the gene therapy basically kind of here to counter the virus. So now come on this AstraZeneca Oxford vaccine. They are in fact one step ahead from the Pfizer. Okay, so they use the DNA vaccine actually. So DNA is little better stabilized molecule, so it does not disintegrate as quicker as mRNA. And here they are using the adenovirus. There they are using the fat nanoparticle to wrap the mRNA and deliver in the cell. Here, this AstraZeneca is using adenovirus to inject the DNA, that is genetic material again. So now you see, this is opening a very new chapter in the field of medical science, because last 30 years, starting from 19, I know I was working on gene therapy, and as I told in 1999, I showed first time that you may insert pancreatic gene to correct the pancreatic gene, and you may express that gene in proper manner. But we could not do the gene therapies for several diseases, and it's very difficult to get the approval to do the gene therapy because you use the virus vectors to deliver the gene in the cell. But because of the COVID-19, they allowed this adenovirus and now they are going to vaccinate every single person. So it means they are going to inject this particular vector to everybody to use the DNA vaccine. So this DNA is, of course, going to nucleus, okay? In that case, it was just lying in the cytoplasm, that mRNA. Here, the mRNA is not this mRNA is not going into the nucleus of the cell. It is just directly going to ribosome and thus translating the protein. Here, this DNA is going to nucleus from here. Of course, they are not going to, they say it is not integrating to the genome, but they are transcribing the mRNA. And from that mRNA, it's the same mechanism, B cells, plasma cells, and making the antibodies for the virus to kill the virus or neutralize the virus. So the point is, because this adenovirus and secondly, that nanoparticle, fat nanoparticle, these two things are going to go in almost, I will say, at least 50-60% of the human being. And that would be extraordinary data. And if this adenovirus successfully, which data is telling it is very successfully transcribing and translating the particular protein. So now using the same adenovirus, because now it is allowed, okay, to use in the humans and at a very mass level. So using the same adenovirus, you may pack any kind of DNA or gene here to deliver in the cell to treat several kind of genetic diseases. Of course, that need a lot of other researches and a lot of other complexities are there, but using the viruses to deliver a particular gene is now here. We are doing it for thousands and thousands of the people. So that is the best part. And why this AstraZeneca does not need -70, they need just -20 because it is DNA. So it is more stable, so you don't need -70 degrees centigrade. And of course, then protein vaccine and all these things, which is not. So this is 2 vaccine, and this is the mechanism of action for these 2 vaccines right now. So credible when the vaccine, and we'll see how this going. And we know that we have to take 2 doses. One is day 0 and 21st day for AstraZeneca, and for Pfizer, you have to take 2 doses, 0 and 28 days at the place of 21 days. So these are the different booster dose timings for these 2 vaccines. And data shows it's very promising. So hopefully it will be very, very promising. But the point again here, there are 2 points here now. Of course, you can neutralize using the vaccine. But if somebody is infected already, have compromised lung, and we are listening each and every single day that people are dying because of multiple organ failure as well, not only the lung or the respiratory failure. So why this is like there is multiple organ failure because of this infection of this virus. So what happening, in fact, when this virus is going and it's triggering a strong reaction, of course, and then killing the lung cells and making the scar of the lung cells, so a lot of cell debris and other things are triggering. So when the cell debris are there, they are going and clumping. Small blood vessels are getting clogged. Blood supplies are getting hampered. So patient may get a stroke, heart attack, kidney problem, liver problem. So those are the problems. So important is somebody is already infected and having problem. So this vaccine may not work. The only thing may work is the traditional way of treatment, like we are using dexamethasone or steroids and all these things. But for those patients, still the mesenchymal stem cell is the best option. And then secondly, even we are seeing nowadays and listening and seeing the patient that when the patient are rescued from and coming out from the ICU, still they have compromised lung and multiple organ problems. So, and damaged organs. So how to manage those damaged organs? So again, to manage those compromised organs, that is the post-COVID effect. I think the most easiest way to manage using the stem cell treatment, that is mesenchymal stem cell, because it is well known to manage the compromised organ. So the combination of both is, I think, one of the best way to manage this particular pandemic. So let's. So I'm at the last of my slide and talk, and I think I am very much within the time, hopefully. So we treated, as I told, that we treated very severe patient who was under coma for 60 days because of COVID infection, and again. We are very thankful to US FDA. They allowed us to treat that so severe patient who was almost on the death bed. So we were on the news, all over the news channels and all these things. So I'm going to show you what happened.
San Diego-based Stem Cell Research Institute believes its discovery may have saved the life of a COVID patient on death's doorstep. Our ABC attendance reporter John Horn explains why the new treatment could be game-changing while we await a vaccine. He was having a stroke. He was under dialysis. Liver function was declining. The prognosis grim for this 53-year-old COVID patient deteriorating in a New Jersey hospital. The man fell into a coma that would last nearly 60 days. Nothing was working, in fact, like plasma therapy and all the classical kind of treatment. That's when San Diego doctor Anand Srivastava and his team at GeoStar sprung into action. GeoStar is an international stem cell research firm based in the Golden Triangle. It got special clearance from the FDA to infuse the 53-year-old man with mesenchymal stem cells taken from an umbilical cord. Slowly, his renal and liver function came towards the normal. He came out from the intubation, and now he's talking. Srivastava says it took about 3 weeks for the man to recover. GeoStar interviewed the patient's family, keeping identities confidential due to privacy laws. Basically, we had no hopes for him, but it turned around. I feel so happy to see, because he has a daughter and wife, of course. Srivastava says this kind of treatment could be key in helping COVID patients survive until a vaccine arrives to protect people from infection. John Horn, ABC 10 News. GeoStar is planning to conduct a double-blind study for the treatment to make sure that its conclusions are correct. That study is expected to take about 3 months.
Okay, so thank you very much. So this is all for today, and please ask if you may have any question. And
Thank you, Dr. Srivastav, for your wonderful presentation. And we have been enlightened with so many facts regarding COVID-19. Though I don't have much exposure in stem cell research, but I have 2 basic questions. Like we know that patients who are suffering with diabetes or having high blood pressure, we have heard that COVID complications, it is being escalated.
you're right.
And does the association between COVID-19 and diabetes differ with the different types of diabetes? This is one question. And the second question is, do anti-diabetic drugs have an impact on disease progression?
Okay, wonderful. So, so diabetes is one of the thing, and this is in fact, we are seeing almost every single patient who are suffering with diabetes, and generally type 2 diabetes, because type 1 diabetes in the kids. Okay, and so what does diabetes do? Diabetes, in fact, creates a big problem for energy production utilizing the glucose, and because of diabetes there are a lot of other secondary problems, and we say underlying problem, health problem, and not only for diabetes, any underlying problem creating a big problem for those people who are suffering with COVID-19. So when COVID infection is there, what it does, it goes and hampers it binds the epithelial cell of the lung and hampers the exchange of the oxygen and means the energy metabolism finally. And diabetes has the same problem. The diabetes also creates the problem in the energy metabolism because of the utilization of the glucose and oxidative phosphorylation and all these things and mitochondrial problem. So the person who is suffering from the diabetes and going to get the COVID-19, so diabetes and COVID-19 together does not only form 2 + 2 = 4. It synergizes the problem, but multifold because of the diabetes. And I would like to say one thing here, that we know that the young people does not get too much problem with the COVID. Yes, they get infected, they feel little bit problem or coughing or something, but they really don't have those kind of problem related to those people who has underlying situations or old people maybe. Why it is? Because the young people has a lot of stem cells in their body to fight this. And secondly, this particular virus does not affect the mesenchymal stem cell because mesenchymal stem cell does not have ACT, that particular receptor, that is ACE2 receptor. Okay, so that is the main point. So that's the diabetes part. And your second question was that is it going to affect different kind of diabetes? Is that? Yeah. So
anti-diabetic drugs, they have any impact on this?
Okay, so it depends again, like anti-diabetic drugs like metformin or something, if you are going to give. So the point is anti-diabetic drugs, if you give, so you are going to, somehow if you managing the diabetes using anti-diabetic drug or something. So I very mixed reaction I can tell at the data, what I have with the patients, that most of the time anti-diabetic drugs, when the person having diabetes and going through COVID-19 infection, those helps because they can manage the diabetes through anti-diabetic drug. And then at the same time, you see the problem is like with the diabetes people, what is the classical treatment? Dexamethasone, Remdesivir, and if you are in India, you get antibiotic also to manage the secondary infection to the lung. So what happens that dexamethasone increases the blood sugar, and diabetic person already has the problem of managing the blood sugar. So in that case, it is so difficult to give dexamethasone and that is very important to manage the cytokine storm of the COVID-19 patient. So in fact, it's a very fine balance. In general, if you can balance probably better way, yes, diabetes drug may help.
Thank you, sir, for your nice presentation. And now for question and answer session, I will request Dr. Anand Premarajan. He's the moderator. Now over to Dr. Anand Premarajan.
Thank you, Dr. Srivastava. It was, in fact, an enlightening and presentation because you started with basics, what is a stem cell, so even a layman can understand. Then you move ahead with the Human Genome Project, stem cells, and you talked about the papers published in the October 2020, and you have updated with what is going to come, right? So you compared both the Pfizer and AstraZeneca vaccine, which was, of course, a very new, and you informed us in a very subtle way that we are able to understand even this minute detail. Even a layman who knows the biology a little bit can understand. And you have also spoken about the how or what are the precautions we can take. So that means when you spoke about the energy management and other things, so in India we are all doing a lot of yoga and other things. So indirectly we can understand that there are so many other things which can be done. So there are some questions which have come in the chat box. So So first question is from Mr. Krishnamurthy Kannan, that is MSC, can it be called a stem cell?
Okay, so that's the question.
Yes, yes, sir. And another one is, sir, as MSC have benefited effect to immunomodulation, but most of these findings are in vitro, and we all are aware that MSC uses LL37, and this molecule is also associated with autoimmune disorders. So, is there any consequence related autoimmune conditions in case of COVID-19 finding? So this is the question, sir.
Okay, good. So first question Professor Chris Murthy asked about to question whether this, we can say it is a stem cell or not. Okay.
Yes,
There's a big debate going on. And again, I personally say, you see, I say it is a brain massage. So according to the definition of stem cell, that it can give same kind of cells when proliferate, and if you give to signals, it may differentiate to different kind of organs. This is not a certainly we cannot say it is a pluripotent stem cell. There are different kind of stem cell, multipotent, unipotent, pluripotent, so there are a lot of definition. So if we see this definition giving same kind of cell, and if given a specific signals can differentiate to different kind of cells, you can define this as a stem cell. But again, there's a lot of debate going on, and this is the point. Sometime I feel that how to really, we before this COVID-19, we were talking a lot of science and debating a lot of things. This is not, this is this, this is this. As we know everything. Certainly, we don't know everything. That COVID-19 told very well to us, and that we struggled a lot, and almost millions of the people died. So that is my answer about the stem cell. Is it a stem cell or not a stem cell? The point it behaves like a stem cell, or whatever you try to say, but this is very important cell which really helps in a lot of situations. Secondly, if you that question that sometime some report tells it may create autoimmunity or something. So in case of COVID-19, we do not have long-term study. We just started 3-4 months, I think 6 months back, and now we are treating and collecting the data. The most important point is that we rescued almost all patients who were very severely sick or affected with COVID-19. And at this time point, when we don't know what is the answer even, and if we can save some life using something like yoga, like respiration, like pranayam, like Ayurvedic medicine, like turmeric powder, like Chyawanprash or Triphala, okay? All are good. But in long term, yes, we have to know whether it is going to really create some other kind of problem. But point is here that this is really working and nicely. And to those patients who already gone through classical treatment till plasma therapy, starting from dexamethasone, remdesivir, plasma therapy, nothing working, then only we can use this. Because FDA only allows when the patient already tried everything and nothing is working. So that's the point here.
Thank you very much, sir. So another question from Seetal is several cases of people infected with COVID-19 have been reported to catch the infection again, and deaths also have been reported. With the primary infection, antibodies against the S protein should be formed. So how is the secondary infection still so severe? And how is the vaccine going to be any different or solve this problem? This is a question from Seetal.
In fact, she asked very nice question. Every single person is talking about that. Okay. And I personally do not want to undermine this vaccine. As I told very well, I told very clearly that this is opening a entire new era. Like we are going to give every single person a kind of gene therapy, basically, which was all continuously stopped by entire world. Even US FDA, just name the regulatory agencies of entire world. Nobody allowed it. Okay. So the point is putting that, is it not possible if you take this vaccine and again you may get infection? Yes, it is very much possible because we don't know at this time point. We just 2 days ago, we started giving the vaccine to the people. And it is also correct that several people who got COVID-19 infection, few of them again got the infection, and those infections were even more severe than previous one. But again, those are, I think, exceptional case. And we have to study those cases because it is also possible that those particular people who got reinfection of the COVID may have some kind of underlying situations which we may not be knowing or may not be reported. So there are several if and buts, but she's right. We don't know right now.
Thank you, sir. And another question which is asked by the Khushi Srivastava, and the first question also was from her when she asked about the immunomodulation. Now she is asking about before COVID-19 pandemic, why there was a restriction on usage of adenovirus and why they have approved now.
That is very good question, in fact. So there are two answers. So, okay, I'm going to talk scientific answers anyway. So, where this is so severe situation, the pandemic, and several people, more than millions of people died, in fact. This is the saddest part of this situation. And what to do then in that case? Okay, making a vaccine going through classical way takes a lot of time, almost 10 years, 9 to 10 years. And when there is a war, and this is a war, in fact, enemy is at the door. So you have to fight with that enemy, whatever you have. You are not going to see what kind of weapon you have in your hand. So this is the situation. So that is the reason they allowed this RNA and DNA and RNA vaccine, and also allowed the adenovirus. Of course, it is chimpanzee virus, and it is well known that it does not harm to human beings because this virus from chimpanzee. And but again, she asked a very good question: why they did not allow it? Because they are always worried if that virus goes and creates some kind of problems, okay, to human being. But again, the research document, their data, which they did already and checked on thousands of the people with double-blind trial, they showed it is quite safe. So now it is safe, and we are suffering with biggest problem of the entire century. So yes, this is the big fight.
Thank you, sir. And another question is that how COVID is related to blood pressure and blood pressure patients.
How COVID is affecting or creating problems?
Yeah, affecting. It's a comorbid situation. So they are asking about how it is related to blood pressure, high blood pressure patients.
Okay, very good. So again, we have to first understand one point here, that COVID is very severely affecting the lung. What is the purpose of the lung? Lung is the lifeline for our body from the external atmosphere. environment. Without lung, of course, every organ is important, but exchange of the oxygen and then finally taking that oxygen towards the entire body and generate the energy, lung is now compromised. Blood flow is needed more and more to oxygenate your body. And then somebody already has the problem of the blood pressure and all these things. So he's going to have more problem because now lung is compromised, oxygen exchange is not there, then energy metabolism is hampered, then there is a scar in the lung, the dead cells are coming in the blood circulation. Those dead cells are clumping, maybe blocking the narrow capillaries of blood vessels, creating another problem. And then if it is blocking the nephrons, glomerulus, then again the kidney problem, filtration problem. So it's a lot of secondary problems to those people who are suffering with, again, blood pressure or any kind of underlying situation, because lung is very important organ to supply the lifeline to our body. All other things come second.
Sir, another question from Khushi Srivastava. I am reading the question. Continuation to the previous question, as you have stated that Oxford vaccine works on genetic material, which is stable molecule, but we have that there is a second wave of COVID-19 and that is more dangerous. So is there any chance that as we are interfering with genetic material, is the reason for development of more dangerous vaccine, more dangerous virus?
These are very hypothetical situation question, in fact, because we do not know. And the second wave of COVID is not because of some mutation in the COVID. Just we heard that in UK, they got some different kind of, or some mutation in S protein of the virus. And again, that again, another debate or discussion you can start that, okay, mutation means what? It is also possible that that particular mutation, which is on S protein, may not bind to ACE receptor. Okay, so that is also very possible. So virus may not create any problem, but we don't know right now. So point is that after giving this vaccine and this DNA, particularly she's talking about Oxford vaccine. So this vaccine is going to translate a specific protein through mRNA transcription. So hopefully, we hope for better outcome. And they already tested thousands of the people. So the data shows that they get 95% people rescued. So that's quite good, in fact. So there is no reason right now, there is no reason to believe that it is going to not work or it is going to create more problem in the future. There is no reason to believe that because data is very good and not in 1 or 2, 30,000-40,000 people, I think.
So last question, sir, and with this we will be winding up because sir, the last question is, in a family, one person is affected by COVID, whereas all the other 4 people are not affected. Why is it so?
This is very, again, I'm telling my own experiences related to this, but herd immunity, you know already that there may be some infection and they develop the antibody for that. And I have seen that people who in one family get affected and young people just get some infection a little bit, or if they go for the PCR test or antibody test, they get positive antibody test and PCR test, but they do not feel any problem or symptoms of COVID. So this is very much possible to develop a herd immunity because of the close proximity of a COVID infected person. So a lot of different things are going on. And that's what I think India is doing very good. Now, 0.1% people are going in the severe situation like dying or something, but most of the people are recovering. So I think it is because of the herd immunity.
Thank you. Thank you very much, sir. So over to Nilendra, madam. Would you like to say something?
So, thank you so much, Dr. Srivastav. We are really very lucky to get you here, and we are thankful that you have spared your valuable time, and we are really enlightened for so many things regarding COVID-19. Thank you once again. Thank you.
Thank you very much, everybody, and Dr. Rajan and Professor Das, Professor Krishnamurthy, and Debashish and Professor Suresh. This is wonderful meeting you all people in India. So for me, it is like 8 o'clock, going to be 7:40 or 7:50 evening. So I'm going to prepare for my dinner now. Getting ready for my dinner. So please carry on, and I may join after a few minutes again.
Yeah.
Thank you.
Thank you.
Thank you very much, sir.
Thank you, sir. Very good lecture, sir.
Thank you, sir. Thank you, Dr.
I'd like to announce our Chancellor sir has already joined. So over to Professor Javed Osborne. He will be the in charge of the inaugural session. Thank you. I'll get it over from him after the inaugural session. Thank you very much.
Yes, thank you, Dr. Debashish. So we still do have another 10 minutes time. So Chancellor sir, should we wait, sir?
Has the chief guest joined?
Chief guest has joined.
Yeah, Professor Krishnamurthy joined now.
The guest of honor has to join, sir.
Dr. Kannan?
Good morning, sir.
Good morning. Which part of Tamil Nadu you belong to?
Nagarkoil.
Nagarkoil, where did you study?
I studied in Delhi, then I was in IIT Bombay. Then
Here in Tamil Nadu?
Tamil Nadu, I didn't have an opportunity to study at all because my father was
School also went out?
I went to Kerala. So my father was posted in Kerala, so I studied there. Then
he was in the central government service?
Central government service.
Oh, then you would have been going around the country.
Yes, exactly. And plus, I got this National Science Talent Scholarship. So that allowed me to go and work wherever I wanted.
Oh.
As a young boy.
You speak Tamil or Malayalam at home?
I speak both.
Both?
Both, yes.
What's your mother tongue?
Tamil. So we are all migrants from Tamil Nadu to Kerala, my grandparents and all. So the original route is still...
The Devikulam pyramid area?
No, not Devikulam, what is that? More towards, more closer to Trivandrum.
Mm-hmm. Yeah.
Because my mother is from Vadivishwaram.
Uh-huh.
So, Vadivishwaram has been for my mother, it has been an inspiring place. It produced the maximum number of civil servants those days. So education became a parameter in our house.
Yeah. Kanyakumari is really known for education.
Yeah. So
earlier days.
Even MS Swaminathan married from Vadivishwaram. His father.
Where is that village, Vadivishwaram?
Nagarkoil. You get down the bus.
It's part of Nagarkoil now?
Yes. It's just walking distance from the main bus stand.
Mm-hmm.
Of course, I was very fortunate that I had very good mentors in my life during my research. So I was also in England in Imperial College.
When were you in Nagaland?
Nagaland, Dr. Abdul Kalam one day called me, Kannan, that state needs somebody. So you better go there. So I said, sir, if you think I'm fit of it, I will accept, because I had no idea what Nagaland was till I landed there.
Chancellor, sir, the guest of honor has also joined. So shall we?
Yeah. Who?
The guest
Guest
of
of
honor.
honor, Vipul Jain, sir. He's the CEO of Advanced Sense. CEO of Advanced Sense. It's a company. It's a stem cell based company.
Mr. Vipul Jain.
Yes, sir. Yes, sir.
Is he there?
Sir, yeah, he joined now. So with your permission, shall we start, sir?
Yeah, if all of them have come, but still we have 7 more minutes. Oh,
Yes, sir.
9:30, yes. It's 9:23. Mr. Jain?
Yes, Doctor.
He is not seen here in the screen.
No, he's here, sir. So he turned on his camera now. He's visible.
Where are you located, Mr. Jain?
We are in Delhi, sir.
Delhi. Okay. You belong to Delhi?
Yes.
Sir, he is the industrial partner for this conference, Advanced Cells, and we are interacting with a person named Dr. Sachin Kadam. He is the Chief Executive Officer, and he is also going to be delivering a lecture today, sir.
Oh, very good. We couldn't visit Delhi for a long time.
You are most welcome, sir. Anytime you are in Delhi, please.
No, we have an office. Of course, one is VIT office. The other one is there is an association called EPSI, Education Promotion Society of India. We have an office near Apollo Hospitals. I'm the president of the association for the last 5-6 years.
Okay.
So we are all doing only through Zoom. Where is your office?
We are actually in Noida. The office is located in Noida.
Okay.
Lab and office both are located in Noida.
This agitation, has it affected Noida also?
Not really. I think only figuratively speaking, they have closed the border, one border, but then there are several other borders which are open. So life remains normal, but figuratively speaking, yes, the agitation is going on.
What is the temperature there now?
It's almost 3 degrees, sir.
Huh?
Almost 3 degrees.
3 degrees. Oh.
Pretty cold now. But I believe Vellore would be still moderate.
Vellore goes up to 17, 18 degrees in the night time.
Still moderate.
Daytime it's hot, 29, 30 degrees. How many employees do you have?
We are now about 35, sir.
35,000?
No, 35. Just 35.
In the office.
In the office, we have about.
How old is the company?
We are now 7 years.
Oh, that's it. Have you been to VIT Vellore?
Personally, I have not, but
You
Dr.
haven't visited?
Yeah, that's on the agenda.
Before we start the inaugural session, I should overwhelmingly thank Dr. Nilanjana Das and concluding the earlier session. Stay back for the inaugural session.
Prakash, what is the strength now of faculty in SBST?
Sir, 76, sir.
76, sir.
Yes, sir.
It used to be more?
Yes, sir. We now we have a YL school and then a CBC, CBST, CBCMT. So bunch of faculty members.
You are divided.
Yes, sir.
How about research students?
Research students, we have 540 students as on roll, sir.
540, yeah?
540, sir.
Oh. Because I still remember we were about 125 or 130.
Hopefully in coming years, it is going to grow up like with the new program, sir.
Start?
Yes, sir. Yes, sir. Jebes?
Yes, sir. A very good morning to everybody. I would like to welcome you all for the 2 days international conference on the update on stem cells, academic and industrial perspectives, and future challenges. So now I would like to request the Dean, SBST, Dr. Pragasam Viswanathan, to give the welcome address.
Thank you, Dr. James. Honorable and beloved Chancellor, sir, the Chief Guest for this international conference, Professor Krishnamurthy Kannan, former Vice Chancellor, Nagaland University, is also an esteemed academic researcher in the field of stem cell technology. The guest of honor, Mr. Vipul Jain, the Chief Executive Officer, Advanced Cells, the convener of this conference, Dr. Suresh, faculty organizers, colleagues, students, and the participants. Warm good morning to all. Due to the extraordinary situations caused by the COVID, we are continuing with the glory of international event had become a challenge, and we decided to go virtual. However, I'm delighted to see that the enthusiastic interest evinced by the brilliant personalities from several institutions and industries from India and abroad. We have invited eminent personalities to update our knowledge on stem cells, their application in various sections of medical biotechnology. I'm also delighted to state that there are 360 registered participants for this conference across the globe from 12 different countries, including India. Stem cells have become regenerative medicine now. Researchers worldwide are extensively working on application of stem cell research, which harness the tissue engineering potential to stimulate the repair mechanism of the body and restore the damaged tissues and organs. Therefore, the development of disease-specific therapies and treatments by exploiting stem cells' remarkable properties holds hope for improving patient quality of life and the survival with chronic disorders. The faculty members of the School of Bioscience and Technology and associated centers are extensively focusing their research on stem cells and its application for humankind. The School of Bioscience and Technology has been ranked between 501 and 600 categories under QS Biological Science subject ranking for the year 2019 and 20. It has also been recognized as top 10 biological school, top 10 research institute in India by the reputed corporate magazines. It is not unusual to witness this kind of a conference with our school. This conference will really bring several network scientific activities among our faculty members with the resource persons of this virtual conference. I hope this two-day conference would really bring several deliberations to the way forward our scientific activity at the school, and our students will undoubtedly benefit from this scientific task. Without further extent, once again, I wish to welcome you all for this international conference, and thank you.
Thank you very much, sir. Now I invite the convener of the event, Dr. P.K. Suresh, to say a few words about the conference.
Thank you, Dr. Jabez. A very warm good morning to everyone. Honorable Chancellor, respected Chief Guest, Professor Krishnamurthy Kannan, respected guest of honor, Mr. Vipul Jain, Dean Sir, HODs, another learned faculty members, and student friends. First of all, we would like to acknowledge the incredible support of the Honorable Chancellor and the other members of the management of VIT in organizing an event of this magnitude. We have organized events of this kind in the past, workshops and faculty development programs. However, this is the first time we are organizing a conference specifically on stem cells, and that too in the virtual mode, even though CBCMT has organized events related to this topic. The idea of, or rather this idea of conducting a virtual international conference on stem cells came after several discussions with like-minded scientists, representatives from the corporate sector, and academicians from VIT and outside. We received a lot of inputs with regards to the identification of suitable resource persons from Advanced Cells, our industrial partner, as well as from resource persons from Narayana Netralia, as well as from the Center for Stem Cell Research, Bagayam, Velur. Also, our respected chief guest, Dr. Krishnamurthy Kannan, has provided inputs in terms of resource person identification, and he has also circulated a quiz via Google Forms for the benefit of the participants, especially the students, going to be the scientists and stars of tomorrow. He will be discussing about this quiz during his inaugural address. We are glad that all the speakers, eminent scientists, clinicians, resource persons from industry, and entrepreneurs from the USA, Sweden, and India have accepted our invitation and agreed to present in this conference. In this regard, as Dean Sir said, we are very happy to state that we have over 300 registrants representing 21 states in India and also 11 countries, including India. Specifically, the resource persons will be discussing topics related to embryonic stem cells, adult stem cells, induced pluripotent stem cells, and hematopoietic stem cells. In addition, lectures will be delivered with regards to stem cells of the ectodermal and mesodermal lineages. Also, other topics to be covered include stem cells and pancreatitis, engineered 3D models for tissue engineering, and topics related to research and clinical aspects pertaining to human bone. In general, this conference is expected to provide a common platform for students, research scholars, academicians, scientists at various levels, government labs, private labs, as well as those in education institutions, clinicians, and biomedical science and biotechnology related entrepreneurs to update themselves with regards to the current state-of-the-art cutting-edge developments in stem cell research, therapy, and related aspects. Also, this interaction would pave the way for collaborative research endeavors and provide opportunities for networking. This joint conference will also reiterate the importance of academia-industry collaborations in terms of academic and research exchanges. This interaction is expected to pave the way for cross-pollination of ideas, potentially culminating in focused translational research-based endeavors. I hope that all the registrants make the best use of this opportunity to acquire more knowledge about stem cells. Thank you and all the best. Now over to Dr. Jabez Osborne. Thank you.
Thank you. Thank you, sir. Thank you very much. It gives me immense pleasure in introducing the guest of honor of today, Mr. Vipul Jain, who is also the industry partner for this particular conference. So Mr. Vipul Jain is a serial entrepreneur who specializes in spotting business opportunities while they are still in nuanced stage. He is passionate about healthcare and how future of healthcare will be revolving around the concept of personalized medicine and cellular biology. In 2013, Mr. Vipul established Advanced Cells, which is a research-based company focusing on providing cutting-edge stem cell therapies for patients with life-threatening diseases, also catering to lifestyle diseases and anti-aging technology. Advanced Cells is slowly rising to be a leader in the space of regenerative medicine, which is routinely called the future of modern medicine. Under Mr. Vipul's guidance, Advanced Cells now have produced more than 10 stem cell lines that are used by doctors, hospitals for therapeutic purposes, by various research organizations, and by pharma companies for drug discovery. Advanced Cells have also produced an all natural plant material based scaffold, which is patented and used in therapeutic purposes, especially in bone regeneration and skin burns healing. Mr. Vipul's moonshot is to be able to print 3D organs and convert them to transplantable organs with the use of stem cells and scaffolds. With these few words, now I request Mr. Vipul Jain to address the participants.
Thank you. Thank you very much, sir. Honorable Dean, sir, Honorable Chief Guest, sir, Mr. Dr. Suresh, all the dignitaries present today, all the student friends, learned faculty, thank you very much for inviting me. Thank you very much for having Advanced Cells as the industry partner for this wonderful conference. Our relationship with the VIT goes a bit back. We have been associated with VIT and working on different projects and trying to work on different projects for some time now. And it is an absolute honor for us to be an industrial partner in a seminar of this magnitude organized by VIT. So thank you very much for the same. While I'm going to leave the technicalities and the technology of stem cells, it's going to be discussed extensively over the next 2 days in this forum. I just wanted to take up this time to see or to basically say how my vision of stem cells is going to pan out in the future. If there is one thing that this current year, the pandemic has taught us, is probably it has very well established that every disease affects the human body, every human body in a different way. There is no such thing as just one way of dealing with a disease. And therefore, the concept of personalized medicine is going to be more and more relevant as the years goes by. And very, very soon in the future, we will see medicine to be extremely personalized. And I believe that stem cells, cellular biology is going to be the cornerstone, is going to be the central point on which the whole personalized medicine field is going to pivot. Whether it is 3D printing of customized organs, whether it is stem cell rejuvenation of the body, whether you talk about human longevity, everything is going to revolve around personalized cellular biology. And as industry, as research institutes, it actually becomes our duty to be collaborative, to join hands in a endeavor to find basic research, to find ways in which collaborations can be done, in ways in which industry participation can be done, and basically all this research can be brought from bedside to the bedside. Because until and unless that happens, until and unless our patients are not able to get the benefit of all this research, then there is no point of doing this anyways. I am a person who strongly believes that the first person to live till 150 years of age is today already born in this world, which means that over the next 10 years, 20 years, we are going to see some massive improvement in the way cellular biology is going to play the role in the healthcare field. And it is not going to be possible till the time institutes of magnitude like the VIT and industry is going to come together, going to collaborate, and going to make sure that all our research is going to get passed on to the patients. I also believe that stem cells are really the future of modern medicine. everything that we see currently is going to get disrupted. Every surgery, every treatment that has been devised right now is going to get disrupted very quickly in the future. And we are not very far away from the time when, all of us can go to a chemist shop and buy a jab of stem cells and basically inject it ourselves into our body and see if any ailment can be corrected by the use of that. Yes, there is a lot of research still pending in that field, lot of clinical trials still pending, lot of data yet to be generated. But if institutes like VIT and industry partners come together, we can definitely generate that data and make sure the governments are able to stand up and take notice. So I'm going to now leave the forum in the capable hands of all the eminent scientists, all the dignitaries that are present here. I hope that the next 2 days are going to bring in a lot of deliberation on how stem cells are going to become the future of modern medicine. And a lot of partnership is going to come out of this session where we can then put our heads together and get solely with one purpose of making healthcare absolutely personalized in the future. Thank you very much. And thank you again for having me here.
Thank you very much, sir, for your inspiring words, especially on personalized medicine and also about bridging the gap between industry and academia. Thank you once more, sir. I now invite our Honorable Chancellor, Dr. G. Viswanathan, to give the presidential address.
Our chief guest of today, Dr. Kannan, the former Vice Chancellor of Nagaland Central University and a senior scientist. And our guest of honor, Mr. Vipul Jain, CEO of Advanced Cells, the Dean, Prakasam, Head of the Department, professors, convener, Dr. Suresh, and other professors, participants in various parts of India and 11 other countries. I am extremely happy to know that the School of Biosciences and Technology is organizing this conference, update on stem cells, academic and industrial perspectives and future challenges in association with Advanced Cells Noida, which is the leading cell company solution provider in India. To keep the body in good health is a duty, otherwise we shall not be able to keep our mind strong and clear. Every human being is the author of his own health or disease, are the popular sayings from Gautama Buddha. Mahatma Gandhi said, "It is health that is real wealth, not pieces of gold and silver." Heart disease is a leading cause for maximum health globally, as per World Health Organization, with about 9 million deaths in 2019. This was followed by stroke with 6.2 million deaths, lung disease with 3 million deaths. In 2020, as of 21st December, 76.8 million people are affected by COVID-19, leading to 1.6 million deaths. Moreover, people living with pre-existing health conditions like heart disease, diabetes, or respiratory conditions are at the high risk of complications and death due to COVID-19. Soon we will be able to get from WHO the complete assessment of the direct and indirect impact of this pandemic virus on mortality and morbidity. To our knowledge so far, none of the human diseases have caused complete lockdown for several months as like this in COVID virus disease. We have learned lessons from the pandemic, which has not only brought physical lockdown of the entire world, but lockdown of its economy as well. Coping with this crisis, which is referred to as new normal, preparedness, understanding the opportunities and challenges, reflection, reaffirming, and resilience, our healthcare system are key points the situation has made us understood. The scientists, healthcare professionals, and healthcare industry have important roles and responsibilities to recover humankind and from this pandemic and many other ailments like heart disease, cancer, diabetes, etc. I understand that stem cells are the body's raw materials, the cells from which all other cells with specialized functions are generated. Blood cells, brain cells, heart muscle cells, and bone cells are formed this way. Stem cells have the ability to build every tissue in the human body. Hence, there is a great potential for using stem cells for therapeutic purposes by tissue generation and repair. COVID-19 mainly causes damage to the lung. Hence, it must be possible to use them cell therapy to combat this virus infection. While the scientific community is working to develop vaccines and drugs against COVID-19 pandemic, novel alternative therapies may reduce the mortality rate, protect the people. Recent use of them cells for certainly ill COVID-19 patients in small group of patients and emergency use authorization of stem cells by the Food and Drug Administration has created excitement among the medical community. At present, it is not only the COVID, there are many health issues people are challenged with: heart disease, cancer, diabetes, stroke, obesity-related issues, preterm birth, infertility, emerging and re-emerging infectious diseases, including dengue, swine flu, bird flu, chikungunya, Ebola, continue to rise among the population with changing lifestyle, lack of immunity, and other environmental causes. All these situations alarm us the importance of strengthening and accelerating our clinical research. The quality of health or healthcare system and services in the society is considered as one of the crucial factors that define a government. However, this pandemic period has proved that we need to improve our healthcare sector. Healthcare is a very important factor to determine the physical and mental well-being of the people. That is the sole reason it is highly recognized as a great contributor to the country's economy. In addition to the research and development undertaken by healthcare professionals in medical institutions and hospitals, in collaboration with healthcare industries, biotechnology has greater potential to contribute. This is because biotechnologists in academic institutions like VIT are well-trained and knowledgeable in fundamental cell biology and molecular biology. The bridge between academic and industry has to be strengthened to achieve transnational research in areas like stem cell therapy. You know that VIT is one of the biggest department or schools in biotechnology in the country, with a number of scientists and researchers. I strongly believe that biotechnology-based stem cell research will come up with affordable healthcare measures, products, and processes. With constant improvement in research and development, medical biotechnology research in VIT and elsewhere can surely become well-received foundation for solving all the problems in human health time to time, including the current virus disease. I'm happy to note that the scientists and industry experts from all over the world are participating in this conference, and researchers going to share their research findings, experiences, and thoughts in this advanced topic in biotechnology. Thanks for recognizing our School of Biotechnology as a partner for collaboration. SBSC, as I told you, is a leading school in our, not only in our institution, but also in the country. We get the best of students from all over India, other foreign countries to join BTech, MTech, and MSc and research programs in biotechnology. We want every student, research scholar, and faculty of School of Biosciences and Technology to contribute to betterment of society. VIT will always support their initiatives. The future of our healthcare sector depends on the commitment and excellence of qualified younger generation. The economy depends on healthcare because people cannot survive without proper healthcare. Technological advancements will only happen if the next generation researchers and medical professionals are equipped with right knowledge and attitude. Hope the deliberations of this conference will contribute to achieve this. I'm happy that the School of Bioscience Technology has taken an important initiative of arranging this conference in contemporary topic. I congratulate the Dean, organizing faculty team, resource persons, and the participants. I also thank the chief guest who has agreed to come. He's not only a former Vice Chancellor, he is a senior scientist. I understand he is doing very well in this, and probably we will invite him later during the normal times to come and visit us and guide our faculty. And also, I would like to thank Mr. Jain for supporting this conference. I wish the conference all success. Thank you.
Thank you very much, sir, for your insightful speech. Now I request the convener of the conference, Dr. P.K. Suresh, to introduce the chief guest.
Thank you, Dr. Jarvis. It's me a great pleasure to introduce the chief guest for today's function. I'm honored and privileged. The chief guest is Professor Krishnamurthy Kannan, Chair Professor Biotechnology, Manav Rachana International University, Faridabad. Professor Krishnamurthy Kannan has played a key role in producing quality manpower for India since 1985 in the area of biotechnology, pioneering fundamental work on hematopoietic stem cell homing and addition in 1992, and mooted the idea of cord blood banking in 1994, involved in the commercial scale production of whey protein from 0.6 million liters of buffalo milk. He has held very senior positions in the diagnostic and pharmaceutical sectors. He has popularized biotechnology and stem cell biology in schools and universities in India as part of their academic program. The then President of India, Dr. APJ Kalam, invited him and appointed him to become the Vice Chancellor of Nagaland Central University, Nagaland, India, 2006-2011. We feel extremely privileged to have you, sir, to be part of the inaugural function and deliver the inaugural address. Over to Professor Krishnamurthy Kannan. Thank you.
So good morning, everybody. Thank you, Professor Suresh, for the kind introduction. I would like to start. Will we call Dr. G. Vishwanathan a MSc or a STEM cell? You might be wondering why I am dragging in Dr. Vishwanathan into the whole thing. In the morning, I raised a question whether mesenchymal stem cells are stem cells. So I want to clarify some of the very basic points because I have spent since 80s, okay. I have spent my life, one ubiquitin, another is stem cells. So, when we were looking at it, stem cell, we were only 2 people in the country. I'm talking of 80s. That too, Cancer Research Institute, my friend, Dr. Ananda Rao. And otherwise the stem cell was not even known to people. Ubiquitin, of course, wasn't. Now, I raised the question whether Dr. G. Vishwanathan will be called an MSc. I didn't use the word mesenchymal stem cell, or will you call him a stem cell? So one fundamental difference between a stem cell and an MSc, it is also called marrow-stromal cells. Stem cells only receive signals. Some signal has to come from outside. You heard Dr. Srivastava talking about paracrine, etc. So per se, stem cells don't know what to become. It is the niche, what we call as the environment, which decides the fate of people. So I would call Dr. Vishwanathan as a person who created the niche. So he is not teaching, he is not doing anything, but he created a niche for VIT to become where it is today. So these are all people whom I call as facilitators. So when people talk about mesenchymal stem cells, personally, I would not agree with that word, because if you are using it, there are two things. One is the environment. Like, you might be the most brilliant guy, but if the environment is horrible, you will never mature to become a brilliant guy. So if the niche is damaged, you put any damn stem cell, nothing is going to happen. It is like a lot of children join the university. The teacher is supposed to be a stem cell because he decides what direction the student should take. So when a school child passes and joins the college or university or wherever they are, depending upon the inputs which the teachers share with the students, the student will become a stem cell biologist or an immunologist or a biotechnologist or whatever. So the poor child doesn't know what he or she is going to become. It is the niche in which they are sitting. Like, somebody gets admission in IIT, he becomes something. If somebody gets admission in VIT, he becomes. So a stem cells per se do not have any this thing. They don't produce any signal. They only receive signals. And they are, so what you are getting, a child which joins the university or a school knows nothing. A 5-year-old child is nascent. It has no qualities, nothing. It is the quality you imbibe in them. If you want to become the musician, you do that. So I want it to be very clear that stem cell is not what is doing the trick. It is the environment. And if environment is damaged, any good stem cell you put, nothing is going to happen. Okay. So I want this to be very, very clear. So a lot of people think stem cell, put it and magic will happen. No. If the environment is not okay, nothing will happen. So I want this message to be very clear. Number two, a lot of people talk about biotechnology, and it is, talked as if it is something out of the world, and let me tell you, when I had to introduce biotechnology in school, I had a very simple definition. A biotechnologist is nothing but an arithmetician who does addition, subtraction, multiplication, division. That's it. For example, you want insulin, so you take out the insulin from the source, whatever it is, that is called subtraction. You put it into a vector, this is an addition. Then you put it in a bacteria, make multiple copies, photocopies, that is called multiplication. Then sort out the cell which makes the insulin from not there, and that is called division. So essentially a biotechnologist only does this. So he is essentially playing around with the biomolecules and creating what is important. Okay. So I want everybody to be very clear on this aspect, that please don't get carried away by stem cells. Stem cells can't do a thing if the environment is not okay. So if there's a damage in the environment, nothing happens. Okay. Now, you have to look at this, which I'll be presenting in greater detail. I'll be presenting in greater detail in the afternoon talk, how all these concepts developed. But I want you to appreciate what I just said. Stem cells don't know what to become. So the most classical experiment were done by one lady called Ethel Brown in Hydra, and another was, Spearman and Mangold. Now, these are very classic experiments in the sense that he discovered that, whenever an egg gets fertilized by the sperm, it rotates by certain degrees. And the sperm entry point to the opposite end, if you take those cells and transplant, some magic happens. For example, Ethel Brown worked on this thing, on the Hydra. So she put something and she could create a whole Hydra just by transplanting that cell. Similarly, Spearman and Mangold also did a beautiful experiment on salamander, and I just want to share this. So they took, as an experimentalist, they took two types of cells, one salamander which was colored and another one which was white. So they had a colonies of these, and then they took the colored one and then fertilized it. It kept on dividing. It goes through various stages. One of the stages is gastrula. So at the gastrula stage, if you take certain cells and transplant it, say, from the colored one to the white one, which is in the gastrula stage, say, and you put it underneath, I don't want to go into details right now, but you transplant this cell, a magic happens. You get a bi-headed salamander. And the bi-headed salamander is white. What you have transplanted is from a colored salamander. So in other words, the cell which is there doesn't know what it is going to become. You put some signal, it will go into that. It will become eye, it will become, whatever you want. So I want you to understand that this is called the Spearman organizer. Okay, and this should be taught to you. So it is a part of embryology and developmental biology. So you should understand how is this signal happening. Now, when an egg fertilizes and divides into several cells, that's why people talk of embryonic stem cells, because at that time it is not yet nascent. It doesn't know what to become. So you take it out, like inner cell mass in case of human or any mammalian system, and then it will, depending upon the environment you give, you do. Now, I also want you to understand that one of the early people who were to work on this was Metcalfe and Leo Sachs. And they developed a very beautiful assay, and whatever biotechnology today claims as a blockbuster company all happened thanks to the original work of Metcalfe and Leo Sachs. Okay. And when they were trying to mature these stem cells, they realized that there are certain factors. So that's how erythropoietin was discovered, GCSF was discovered, the whole range. So whatever biotechnology success came from the hematopoietic field, how blood cells are formed. So you have an institute, or Dr. Vishwanathan believes biotech is great because it is making money. And he wants that we should also, our children also should be benefited by it. Okay. Now, coming to a very fundamental question that a lot of people, I want also to appreciate Lance Steiner. I don't know how many of you know him. Before, every disease was treated by removing the blood, and a lot of people died. Then later, people started injecting blood from cow and buffaloes and doing it. People died. There was an anaphylactic reaction. Then later, people started transferring human blood. So some people succeeded. So they said, wow, I know how to treat disease. But a lot of people also died. So this is where Lance Steiner's contribution becomes very important. How he discovered the A group, B group, AB group, O group, etc. So now Lance Steiner started working because he worked with some of the greatest minds as a early training. He was matured into a good scientific thinker. Today we hear all kind of crap being talked about. Stem cells can do this, that, whole lot of people. I feel very disillusioned because we are taking people for a ride and we cannot afford to take people for a ride. And this is where I want to come to Donald Thomas, who actually developed the bone marrow transplantation. When the first patient died, he said, no, I must relook. Then he looked at the dog system. He said, no, blood grouping is not important. HLA typing is very important. I'm getting a lot of noise from behind. Can somebody unmute their thing, please? Okay. So, I lost my line of thought. Let me just recall. Okay. So basically what is happening is that there's a lot of misnomer, and right now mesenchymal stem cells is everybody wants to earn cash on it. But we don't have any evidences what's happening. it is like the earlier days before Landsteiner did. Some people succeeded, so you can claim, oh, it's a fantastic thing. Then some people die, then you will not even talk about it because that is removed from the data. So I remember when I go through the Donald Thomas, he did experiments in this thing. He discovered dog lymphocyte antigen. Then he discovered, okay, there's a similar one in HLA typing. So now this cord blood banking, why I was talking about it, everybody think cord blood banking is the next one to do. A lot of people started. But the amount is small. Number two, we find that because of this HLA typing problem, there will be host where, the tissue rejection can happen. So it's a very complex subject. And when people like me have spent a lot of time trying to understand this. And that is why we never claimed anything. I was never in the media to tell about this can be done, that can be done. Because I had a mentor like Fred Sanger, who always said, let others do all that. We are nobody to open our mouth. He didn't even, when he did the sequencing technique, he never said what the future of sequencing will be. The noise is made by people who never did that work. So I want you to be very, very careful that stem cell therapy requires a lot of, proper clinical trials, and our government of India, ICMR, is still not geared for that kind of stem cell research. We like to import things from outside. But the point is our patients are going to react differently, exactly like COVID reactions are different in Europe, you're different in USA, different in India, different in Southeast Asia. So basically, I just want to say that our body has something like 10 to the power 13 to 10 to the power 14 cells. The type of cells are about 218, probably might be more, because as we learn about astrocytes, etc., neuronal thing, it might even increase the number. Now the question is, out of these 218 cells, from where are they coming? So I have done a rough calculation. It may be about, our body only has about 40 stem cells. Hematopoietic stem cell is one, which creates the 12 T cell, B cell, neutrophil, basophil. You can create any damn thing out of that. But that's all what it can do. That's all the capability. That's why we call it progenitor cell. Whereas something which can create a whole thing we call totipotent, like a university teacher can only take children who have passed from school to this thing. So that is a multipotent. So they might, so your fate is decided at different stage. In school, you can be anything. You come to 9th class, you become, okay, art stream, science stream, This is exactly what happens in our body also. That cells multiply, you have ectoderm, endoderm, mesoderm, then it starts going in. So just like every profession. So today's life, and that is this thing. Now the question is, what is stem cell then? So I give the example of two people. One is Rutherford, another is J.J. Thomson. They are the ideal definition of a stem cell. Why? J.J. Thomson produced 8 Nobel laureates. Rutherford produced 13 Nobel laureates. Both and Rutherford was student of J.J. Thomson. So between them, they produced 18. So you should be able to produce your own Nobel laureates. That is what is a stem cell. The critical mass has to be produced, and the rest of them will become scientists, whatever it is. So a teacher is a very crux of it. So Dr. Vishwanathan, my suggestion is, how do you pick up people who are true stem cells, who can make people differentiate properly? People, nowadays we recruit people because they have so and so papers. That's not the way to go about it. There are people who look at it in a broader perspective. So stem cells are different diversity that what we create. So, so this is, unfortunately, what is happening is we are not looking at this aspect, who is it? And those people will never open their mouth. You have to go and look for them. That is why I remember country had a national science talent search. And you have Venkatraman, who got Nobel Prize, is also my batchmate in science talent. So country had a mechanism to plot talent. So it is very important to pick up teachers who can create that. So stem cell per se has no function until the environment is okay. So how do you create the environment is the critical aspect of stem cell research. Now, one of the greatest hopes, which has been shown in case of, I remember Fanconi's anemia, cord blood was used because the mother became pregnant again. The child was already, they had a child with Fanconi's anemia. Luckily, the HLA matched, so they did transplantation of the cord blood, and then the child is still alive today. That's how all the noise came about cord blood, but it went through strict scientific quality assurance. So Donald Thomas was involved in that. So a good worker won't make noise. He's worried. Everybody says I had 100% success. It's not possible. Because it might be 10% success, 20%. We have to keep on improving. Right now, stem cell success rates are minimal. Number two, bone marrow transplantation means you need a big you need an institution like VIT to do it properly. There are so many divisions involved. I remember I was in the committee when Alok Srivastava was permitted to start bone marrow transplantation in Vellore, because I was in the stem cell committee. And I said, no, this has to be developed. But the question is, it is just like we are talking of probiotics, how much of bacteria or how much of stem cells are required, which will go home and adhere to that place and replace the defective one. So homing and adhesion is the most critical thing in the whole thing. Monoclonal antibodies all will identify, but it doesn't give a functional assay. The functional assay has to be done, and today afternoon I'll be talking about how functional assays are done. For example, mouse, Till and Matlock showed that I can take from a syngeneic mice and make the mice live. Otherwise, they would have died after irradiation. That's why I say Hiroshima, Nagasaki, one question I've asked in the question, could we have saved their lives? I think you must go and read Till and Matlock's classical work, because any radiation will destroy the hematopoietic system. So you'll have to put those cells back. And where do you find it? It's a very interesting thing, but nobody has actually seen a stem cell. All these are claiming the CD34 positive. Basically, you're always injecting 10 to the power 5 cells or 10 to the, so that hopefully there are 1 or 2 stem cells. So it is still a mystery. Nobody is injecting a single cell. So whether in your pooling, you are able to get the right pool. So which density gradient of cells to use, probably finding, is a lot of mathematics involved, and people have done that. So people like Metcalf and all have done this in great detail. So please, I want today's audience to understand, please, this is not a honeymoon. You are not going to make a money tomorrow, no way, because patient safety is at most. And I think until you are honest about it, that look, I want to save a life, please don't get carried away. That's my little bit of experience I can tell you, because I could have jumped into stem cell way back in 80s, but no, because there is a ban on using embryonic stem cell. IVF clinics were available until Shinya Yamanaka came with the factors, but that is still not clear. We don't know what are the issues involved, but it looks promising. But they are also talking about haplobanking. I think Shinya Yamanaka's work has led to some progress in the field. But I want people to appreciate that there are a lot of question marks. Please don't get, feel great. Until it becomes like Landsteiner kind of work, we are still very far. So with a cautious note, I would like to advise, it's exciting tissue engineering, any damn thing you can talk about, because they are all self-organizing systems. So it is not that the scientist has created, you put the right cell, nice environment, the tissue will form. So it's the beauty of stem cells and the initial, they are self-organizing system. We do not do anything. As long as you know what factors need to be put, it will form. So when tissue engineer claims I made this, it is happening naturally. That's the beauty of stem cell and its environment. So it is a self-organizing system you have to understand. And so when people say I did tissue engineering, nothing very great about it. You put two things together, it will happen. Like you take an octopus, and if a shark comes and eats away one of its limb, it has a natural capacity to regenerate. That is why it is called regenerative medicine. You have to do nothing. It just happens. Make sure there is no infection. That's all what we do. And this is where this theory of spontaneous generation, which we talked about food spoilage and all that, which the classic work with Louis Pasteur did, but here stem cells are automatically regenerate. And, so this is where we differ from Pasteur when it comes to stem cell. I don't want to go on and on about it, but I hope I gave my message very clear. Please be very, very cautious. If it's a businessman entering, please be very clear. You can't take people for it because tomorrow you'll be behind the bars if something adverse happens. So please is my advice. It's exciting, but people like Dr. Viswanathan should create an environment to make sure, a group of people are able to provide quality stem cells, properly validated, and assuming risk is there. It is like vaccine also, some people might die. We all know it. So it is not that anything is 100%. We measure the risk to benefit analysis, and that's all what we can do even in stem cell research. People only talk about positive results, negative results, nobody talks, and that's very unfair way of doing business. Or even academicians do it, we delete the points. So my request is that I hope I could convey what is the real meaning of stem cells. This is all what I learned in the last 40 years, and I've just shared with you. And thank you so much for having me, and I hope I didn't exceed my time. Thank you.
Thank you. Thank you very much, sir, for your inspiring and motivational talk on the fundamentals of stem cell research. And now I invite Dr. Asha Devi, Head of the Department of Biomedical Sciences, SBST, to give the vote of thanks.
Thank you, Dr. Jebus. Good morning, everyone. I would like to take this opportunity to thank our Honorable Chancellor, Vice Presidents, Vice Chancellor, Pro-Vice Chancellor, and the VIT management for their constant support and motivation in organizing conferences. I would also like to thank our chief guest and guest of honor of the day for accepting our invitation to be a part of this event. My heartfelt thanks to the Dean SBST for his encouragement and support. I extend my gratefulness to the organizing team convener, organizing secretaries, head of departments, chairpersons, and moderators who have been as a backbone in organizing this conference. I'm also thankful to the CTS, VIT for always being there for any technical assistant. I would like to extend my gratitude to the secretaries of the School of Biosciences and Technology for rendering their support. Last but not the least, I would like to thank the participants without whom the event is not possible. Thank you all once again. Have a good day.
Thank you very much, ma'am. Now we have come to the end of the inaugural session. So without wasting much of the time, I hope we are directly going ahead with the first session of the day. Thank you very much, everybody. Thank you, Chancellor, sir. Thank you.
Javed, I'm sorry to interrupt. Before we start the next session, we would like all of us to switch on our videos and we can have a gallery snapshot. All of us, let's switch on our videos, at least those who are the co-hosts and the snapshot now. Yes. Sir has yes.
Okay, Dr. Devashish.
Yes. Thank you, Professor Javed. Thanks for coordinating the inaugural session. So
1 minute, 1 minute. So I have another announcement. So yes. All the participants are requested to attend minimum of 5 sessions to receive the certificate. In fact, all the sessions, as it is definitely going to enrich your knowledge in the field of stem cell. And with no more delay, I'm passing it to the moderator of the first session, Dr. Shatarupa Banerjee. Over to you, ma'am.
Very welcome to the next session of this conference. So, in this
Shatarupa
conference,
ma'am, would you like to hold a bit because the guest introduction is to be done by the chairman, and you'll be there to collect the questions and finally,
Sure.
Yeah, thank you, ma'am. I'll just give me a second to make everybody a co-host. Yes, I welcome everybody to this session. Here, the speaker is Dr. Sachin Kadam, and the chair of this session will be Dr. K. Kannaviran. He is a senior professor of our school, and joining him, our moderator will be Satyarupa Banerjee. Over to
Hello, good morning, Professor Debashish.
Yeah, thank you, sir.
Am I audible?
Yes, sir. Yes, you are very much
yes.
audible.
Okay. So it's very good morning to one and all. And on behalf of VIT University, I welcome once again all the participants for this international virtual conference on update on stem cells, academic and industrial perspectives, future challenges. It's my pleasure to introduce the next speaker of the next session, Dr. Sachin Kadam. He is a very eminent researcher and scientist and stem cell biologist, and with the research interest on stem cells, biomaterials, and their applications in regenerative medicine, and especially making bioartificial organs. Dr. Kadam has earned his doctoral degree in National Center for Cell Science, Pune, and he is a very eminent researcher. I did his postdoc in the Johns Hopkins University, USA. And after that postdoc study, he joined as a faculty in DRDO's Defense Research University and the Defense Institute of Advanced Technology. And he also worked in D.Y. Patil Medical University and Amity University as well. So after I started as a faculty, I joined in Advanced Cells as a CTO, Chief Technology Officer. And he is presently leading as a CTO of Advanced Cells and an eminent researcher in Advanced Cells. Dr. Kadam has over more than 40 peer-reviewed publications in reputed journals or high-impact journals. And he has filed 3 Indian patents as well as an outcome of his research, and also contributed several book chapters. And he edited also a few of the book chapters. And he is also the editorial board member for many of the reputed scientific journals, peer-reviewed scientific journals. He has presented his research work in many national and international conferences. And Dr. Kadam has also been awarded with several awards. And just a few to mention, he received a Elsevier Young Scientist Award in 2008 and Distinguished Scientist Award in 2015 by Society of Bioinformatics and Biological Research. And it's very, fortunate. We are very fortunate to have him as a speaker for this session. And on behalf of organizer, School of Bioscience and Technology and Vellore Institute of Technology, I welcome Dr. Sachin Kadam to give his talk. Over to Dr. Sachin Kadam.
Thank you, sir. I hope I am audible.
Yes, sir. You are audible.
Okay.
Sir.
Yeah.
Yes,
thank you so much for your kind introduction, and I especially like to thank Professor Suresh for giving me an opportunity to present my work here. Without further delay, I would like to start with my talk. Is the screen visible now?
Yes, sir. Yes, sir. It is visible, sir.
Yeah. Okay. Great. So what I'm going to talk is basically on gingival recession defect and how to cure this defect using mesenchymal stem cells. But stem cells per se will not help. So what I have done is I have used a PCL-based bone regenerating scaffold, which we have created and we have patented also. So let's start with today's talk. A beautiful smile is the best ornament of the face and is the most primitive form of human communication. The harmony of smile is determined by especially a shape and positioning of the teeth. But when we talk about gingival recession, what happened is there is a displacement of the gingival margin from apical cemento-enamel junction, exposing the root of the teeth. And this creates an aesthetic issue and spoil the look of the person. There are various reasons why this gingival recession happens. Some to name is like there is something like predominant periodontal diseases, inflammation, improper flossing, aggressive toothbrushing, incorrect occlusal relationships, and dominant root. So these are some of the reasons why this gingival recession happens. If we see, generally this gingival recession, it's been seen in adults over the age of 40. There are mainly 4 classes of this gingival recession: class 1, 2, and 3, based on their severity. The class 1 is less severe, and just a small, defect in gingiva happens, which can be cured without any surgical intervention, and there is a 100% success rate when we are talking about class 1 gingival recession. In class 2, there is no proper periodontal attachment. Like, there is a loss of this attachment. There is no loss of this attachment with the tooth, but the gingiva starts receding further. Still, at this stage, we can recreate the gingiva, and 100% success rate can be achieved. Class 3 is where there is a loss of periodontal attachment, and the tooth is exposed, you can see in this particular figure, like tooth is exposed. And when we are talking about the correction, only 50 to 70% chances are there where it can be corrected. In class 4, it's a severe recession where a bone or a soft tissue loss happens, and root coverage is unprecedented, and there are only 10% chances by which we can achieve the success for gingival recreation. So when we have to treat this gingival recession, there are 2 methods: non-surgical and surgical. Non-surgical, like, you can use antibiotic, correct your brushing methods, soothe the tooth surface. So these are some of the non-surgical method. And surgical, it's a soft tissue grafting to prevent the gum recession. And this is a correct treatment plan. Like here you can see the classifications are shown in 2 corners, and as we go further, like from class 1 to class 4, the treatment of these particular classes it get minimized, and for class 4 the treatment is very poor, and you can say that there is no substantial treatment available. You cannot create gingiva again using the gum surgery, and there is a really need to look into this particular matter. So this is how the class 3 or class 4 gingival recession looks like, wherein there is a bone loss. There is a the skin of gingiva gets further down, and the tooths are exposed to the environment. If we are talking about the surgery, like the wherein we can use the gum graft from the healthy gum of the roof of the mouth. What is been done is you can see the roof of the mouth. From here the gum is taken out, some part of skin is taken out, and that skin is used for covering the exposed part of the tooth. So this is how the surgery is taken place. It's a invasive surgery. It needs cutting here and there, and there are many multiple scars has been created. So considering this in my mind, like we have decided like to develop something, some sort of treatment wherein we can cover this thing and create a gingiva and recreate the gingival recession, gingival tissue. For this, we need to have a base material, something like a scaffold. And this scaffolds, when we are talking about the scaffold, like since the bone and a soft tissue is being exposed during the gingival recession, the scaffold needs to be the bone regenerating scaffold. This scaffold should have the ability of osteoinductive, conductivity. It should be a biocompatible. So considering all these property, we have decided to go ahead with the preparation of scaffold. And scaffold was not just enough, because scaffold per se, like on its own, cannot give rise to gingival tissue. And for that, we have decided to use stem cells. So the main objective of this particular study is, first objective was stem cell isolation and propagation. So we all know that stem cells are a specialized cell of the body. Sometimes we call them as a mother cell. And these cells have an ability to self-renewal. That means they can give rise to the copy of the self. At the same time, these cells have an ability to differentiate them into different kinds of cells when they divide. So considering these two properties, these two properties are generally seen in many different types of stem cells, be it pluripotent stem cells, hematopoietic stem cells, or mesenchymal stem cells. So what we have decided is to use a human umbilical cord. From that, Wharton's jelly portion was taken out, and the mesenchymal stem cells were isolated, and the isolation protocol was standardized in our laboratory. Then these isolated stem cells were characterized for their surface markers like Ki67, which is a proliferation marker, smooth muscle actin, nestin, desmin, vimentin, and embryonic stem cells marker were also we have seen in these cells, SSCA4 and OCT4. By FACS, we have studied that these cells are CD44, CD73, CD98 positive, CD117 positive. This confirms that whatever cells we have isolated from umbilical cord Wharton's jelly are stem cell population. We also know that when stem cells are externally forced, or we supply some chemical or differentiation cocktail, they can differentiate into different lineages. Generally, a tri-lineage differentiation is studied for the stem cell characterization, and these tri-lineages, chondrocytes, adipocytes, and osteocytes. But in addition, we have also studied the neuronal and pancreatic lineage. You can here you can see that we have differentiated the isolated stem cells into adipocytes, osteocytes, chondrocytes, and neuronal cells using a chemical stimulation, like for adipose dexamethasone, IBMX, and indamethasone, osteogenic ascorbate, beta glycophosphate, etc. Just plants tell although for differentiation we can use chemical stimulation, but we were thinking is there a possibility by which we can use our Ayurvedic knowledge and differentiate these stem cells without any additional chemical stimuli? Like what we wanted to do is use the plant cell extract to differentiate these stem cells into adipocytes. So what we have decided is to use the plant stem cells for this particular purpose. And generally we have seen that meristeme is a plant portion where plant stem cells reside. So we have used the plant called Ciscus quadrangulus. This generally known as a hardzod in Hindi. And this plant has been very well known for bone regeneration, cartilage, and cartilage renewal, and so on and so forth. So what we have done is we have decided to use the stem of this particular plant for a differentiation, to see the differentiation of human stem cells into bone-like lineage. So from stem, what we have done is we have prepared a callus culture, because callus is the part, in plant stem cells, like it has a maximum amount of stem cells like a property. These are generally undifferentiated cells. We can, those who can regenerate into shoot or root based on the media which we provide. So whatever callus we have isolated, we have confirmed that by Salowski stress, by a formation of brown ring, that whatever a portion we want basically a phytosterol portion of the plant stem is being isolated, and that particular phytosterol can be further used for our human stem cell differentiation. So once we have isolated and characterized stem cells both from human and plant, we have decided to synthesize the scaffold for bone regeneration. So there are various different types of scaffolds available in the market. These are some of the list of the scaffolds which you can see for bone tissue regeneration people are using. What we have decided to go for electrospinning techniques. Like in electrospinning, there is a you all know, like there is a electrode difference, a positive and negative electrode. There is a polymer, and this polymer, under the charge condition, it comes on from positive to negative electrode, and at the negative electrode surface, it's form a paper-like sheet. So we have decided to use this simple method for generating scaffold. And the polymer which we have decided to use is PCl. Why PCl? Because PCl is FDA approved, and it's been shown that many bone or cartilage regeneration scaffolds had PCl already in their composition. So if you see this particular slide, the first picture, it shows that how scaffold looks like. It's a paper-like structure. And the second figure is the electron micrograph of the same scaffold, wherein we can see there is a weaving pattern of the polymer thread which give rise to a scaffold. So this scaffold is quite porous in nature. As I have already mentioned, only PCl per se, it will not be useful to regenerate bone. So we have to surface modify this scaffold, and for the surface modification, we have decided to use a plant called Ciscus quadrangularis, and the graphene is also used as a carbon source. So what we have done here is we have prepared a solution using graphene and Ciscus quadrangularis in a water by sonication, and the PCl scaffold was 60. We have dipped it in this solution, taken it out, dried it, again dipped it. So this particular cycle was repeated for almost 60 times. And here you can see the first is our PCl scaffold, and the third here is a PCl graphene oxide and Ciscus quadrangularis scaffold, wherein multiple layers of graphene oxide and Ciscus quadrangularis has been prepared on PCl scaffold. Then we have characterization of scaffold was done. What we have did is we have studied the scaffold underneath the electron microscopy again, and we have observed that the thickness of the fiber is been increased to a greater fold. Like initially it was 226 nanometer, which increased to 2369 nanometer. Even if we see the atomic force microscopy, the roughness of the scaffold is also increased when we modify the surface of the scaffold. So both these increase in thread and roughness can furtherly enhance the cell attachment and penetration and proliferation of our stem cells. We have also studied the contact angle method to see whether the scaffold is hydrophobic or hydrophilic in nature. What we have observed is the initial scaffold, only PCl, was hydrophobic in nature. But as we modify the scaffold by layer method, the scaffold becomes hydrophobic, and this hydrophobic nature of the scaffold further helped us to help cells to attach on the surface. Even we did the thermogravimetric analysis. So this TG analysis showed the degradation pattern of the modified scaffold, and this degradation pattern was found to be the same as that of the native PCl scaffold. So now the scaffold is ready. Now what we wanted to see is if we seed the mesenchymal stem cells on these scaffold surface, what will happen, like whether there'll be a bone regeneration or not. So for that, we have first what we did is we inoculated the mesenchymal stem cells, and we have seen whether the scaffold promote the growth attachment and growth of the mesenchymal stem cells. So here you can see the confocal images and electron microscopy. The day one, you can see like the electron microscopy, this is DAPI stain. The day one, you can see that there are very few nucleus you can see on the scaffold, whereas how the way we progress from day 1 to day 7, the number of nucleuses are increasing. Even if we see the scaffold, native scaffold and a modified scaffold, the modified scaffold showed more number of adhere cells on the surface than the native scaffold. Same was proved by electron microscopy. Further, we have performed the MTT assay. So this MTT assay basically tells us the metabolic activity of the cells, wherein MTT is reduced to a purple color formation compound by the NADPH secreted by endoplasmic reticulum of the active cells. So if we see, there is always an increase, this graph shows the increase in the MTT activity. That means cells are metabolically active, so from day 1 to day 7, cells are actively growing on the scaffold surface. And here we can see that the PCL scaffold, PCL GO and CQ modified scaffold shows the maximum number of cell proliferation than the native scaffold, which is PCL. Now, what we did is we exposed the scaffold the cells were seeded on the scaffold, and we observed the cells for continuously for 14 and 21 days. So after 41 days, we have started seeing that there is a bone, a calcium deposition has increased on the scaffold surface. So here, the A panel shows the plain PCL scaffold, and the C is our modified scaffold, whereas E is wherein the cells were seeded in tissue culture plate, and we have used the differentiation cocktail medium for their differentiation. So you can see there is a comparable calcium deposition is observed on both scaffold, which was not induced by chemical stimulation. It was formed only because of Ciscus quadrangulus, and the tissue culture plate, wherein we have added the differentiation cocktail. The same was observed, like after 21 days, this calcium deposition was increased in the number. The same was the case with tissue culture plate. So that shows that by the end of 21 days, like our scaffolds have successfully differentiated these stem cells to a bone lineage, and that too without any additional chemical stimuli. That means the graphene oxide and a plant, Ciscus quadrangulus, helps the differentiation of stem cells into osteogenic lineage. Further, we have quantified the alizarin S red stain, and we could see that the final scaffold, the layer by layer scaffold, have the maximum number of a differentiation into stem cells from bone lineage. So that proves that the scaffold which we have created can differentiate into bone lineage. Now, we have is a differentiation happens, but what about the in vivo case? Like, will this scaffold, when transplanted into animal, will it be able to differentiate into bone or recreate the bone? Because we have many times we have seen, like, cells or our experiments behave differently in a lab, and when we go to a bedside, the results are different. So just to prove the usefulness of these scaffolds, we have performed the animal study, wherein we have used the V-star rat, and these these are the different groups which we have created, and 5 animals per group we have used for the study purpose. So what we have done here is we have created a critical size defect of 8 mm on a rat skull, and this critical size defect were further used further covered by a scaffold with stem cells, like a final scaffold, PCL, graphene oxide, and Ciscus quadrigelis scaffold with stem cells on it. We have covered it. This defect was covered. So here you can see that the hole was created using dental burr, and the critical size defect was then covered using the scaffold. After this, like, animals were observed for continuously for 12 weeks, and after 12 weeks, during this observation, we have monitored their body weight, which was comparable to a sham control, which was non-surg like the animals in which the surgery was not happened. So it was comparable. In fact, the bone, the body weight was somewhat constant, or it was increasing in all the groups. After 12 weeks, we have sacrificed the animals, and all the organs were collected, and we have weighed these organs and observed that all essential organ weight was constant, and it was it was comparable to the normal group. Hematological parameters were seen, and again they were observed to be normal. Like we have seen the mean corpuscular hemoglobin level. We have checked at 12 weeks also, and 6 weeks also. Packed cell volume, neutrophil were observed. Again, the PCV and neutrophil counts was found to be in normal range. Lymphocytes were in normal range. Eosinophils, monocytes were in normal range. So all basically the body parameters were found to be normal. Like even the platelet counts, they are of normal range. So most of the hematological parameters like hemoglobin, RBCs, WBCs, platelets, neutrophils, eosinophils, MCV, PCV are all in normal range, even at the 6-week time, and even after 12 weeks of transplantation. That indicates that there is no severe inflammation in the animal happened, and all the animals were doing fine even after surgery. We have also seen some of the organ parameters like SGPT, SGOT was seen to check the liver activities, and the values were found to be in normal range. That confirms that the liver was healthy and normal, functioning normally. Even the kidney, for the kidney functions, we have studied the urea and creatinine, and again the values were in normal range, showing the functioning of the kidney is normal, and all the parameters, like a major organ parameters are of that of healthy animals. These are alkaline phosphatase studies which we have done to check the bone formation. We have seen that there is a higher alkaline phosphatase activity at the 12 week, indicating that the tissue healing is happening, and it's happening at a very good speed. Further, we have did the X-ray. Although X-ray could not show a good image because the tissue was very soft and the X-ray could penetrate, but you can see the clear a very light tissue formation at 6 weeks and 12 weeks of final scaffold. So what we have decided to go ahead with micro CT tomography, and here you can see that after 12 weeks, like you can see the sham control, the critical size defect after 6 weeks and 12 weeks was almost same. There was no proper healing. Even the normal PCL scaffold, there was no healing, but where we have used the PCL and mesenchymal stem cells, there was not much of a healing was seen. But if we see the scaffold where we have used the graphene oxide and a plant extract, Ciscus quadrigelis extract, and stem cells, was happening very fast. Like the critical size defect of 8 mm, which cannot heal on its own, started healing. Like the bone formation is quite substantial, showing that the scaffold was working in the animal. So this particular differentiated bone was then further, we have carried out the bone mineral density to see whether, the bone density is increasing from the sham level or from only PCL level to a PCL GOCQ mesenchymal stem cell, and it was seen that the bone mineral density is continuously increasing. That means there is a proper healing or new bone formation is happening at the transplantation level. Also, the histological analysis shows that new osteoblast and there are some fibroblast or osteocytes are developing in the transplanted bone region. So these are some of the histological analysis wherein you can see this is the final scaffold, where you can see that maximum number of osteoblast and osteoclasts are getting formed in the transplantation layer, showing that a good bone regeneration is happening with reference to PCL GOCQ mesenchymal stem cell scaffold. Now the animals study was functioning properly. Now it was a time to see whether this particular scaffold along with mesenchymal stem cells can heal the gingival recreation or gingival recession and give rise to a gingival recreation. To this, we have, we were doing, the clinical trial is still on. This is one patient wherein we have seen the very good results. This was the first patient which we have studied. So he was the 35-year-old male with Miller class 3 defect, a gingival recession. He had 6 to 8 mm of recession of gingival tissue on a mandibular central and lateral incision and left mandibular canis. This was a clinical problem, and the depth was even the soft tissue was disappeared, and this particular depth was somewhere around 2 to 3 millimeter in size. So a critical case of gingival recession. What we have done is we have taken this scaffold of size 1 centimeter square, seeded around 3 into 10 raised to 4 cells per ml per centimeter square area. And this cell seeded scaffold, after 12 hours of inoculation, it was taken, like cells were seeded on one surface, not both the surface. So this scaffold then was used for transplantation. What we have done is, here you can see we have placed this particular scaffold on the defect area. So this particular scaffold was placed, and then we sutured the defect. The scaffold was then stabilized by suture, and then the wound was closed. What happened is, here you can see the remarkable difference. Like just 2 months after post-operation, the gingiva was completely recreated, which was unable to heal almost for over 2 years. The the soft tissue was formed. Even the bone recession was recovered, and the person's gingiva was completely went back to a almost near normal gingival tissue. So this particular study shows that the Ciscus quadrangulus, when used in a PCL scaffold, can differentiate stem cells into a bone-like lineage. And further, if we use these scaffold for a tissue regeneration, this tissue, like gingival class 3, can in class 3, the soft bone regeneration happen very fast, and the recession can be avoided, and gingival recreation can happen. So this was the entire study what we have performed, and I'm happy to tell you that there are almost 10 patients are lined up in this particular clinical trials, and results are quite encouraging. So this is what the final outcome of my study. Just to brief you about the Advanced Cells group, like Advanced Cells is actually a group of 4 branches. Koshika, wherein we are producing primary cells for research purpose, though. So research institutions and CROs are our main client. Advanced Cells, wherein we are using or preparing stem cells from adipose tissue, bone marrow, and umbilical cord, dental pulp, and giving those tissues or cells to hospitals and clinics for therapeutic purpose. Youthful Me is is a very young branch wherein we are trying to create a stem cell based cosmetic for aesthetic purpose. Advanced Cells Biopredict is a new JV which we have formed recently with a French company called Biopredict. Now it's known as VPredict, and we are partnering with these companies. We are distributing their products in India and the Asian continent, and they are helping us to promote our products in European continent. So here you can find us. We are based in Noida. Thank you so much for your attention, and if there are any questions, I would like to take those questions now. Thank you so much.
Thank you, Dr. Sachin. It was quite interesting, and also really proved it in vivo also, and it was very successful. Whatever scaffolds which you have produced using MSc, it was quite successful in rectifying the gingival recession, and I hope this will be a very inspiring talk to many of our young researchers, faculty colleagues, and also other participants. And over to Dr. Satrupa for any questions from the participants. Dr. Satrupa?
Thank you, Dr. Kannapiran sir.
Yes,
and
yes,
so now we have 2 questions for Dr. Kadam. Munaza Rashid is asking, what are some facts about stem cells? What are the obstacles that must be overcome before the potential uses of stem cells in cell therapy will be realized? And why are doctors and scientists so excited about human embryonic stem cells? So this is the first question for you, sir. Hello, am I audible?
Yes, madam, you are audible. Hope you can repeat the question. It wasn't very clear. Could you please repeat?
Yeah, it was a very basic question basically.
Yes.
What are some facts about stem cells? And why are doctors and scientists so excited about human embryonic stem cells?
It's 11 hours.
And what are the obstacles that must be overcome before the potential uses of stem cells in cell therapy will be realized?
For the time being, Kadam sir has a very low band.
He's not around. He's not around.
Yeah. No, he's not in.
So suddenly maybe disconnected, sir. Maybe.
Yeah, he was disconnected suddenly. I think we can wait for a second or two.
Yeah, that's what
for him to join. I think he must be trying to rejoin.
Yes. Maybe some issue with the network.
If there is any more questions, all the participants may also shoot it.
Yeah,
I think
in the meantime.
Suresh sir can answer now. Question about the stem cells.
Yeah, Suresh.
What are the question again? What
are the stem cells and how it can be used by the scientists? It's a very basic question.
Question, sir. What are some facts about stem cell, like which already was discussed, and why are doctors and scientists so excited about human embryonic stem cells?
Yeah. Now, stem cells have the inherent capabilities of self-renewal, right? Making more copies of itself or themselves. And then under suitable conditions, they can be made to differentiate into cells of different types. For example, the mesenchymal cells that Dr. Anand Srivastava talked about this morning can be made to differentiate into cells of the bone lineage, cartilage lineage, or the adipocyte lineage. Okay, now excitement about embryonic stem cells is because that they represent one category of stem cells, right? But there are ethical issues and...
I'm sorry to interrupt, sir. Dr. Kadam is back to the chat room.
Dr. Kadam?
Yeah, I'm sorry,
Yeah,
I
please
had
continue.
some internet issue.
Yeah, we understand, sir. Very much understand. The first question was being answered by Dr. Suresh now. You
Oh,
can
thank you, sir.
you can take the next one also, or you can also add up to this question. Thank you.
Sure.
So should I ask you the first question or should I go to the next question?
No,
sir?
Professor Suresh, let him complete.
Yeah, sure.
Please, sir. Please continue, sir.
Yes,
And-
yes.
Yeah.
Talking about embryonic stem cells, it's a category of stem cells that are capable of being renewed in the laboratory and to differentiate. But there are some ethical issues associated with embryonic stem cells, safety issues. One is, are we destroying life? Are we playing? So are we allowed to tinker with Mother Nature? So those are some ethical issues. And when does life actually begin? So instead of embryonic stem cells, there's a lot of excitement with induced pluripotent stem cells, Yamanaka's work. So you have differentiated fibroblasts being made to de-differentiate into pluripotent stem cell-like cells. So that way we'll get around the ethical issues and also the rejection issues are taken care of. So the excitement is there with stem cells, but I think the world will start veering around more towards iPS-based technology. So it has remarkable potential in research and therapy, right? And also regenerative applications. Dr. Sachin, please go ahead.
Dr. Sachin, if you are able to hear, you would like to add up to this why people are excited on stem cells? Yes.
So, sure.
Embryonic stem cells. Most.
You just repeat the question because I missed that.
Dr. Saturupa, could you repeat that?
Yes, sir. Why are doctors and scientists so excited about human embryonic stem cells, sir?
Okay. See, like, stem cells, like right from we have categorized them into totipotent, multipotent. So if we are talking about the embryonic stem cells, as Professor Moti Kannan have said, like we have somewhere around 218 cells in our body. So these embryonic stem cells have a capacity or a capability to each and every type of cells, including germ cells. Okay, so an added advantage to embryonic stem cells. But when we are talking about the adult or a mesenchymal stem cells, in that case, what happens is these cells cannot differentiate into all possible lineages. Again, like as we grow old, like stem cells lose their characteristics, because although we say like when one daughter cells divide into two daughter cells, the exact number of chromosomes are being transferred, but it's not actually true. Like some amount of chromosome, like a telomere from the chromosome is, we have seen that it get lost somewhere. And as we grow old and as the telomeres, the size starts shrinking, the stem cells activity also stops. So adult stem cells, although they you can use them for your own body for therapeutic purpose, but they have some limitation. Whereas embryonic stem cells, they can grow enormously, but that enormous growth can also lead to a cancer formation, teratoma formation. So this could be a disadvantage with embryonic stem cells. Now, as Professor Suresh said, like, induced pluripotent stem cells, in fact, scientists are more keen to work on induced pluripotent stem cells now, because what is happening is by using fully differentiated cells like skin cells, you can give rise to a or you can program these fully differentiated cells to behave like a embryonic stem cells, and then you can ask these de-differentiated cells to differentiate into whatever possible lineage you want to work on. So this particular capacity or capability give added advantage because we can use our own body cells to create cells for therapeutic purpose. So if you see the research trend now, you will see that maximum labs are working on induced pluripotent stem cells than embryonic stem cells. So what I could say is the induced pluripotent stem cells are in trend. And if you see the Japan, they have already started, regularizing induced pluripotent stem cells for therapeutic purpose. Like there were few clinical trials happened successfully for eye surgeries, like some, I forgot the disease. What they have done is they have used these cells in older people, macular degeneration. Yeah. So they have seen that using these induced pluripotent stem cells, macular degeneration can stop and revert to a normal vision also. So induced pluripotent stem cells are gaining importance. So I hope I have answered the question.
Dr. Sachin, just to add
Yeah.
on the point about pluripotency, to satisfy the criteria, capability of forming the 3 germ layers, chimera, and the fact that it can form teratomas. I think Dr. Deepa Bhatia, who is going to present next, will be discussing more about the bona fide characteristics of stem cells and about the pluripotent nature and what criteria need to be satisfied to say that in fact the stem cell is a pluripotent stem cell.
Yes.
Thank you, Dr. Sachin.
Thank you, sir.
So next we have 2 more questions.
Okay.
One question is very interesting. How is the scaffold inoculated into human body? Asked by Shauna Srivastava.
How it's inoculated, I have shown in one of the figure, like, where there was an open area. What we have done is the cell facing scaffold side was placed on that open area, and then the rest of the tissue which was present, like that was sutured with the scaffold, so that scaffold will not move. And patient was, he was given some restriction, like after 2 weeks of when we removed the suture, the patient were given direction for oral hygiene, a proper brushing, like the circular brushing, and then this is how we try to maintain the scaffold intact at that particular area. And this particular scaffold, what we have observed is once it weight, like it just goes and stick to that particular surface, and it does not move. So even if we are talking about the bone regeneration and we want to see or use the scaffold on a bone, I really don't see the scaffold will move that much. But in case of oral cavity, there is lots of movement, like our lips were continuously touching to the gingiva area, saliva formation, we drink something, eat something, so there is a lots of movement. So just to make sure that scaffold remains on its place, we have sutured it. Otherwise, when we did the animal surgery, what we have observed is scaffold stuck to the bone surface as soon as we keep it on the defect. So there was no movement of the scaffold was observed even after the sacrificing animal. After say around 24, the scaffold was the partial degraded scaffold was still there on the surface. It didn't move. So same way we can use for bone surgeries in human also. So this is the way we can use the scaffold in human transplantation purposes.
Dr. Sachin, I have one question for the students about patenting. You mentioned the name of a plant which you have used, the CQ.
Yeah, Ciscus quadrangulus.
I'm assuming since you have patented it, you have added, PCL is known and this plant is known. So you would have added other components, or the stoichiometry would be different in order to go for a patent.
See, although the PCL is known and even the plant is known, but their combination and the process of creating the scaffold, basically, if you see, this is not a product patent, it's a process patent, like the way we have created scaffold and then the uses of this particular scaffold. So if you see, like Ciscus quadrangulus is known, stem cells are known, PCL is already been patented. So when we combine these three, we didn't interfere into existing patent, but we have modified the scaffold. We have used the plant extract into that which was not been used so far, and then created a scaffold. So the process in which we have created the product was patented in this case.
Yeah, thank you, Dr. Sachin. Over to Dr. Satarupa. Sorry to interrupt.
Yeah, thank you very much. Now the last question is that Anand is asking that speaker said rigorous brushing leads to oral inflammation and gum diseases. Which is the best way to brush teeth?
In circular motion, and if you use the there are various kinds of bristles available in the brush, like a soft bristle, medium, and hard. So generally we should avoid hard. For kids, it's always good to give them a soft bristle. For adult, we can use a medium bristles toothbrush. But if you have a sensitivity, it's again that you should go for a soft bristles. And when you brush, like rather than up and down movement, it's always good to brush in a circular movement. And brushing should not be you should not brush your teeth for more than 2 minutes. Like otherwise you tend to create hurt the gingiva in that particular process. So medium bristles, circular motion, 2 minutes brushing is good to go for not to hurt the gingiva.
Thank you very much, sir, for your answer and enlightening talk. Now I'm asking Dr. Debashish Mishra to take over the session.
Thank you, ma'am.
Thank you, Dr. Satyarupa. I would request Professor Kannabiran to give
Yes, sir. Yes, sir.
Thank you note to.
Yes, sir. Yes, sir. So I would like to thank Dr. Sachin for such an interesting and mind-blowing presentation. It's really very, very interesting.
Debashish, can you unmute? Can you mute Samantha? There's a.
Yes, sir. Yes, sir. Kora, sir. Kora, sir is I think is unmuted.
Okay.
That's the problem we are facing.
Thank you very much.
So I would like to thank on behalf of the organizers for accepting our invitations. And also, even though the Advanced Cell is the partner for this international conference, and I would like to thank you very much. So it will be very, very interesting, and it's a very, very inducing talk for many of the young scientists. So thank you very much, sir.
Thank you so much, sir. Thank you so much for inviting. Thank you.
Thank you, Dr. Sachin. I hope you'll be on for the next 2 days.
So over to Dr. Debashish Mishra.
Thank you very much. Thank you for your chairpersonship and moderation, Dr. Satyarupa and Dr. Kannabiran. Thank you very much for your time. I should thank Dr. Sachin Kadam for being a part of the conference, also being a resource person for this. I happened to meet you once in Dr. Amit's house, so hope you remember me. Thanks for this exciting and stimulating talk. We can take a short break to all the students. Hope you have had a very nice, interesting sessions all morning. We can take a short break. We can join within 10 minutes, 11:25. You can go and take a bit of break, go out and have some, get some water. Meanwhile, we'll try to set the slides for Dr. Bharatiya. She is already here for the next session. Okay, so we will start her session exactly at 11:25. Thank you very much, everybody, for being patient, and for the resource persons also. They were really helpful and really informative. I'll request Dr. Bharatiya. I'll welcome her once again. If she wishes to check her slides, we can do that. Deepa Bharatiya. She went out for Dr. Jashwal. Yeah, Dr. Bharatiya. Very good morning to you, madam. Great to have you here in VIT. You are muted, madam.
Yeah. Now you can hear.
Yeah.
Okay. So let me share my screen with you all.
Yeah. We would like to see that. Let's if it is working. Without any hitch.
So how do I do that?
You can see this green button, madam, below,
okay,
share
okay.
screen. Okay.
Dr. Devashish?
Yes, sir. Professor Suresh, yes.
Who is going to project the slide for the introduction? Shall I project?
Whoever is comfortable. We have Professor Amit, he has joined now. Dr. Godwin is also with us. If they are comfortable, if they want to project, because we have shared the slides for introduction. So
yeah, we would like to share the introductory slide for Dr. Bharti.
Can you see my slides now?
Yes. Let Dr.
Sure.
Dr.
Yeah.
Bhartiya finish up checking up. Would you
so
like to put it in the slideshow mode, madam?
Slideshow?
Yes, madam.
This way?
No. The other way.
Yeah, this way, no?
Yes. And would you like to scroll a bit, like, so that we can see what other slide? Yeah, it's all going smoothly, madam. Not a problem.
Okay.
You can choose to unshare now. We will put your introductory slide.
Sure. Let me.
A wonderful talk. I think I need to go back to the video.
Professor Suresh, you may consider coming closer to the microphone and speak. Your voice is very faint.
There is some background noise. Some children were talking in one of the.
Maybe, madam.
Okay, it's okay. That's all right. It's excellent. The audio and everything is very nice.
Thank you very much, madam. Thank you very much, madam. From your side also, it is very well. The video and audio is very good.
Yeah. Fine.
Dr. Devashish.
Yeah, Dr. Amit. You want me to share the slide?
Rights.
Acha. You are a co-host now, I think.
But I don't have sharing rights.
Wait. You are yet to be a co-host. Wait. You will have the sharing rights. Are you able to share?
Yes.
Dr.
Full screen mode?
I hope
yes.
Thank you.
Your meeting got over on time, I suppose.
Yeah, really, we should be thankful to Dr. Kadam to wind up very fast. Yes, it is really good. Dr. Deepa, how is everything in Mumbai, madam? Are you able to go to the lab?
Yeah, we are going every day. Today I'm working from home today.
Yes, it's really.
Hello.
Is all of your scholars coming to the lab, ma'am?
Yes, all of them.
That's really good. Because the transport and everything has suffered a lot.
Yeah. Some of them are there in the hostel only near the institute.
Okay, that is really great. They can come down.
And the trains are working for the government employees. Otherwise, train is not open to the common public, but for the employees, there is working.
That's really
Madam,
good.
Madam,
Yeah.
you're going to talk about pluripotency, the factors that need to be taken into account for a stem cell to be considered to be pluripotent.
Yeah, I really enjoyed the discussion in the earlier lecture. So I'll answer those questions.
Very good, ma'am. Thank you, ma'am.
No, it's very nice. The students are very alert.
Thank you, ma'am.
Yeah. So the whole thing is going very well. The inaugural plenary talk was also nice.
Thank you, madam. Thank you.
Thanks. I think sir is still there. Krishnamurthy sir is still there with us. Yeah.
Thank you, Dr. Deepa.
Yes, thank you, sir.
My students have read your papers. They have communicated.
Okay, yeah.
You had a tough time pushing your idea at one point.
Yes, I had. Yeah.
I hope you got over it, because it's very difficult to impress the people. See, they convince them that you have done the right experiment.
Yes, correct. It is still difficult, but we are strong.
Yeah, I loved your experiment, so I made my students read your paper. And one of them contacted you also.
I must have responded.
Yeah. That's what I'm saying. I've been following your work. Thank you so much.
Thank you, sir. Actually, for saying anything novel, the protocol has to be very robust. How to enrich those stem cells. That was the main thing, which we have now achieved.
Very good.
With 4 publications in 2020. Now we have achieved a very robust protocol. I'll discuss that.
Sure, lovely.
So my voice is clear, no?
Yes, ma'am. Very clear.
Okay.
So I think we can start with the introduction to ma'am. The students have rejoined, I guess. Yes.
Okay. Yeah.
Let me first introduce the chair and moderator.
Sure.
I would like to introduce. I would like to welcome all the students back to the session 3 lecture session, and I would like to welcome Dr. Deepa Bhartiya for finding time from our busy schedule and to give this fantastic lecture today. I would like to welcome the chair, Dr. Amit Jaiswal. He is also a tissue engineer, works in various aspects of regenerative medicines and biomaterial. I would like to welcome Dr. Godwin Christopher. He will be the moderator of the session. Over to Professor Jaiswal. Just basic rule, all the students should remember that they should attend minimum of 5 sessions to get the certificate. Second thing, they have to post their questions to our moderator, Dr. Christopher Godwin, so that our moderator will be able to ask all the questions to Dr. Bhartiya. Thank you very much. Over to Professor Jaiswal. Thanks.
Thank you, Dr. Devashish Mishra, for introduction. And on behalf of conference committee as well as VIT family, I would like to again welcome our esteemed speaker, Dr. Deepa Bhartiya. Dr. Deepa Bhartiya is Scientist G, Director Grade and Head of Stem Cell Biology Department at National Institute of Research in Reproductive Health and ICMR Institute, ICMR NIRRH, which is located in Lower Parel, Mumbai. She did her PhD from CDRI Lucknow, a CSIR lab, and then postdoctoral training in USA. There she was a Fogarty Fellow and later returned to Mumbai. She worked at BJ Wadia Hospital for Children and Reliance Life Science before taking a faculty position at ICMR NIRRH in 2003. She has 2 human embryonic stem cell lines, grown on human feeders to her credit, and did a lot of work on their differentiation into pancreatic and cardiac lineages. She carried out studies in mice using pancreatic progenitors. Later, her interest shifted towards pluripotent stem cells in adult tissues. She has more than 100 publications in peer-reviewed journals and associated with Mumbai University as guide for PhD in applied biology. For the knowledge of audience, I must say that Dr. Bhartiya is pioneer to work in the field of embryonic stem cell lines in the country. And then I also had a chance to meet her for a while when I was in Mumbai. So it is going to be very, fantastic and exciting talk for all the audience, including me. And I welcome once again Dr. Deepa Bhartiya. Over to you, ma'am.
Yeah. So is my screen visible now?
Yes, ma'am.
Okay, and you can hear me also.
Yeah.
Can you hear me?
Yes.
Yes.
Okay, thank you so much for the kind words. The title of my presentation is Pluripotent Stem Cells. And you may be surprised because instead of embryonic or iPS, I'm talking of pluripotent stem cells in adult tissues. But the lectures and the discussion earlier was very stimulating. And maybe the very first slide gives the answer to the question raised by one of the students. So the question is that what is the current status of stem cell research? I agree totally. There was a lot of excitement about embryonic stem cells and pluripotent stem cells, embryonic and induced pluripotent stem cells. And the scientists are very much attracted by these stem cells. But the thing is that what has happened to them? Are they of any use for regenerative medicine? Actually, to tell you the truth, the field is stuck. Because the reason is that both embryonic and induced pluripotent stem cells have failed to deliver, because when you differentiate them, they don't make their adult counterparts. They don't make their adult counterparts, and thus generally you need to regenerate tissues in adult people. So these cells are actually falling short because they are not able to differentiate into the adult counterparts. So initially about 7-8 years back, what they tried was, okay, we can transplant immature cells in the body and they may mature within the body when they are given the adult niche. But that has also not happened. For example, for pancreas and diabetes, they were transplanting immature progenitors in the body packed in an immunoisolatory device, and they thought that it will help. The proof of concept is there that the stem cells can help. But these stem cells are not able to sustain the beneficial effect for a long time, both pluripotent embryonic and induced. For example, one of the first application everybody thought with these cells was to make gametes, but it has not happened. You are not able to differentiate these cells into sperm and eggs for use by the infertile couples. So where is the whole thing falling short? The whole thing is falling short because they are not able to turn into primordial germ cells, into progenitors, which are the natural precursors for gametes. So there is something inhibiting these stem cells to make mature cell types. And side by side, when there was so much of excitement with embryonic stem cells, iPS or embryonic stem cells, there was a group, very strong group, which said, okay, we don't need embryonic stem cells because life is an issue and ethical issues. So we think that hematopoietic stem cells have the potential. They have the plasticity to differentiate into all types of cells, and they can be used instead of these stem cells. And there are so many trials which have been done over the last 1 decade, but everything seems to have failed. the hematopoietic stem cells, when you transplant, is good only for bone marrow transplantation. They are only able to regenerate and reconstitute hematopoiesis and some immune cells. But if you transplant hematopoietic stem cells into the kidney or into the spine, here and there, these stem cells don't have the potential to transdifferentiate into different cell types. As a result, now ICMR has it written on their website that cord blood banking is not a future use for health. It will not treat 80 odd diseases. It is only for blood diseases. So cord blood banking and autologous bone marrow cell trials, they are only for hematological diseases, not for anything else. So transdifferentiation and future health, all those promises were wrong, have proved wrong. So what are hematopoietic stem cells actually? They are adult stem cells, and I would actually classify them as progenitors. They are specific lineage restricted progenitors which can only differentiate into blood cells. So as you see from this slide, and now what has happened since the embryonic stem cells did not differentiate into their adult counterparts for regenerative medicine, you know what a scientist does? Finds an alternate area for research. So the whole thing, you know, the present focus is on making embryos. So they made a brain in the vitro in culture, and so many different kind of embryos can be made. And it's a very in thing right now to study embryos. I'll come to this later on also. So how do you define an adult stem cell? So now I'll come to the different definition here itself. So in case of adults, what is a stem cell? They are expected to be quiescent in nature. They are less in numbers. And a stem cell is supposed to undergo asymmetrical cell division, whereby it can self-renew, divide to keep its numbers same, and also give rise to a progenitor by undergoing asymmetrical cell division. Asymmetrical, I mean, the small stem cell will give rise to slightly bigger progenitor. Therefore, it is asymmetrical division. And these progenitors, they have the property to divide more rapidly. They undergo symmetrical rapid divisions and clonal expansion. Clonal expansion is a very interesting term, and it is rapid division with incomplete cytokinesis to form spheres. And once this you get, then they undergo further differentiation and into different cell types where these cells are sitting. So this is how the lifelong tissue homeostasis in the body organs is maintained by the adult stem cells. But Abhi, just now I told you that hematopoietic stem cells actually are the progenitors. They are not the stem cells per se. So back in 2010, there was a paper in Science that all adult tissues have two stem cell populations. One population is quiescent and the other is active. Similarly, a paper came in Nature also that our adult body tissues have two types of stem cells, dormant and restless, meaning quiescent and actively dividing. So this was the concept, and different types of tissues in the body, they were saying that these two population of stem cells exist in adult tissues. But what has happened over the time? Now in 2019, 15, 20, now this year also, these people, this person, Hans Clevers, he is an architect. He had published this paper in 2010 in Science. Now he says, because it is proved very difficult for these people to isolate the quiescent stem cells from different adult tissues. So what he says that possibly not been able to isolate these stem cells as a physical entity. So he says that we, instead of quiescent stem cells, maybe they don't exist in adult tissues. It is better to define stem cells based on their function rather than as a physical entity. And there is a strong belief that instead of stem cells in adult tissues, these somatic cells, the mature cells, undergo de-differentiation to behave like stem cells whenever the body needs. So whenever there is a very huge damage to any tissue, the somatic cells there de-differentiate and become stem-like cells to ensure regeneration. So this is what I'm saying, that they are now suggesting in 2020 that we need to redefine these stem cells simply on the basis that they have the ability to replace the, so there's a whole confusion. What is an adult stem cell? And they are saying that they may possibly do not exist, the quiescent stem cells. So there is this story I like to share with you about the prostate. In 2020, there's a paper published in Science. And what is prostate, as you know, is part of the male urogenital system. And it has been studied since long. And what they observed that if you do castration, remove both the testes, the prostate undergoes atrophy and regression. It totally shrinks. But if you give back the androgens, it can regenerate back to the normal. So they believe that the prostate has stem cells. This has been believed since long that it does have stem cells. But this recent technology of single cell RNA-seq, it was performed on the prostate in mice and humans both, and the data was published in Science in 2020, and they found no evidence of stem cells. All the mature cells they found in the prostate, but no stem cells. And they came out very strongly in this paper by saying that there are no stem cells in adult prostate, there is a concept of dedifferentiation, and maybe stem cells don't exist in adult prostate. Similarly, there was a paper in Nature, which also said from Sloan Kettering, this is from Sloan Kettering regarding that pancreas also doesn't have stem cells. There was a paper published in Nature Communications from which again concluded by single cell RNA-seq that there are no stem cells in the ovary. And it was reviewed by Deepak Srivastava and Christina Mamre. So these three people are very great people in the field of stem cells because all the three have remained as presidents of ISSCR. She is the current president. So it was like a full stop to the field of adult stem cells. And this cardiac paper in PNAS, by using very sophisticated techniques, said there are no stem cells in the heart also. So what is happening? So, but I have got, and I immediately realized what the problem was. So we published a paper in response to the Nature Communications. How is it possible that single cell RNA-seq is not picking up stem cells? Because I can pick them up very simply. You take a sheep ovary, bring it from a slaughterhouse, scrape it under a microscope, and you see the stem cells. See these stem cells? These small darkly stained cells, these are the stem cells. And these pale stained cells, these are the epithelial cells, the somatic mature cells. So you know what is the mistake the scientific community is doing? They don't understand the difference between a stem cell and a mature cell. A mature cell has lot of cytoplasm, lots of cytoplasmic organelles, which are required for it to remain active metabolically. Whereas a stem cell, as you heard me earlier, is very, very quiescent. It remains quiescent, doesn't have so much of cytoplasm at all, and has just one or two mitochondria. Those also are spherical because these are inactive cells. So just imagine if you have these two cells in a bucket together and you process it for single cell RNA-seq, what has been happening is that you discard the stem cells in the process of processing. They get unknowingly discarded. So a single cell RNA-seq is actually being done only on these cells. And that is why these papers published in Nature Biotech and Science from prostate, they have reported negative results. That is when the paper was published in Science also, I wrote a letter why the stem cells remained elusive during single cell RNA-seq of prostate. So I had written the reply and I had tried to explain them that the scientific community need to understand that a stem cell is very different from a mature cell, and you can't put both the cells in the same basket for processing. But this is where so much of confusion, and everybody is unknowingly discarding the stem cells from the adult tissues, and they are saying there are no stem cells. That's why the whole controversy. So what I am suggesting is that there are pluripotent stem cells in adult tissues, and these stem cells have been studied by different groups. And they have been labeled differently by different groups. Some of them call them VSCLs, some call them Muse cells, MAPCs, spore-like cells. So in my today's presentation, we'll be using the term VSELs, very small embryonic-like stem cells. Yeah, VSELs. And these VSELs are present in all the adult tissues of the body. And what is their function? They maintain lifelong tissue homeostasis. All the organs are working normally, and normal homeostasis is maintained in all the organs because of the functioning of VSELs. And whenever there is any kind of stress or trauma to the tissue, these cells become active and ensure regeneration. They have a lot of role in the process of aging, why diabetes occurs with increased age, why cancer incidence is increased with age. And also it is these stem cells only which get transformed into cancer stem cells. Because you must understand and think very clearly that if a person gets cancer and stem cells have a role, a new stem cell will not come from anywhere. It is the tissue resident stem cells only that become converted into cancer stem cell. And one very strong evidence to support that is these VSELs in normal tissues, they express OCT4. It is a pluripotent marker, OCT4. And cancers affecting different organs in the body. Any organ you think of, any type of cancer, they express embryonic markers. They express embryonic markers, including OCT4. So what is cancer? Cancer is uncontrolled expansion of VSELs. I'll come to that later. And I've got a lot of students working in this field of research. And we have been working in this area for almost 10 years now. So, yeah, I'll come to the definition, how to define a pluripotent stem cell. I'll come to it in a minute. So what are the VSELs? And they were reported for the first time in 2006, when the induced pluripotent stem cells were also reported first time in 2006. So what are these stem cells? You know, during development, there are primordial germ cells that appear. They migrate along the dorsal mesentery and sit in the two gonadal ridges where they'll make the germ cells. And in the process of this, it is thought that when a baby is born, there are no more primordial germ cells. They are all lost. They are all differentiated. However, this person, Professor Radzak from the University of Louisville, he showed in 2006 that rather than moving only to the gonads, these stem cells actually migrate and settle down in all the organs of the body, in all the adult tissues. They are sitting, and they are pluripotent in nature. They become very small in size, and they survive in all the adult tissues in small numbers. And the primordial germ cells, as I told you here, they don't just disappear with development. A small set of these stem cells survive in all the adult tissues, including the testis and ovary. So even in the testis and ovary in an adult human being, these stem cells are present in small numbers. But how to enrich them, how to study them, that is a big question. And back in 2006, 2009, he showed that if you take a mouse, different organs of the mouse, and study them for VSELs, they can be picked up. VSELs can be picked up in all the different tissues. Also, of course, they vary in numbers in different organs. So basically, it is the primordial germ cells that survive in few numbers in all the adult tissues as pluripotent stem cells for their important function during adult life. So as of today, almost 30 groups, these cells are controversial because whether they really exist or not exist is the big question. However, by this year, there was a review article written last year that almost 30 independent groups have now confirmed the presence of VSELs in adult tissues. They are the most primitive stem cells in adult tissues. And why they become important? Because compared to human embryonic stem cells and iPS cells, which I told you in the beginning differentiate into their fetal counterparts, the VSELs, they differentiate into their adult counterparts. They differentiate into adult cells because they are sitting in the adult tissues. They differentiate into adult cells. They are of autologous source. So no immunological rejection or anything, and no risk of formation of teratoma. So as you all had a discussion earlier that how do you define a stem cell? Pluripotent stem cell. So what is the basic definition of a pluripotent stem cell? They should express pluripotent markers, including OCT4, SOX2, NANOG. They should have the ability to differentiate into 3 germ layers. In vitro, people have shown. And also in vivo, by making embryonic bodies, and in that you should show the 3 germ layers. And they should also form a teratoma when you transplant in a SCID mice, or they should incorporate in a developing embryo by forming a chimera. So these are the basic properties of a pluripotent stem cell, embryonic stem cell, and iPS. But a VSCL differs from these ES and iPS. Just imagine if the VSCLs in our body were active throughout life. They were actively dividing rather than remaining quiescent. Then our body will burst tumors all the time. We'll have tumors throughout the body all the time. So what has Mother Nature done? Mother Nature has kept these cells very quiescent. They are very, very quiescent. Very rarely they undergo asymmetrical cell division. And as a result, these stem cells, if you transplant in a mice, they don't form teratoma. If you take VSCLs, put in a culture, they don't actively divide. However, they can differentiate. So, and neither do they form chimera. So this is what sets them apart from iPS cells and embryonic stem cells, which also do these properties, whereas VSCLs only express pluripotent markers and can differentiate into 3 germ layers. But they don't do this. So the work that we have done already, which I may not be covering today, is like if you take VSCL from a testis or the ovary, let's say after chemoablation, you treat the mice with busulfan, the whole testis or ovary gets totally chemoablated, take out the VSCLs and put in culture. They immediately make egg and sperm. Whereas iPS and human embryonic stem cells are not able to make gametes. Why? Because VSCLs are equivalent to PGCs. So if you have a PGC in culture, it is a natural precursor for gametes. So spontaneously you'll make gametes, but human embryonic and iPS cells, making primordial germ cells from them has proved extremely difficult, because you can control gene expression in a Petri dish in culture, but how can you regulate their epigenetic profile? That is not, it's very difficult to make primordial germ cells from human embryonic and iPS cells. But VSCLs being equivalent developmentally to primordial germ cells, if you put in culture, they make sperm and eggs spontaneously. The data is so good to be true that people in the field do not even believe that what we are talking is correct. There's a lot of disbelief in the field. And of course, this I told you that they get transformed into cancer stem cells. So what happens is these VSCLs exist in very few numbers in adult tissues. But if there is any kind of trauma, they rapidly increase in numbers. So they increase in numbers in case of cancer also. Now we have data in the lab. If there is a cancer, they increase 10 to 12-fold more. But if there is some kind of trauma, like for example, if we treat a mice with busulfan, the testis becomes totally non-functional. All the germ cells are lost. So VSCLs double in number. They double in number. Why? Because they want to restore homeostasis in the testis. Or same thing happens in the ovary. If you treat with cyclophosphamide and busulfan, the whole ovary becomes non-functional and VSCLs become activated because they want to restore homeostasis. 2-fold increase. We have published all this data. They increase 2 folds, but they are not able to restore homeostasis. I'll come to that in a minute. Whereas if there is cancer, the changes in the VSCLs are very different and they will increase 10 to 12 folds. I'll come to that also. So basically these VSCLs, as I told you, they're very small in size. So they have remained controversial. So in 2012, you know that iPS cells got the Nobel Prize in 2012. And what happened to VSCLs? In 2013, people challenged their existence. Why? Based on flow cytometry data. This group in Stanford, Professor Wiseman's group in Stanford, he ordered the same machine like Professor Radzak and performed the functions in his lab, and he could not replicate the results. He was not able to detect VSELs. So this negative data in 2013 produced a lot of negativity in the field. So that is the reason the IPS got the Nobel Prize and are very attractive stem cell candidates even today. But VSELs, their very existence was doubted in 2013. So the Radzak's group gave a lot of explanations and technical shortcomings were pointed out, etc. But the controversy was very huge and a lot of negativity came to the field. I have also gone through very tough times because of this controversy, but our group has stood strong. Why? Because we believe in what we see under a microscope. If you can see a stem cell under the microscope, you know they are there, isn't it? And we continued working on these cells. So ICMR had set up a committee also to validate our protocols. Yeah, we have gone through, and extramural funding was stopped to my lab. And that I will tell you that embryonic stem cell research is very, very expensive. You need so many growth factors and cytokines, so much culture media and all that to maintain embryonic stem cell culture and to differentiate them. Whereas VSEL research is relatively much less financially expensive. And you need to do more of biology with VSELs. And we were very happy that despite less of funds, we were able to do good research. So this paper, this is something very interesting you should understand. We published this in 2012, that if you take cord blood or you take bone marrow or peripheral blood, you all know about density gradient centrifugation. So if you do density gradient centrifugation, the whole world works on this Buffy code, isn't it? The stem cells are thought to be here. But what we had published, that the VSCs, the pluripotent stem cells with regenerative potential, are actually sitting here with the RBCs. They were here. So if you take this RBC pellet and you lyse all the red blood cells, then make a pellet and do RT-PCR, you will get all the pluripotent markers. So the VSCs actually sit here. They are not here in the Buffy code. So this we published. So you know what was happening? All the cord blood banking, autologous mononuclear cell trials with HSCs, bone marrow cell trials, etc. They have all focused on this. These cells have been evidently discarded in all those clinical studies. So till now, the clinical potential of VSELs has not really been tested in a lab or in a clinical study. So my student, I had a PhD student at that time. She published a paper in leukemia, which has impact factor more than 12. And what she did, she took cord blood, double diluted it, did density gradient centrifugation. And then the whole paper, you won't believe, she has worked on the red blood cell pellet, RBC pellet. She finally lyses the RBCs and she gets a pellet here, which are the pluripotent stem cells. See, this is how many, so many cells are there in this. And she has done a lot of immunolocalization on these cells, flow cytometry. She shows that these cells are diploid. They are not in the active cell cycle stage. They are in the G0, G1 phase, not actively dividing. And they express nuclear OCT4 and primordial germ cell specific fragilis, etc. So a lot of characterization, and we also look at the ploidy status, etc. They are diploid. And she did flow cytometry to study these cells, etc. So we got the paper in leukemia. After working on the blood sample, then we started working on adult tissues. Like, you know, we are working in a reproductive health institute where we need to work on reproductive tissues. So like testis, ovary, uterus, etc. So let's say if you have a solid tissue, you do enzymatic digestion to get a single cell suspension. So like this, you get so many cells. So what is happening? The mature cells have a lot of cytoplasm, lot of cytoplasmic organelles. Therefore, generally, all the scientists globally, they try to pellet at a low speed. Their cells are pelleted at a very low speed in order to get all these somatic cells intact. And their cytoplasm should remain intact. Their cell surface profile should not get affected. So they pellet at a very low speed, like let's say 1000 RPM. So then you get a big pellet, 1000 RPM, and this is how the cells look. But you know, when you do this, what happens? The stem cells don't come down. They don't pellet down. They remain floating. And what does the global scientific community do? They just throw away the supernatant. So, but we realize that the stem cells are buoyant. They are floating here. And if you take the supernatant and spin at 3000 RPM, instead of 1000, 3000, you can make a pellet of these stem cells. And this is how they look, small stem cells, this. And they all have nuclei, DAPI stain, and they have cell surface scar. Scar one is there. So the stem cells are actually here, and you just discard them. So a person published a paper in Nature and JCI. He was studying stem cells in pancreas. And he made a single cell suspension. And for the whole flow cytometry, he used a speed of 1200 RPM. And he said at the end, there are no stem cells in the pancreas. Paper published in such a reputed journal. But why he got no stem cells? Because he discarded them in the process. This is the same thing. You take any solid tissue you take, if you will spin at 800 to 1200 RPM, you will get the somatic mature cells. But the stem cells remain suspended, and you can get them by spinning at a higher speed. And how the stem cells look different? They are darkly stained because they have a lot of euchromatin. They are darkly stained, spherical, with very less pink color. Pink color is for the cytoplasm. They have minimal cytoplasm. And so we had patented this finding of ours. This is what I want to show you about symmetrical and asymmetrical cell division. See this, like for example, yes, one small cell, one bigger. One small cell, one bigger. This is asymmetrical cell division, where a VACL is giving rise to a progenitor. These progenitors divide rapidly by symmetrical cell divisions, and they undergo clonal expansion. So look at these spheres. What are clonal expansion? Where cytoplasm doesn't divide, nucleus divide rapidly to form clonal expansion. So these are from the uterus, clonal expansion, and after this, they will undergo differentiation to differentiate into uterine epithelial cells, myometrial cells, etc. So when we were publishing this paper in Stem Cells and Development, the reviewer said that why don't you show us pictures at higher mag, 100x. So we did that. It is very nice. You see the cytoplasmic continuity between a small cell and bigger cell. This is asymmetrical division. This is symmetrical division. And look at the cytoplasmic continuity here. These are clonal expansion. And we also looked at the expression of NUM, OCT4 and NUM. So stem cells, both the cells express OCT4, but NUM is exclusively expressed by the differentiated cells. Only after doing all this work, we could publish this in Stem Cells and Development. That there are two populations of stem cells in adult tissues, the smaller quiescent VSCLs and the bigger tissue-specific progenitors. So now let's come back to the definition once again. The hallmark feature of stem cell is asymmetrical cell division. Stem cell undergoes asymmetrical cell division. The progenitor undergoes symmetrical cell division and clonal expansion before further differentiation. So this is how a stem cell gets defined in adult tissues. So last year we published this, that using the simple protocol of different speeds for centrifugation, we can isolate adult stem cells, VSCLs, from the pancreas, from the spleen, from the lungs, from the gut, from the bone marrow, testis, uterus, gut again, and from the cardiac tissue also. Okay, so now I'll just give you one or two examples. Like for example, in the bone marrow, we can isolate these stem cells, purify them, and we can study their differentiation in culture. So it is a pluripotent stem cell, isn't it? So it is expected to give rise to all the three germ layers. And that is what we found, that these stem cells differentiate into ectoderm, mesoderm, and endoderm. And so all the three germ layers we got, we have characterized them also. And interestingly, we also found these stem cells from the bone marrow. If you put on a feeder layer, like a Sertoli cell bed, they make male germ cells also. So bone marrow stem cells, bone marrow pluripotent VSCLs can also differentiate into germ cells. And all this work is published here. And something very interesting we got was that if these VSCLs, they are CD45 negative. Yeah, so initial culture, 99% cells are CD45 negative. But if you put them in culture for hematopoietic stem cell differentiation, we find that over a period of 2 weeks, almost 20% of these cells become CD45 positive. What does that mean? That these VSCLs in the bone marrow also differentiate and give rise to hematopoietic stem cells, hematopoietic progenitors in culture. So this is in bone marrow, we picked up asymmetrical division, symmetrical division, and clonal expansion. So this is a property of stem cell in all the tissues. In all tissues, you must get asymmetrical division, symmetrical, and clonal expansion. And we have also studied the NUMB, selective expression of NUMB in the differentiating cell while both the cells express OCT4. So all this is published. So I will not share all the work that we have done in different organs, just one more organ, that is the testis. So this work we had published in 2010 using human testis. At that time, you won't believe we did not have the antibody for OCT4 in my lab. So we borrowed it from our neighboring colleague lab, OCT4 antibody, and we did immunolocalization on testicular cell smears. Something very interesting. We found these small cells having nuclear OCT4. See, these two with arrow. And they were bigger cells with cytoplasmic OCT4. See this? Smaller cell with nuclear OCT4, bigger cell with cytoplasmic. This was the basic foundation for all the work that I have done in my lab till now. What do you see? Our students spent almost 2 months trying to get rid of the cytoplasmic because we thought it was all non-specific. But this is very, very specific, and there's a very beautiful biology which is shown in this slide, that a pluripotent stem cell expresses nuclear OCT4, and when it starts differentiating, the nuclear OCT4 is no longer required, shifts to the cytoplasm, and as this cell will differentiate further, the cytoplasmic OCT4 also gets degraded and disappears. So then after working on the smear and the section, then we looked at different sections for OCT4 expansion, OCT4 expression. See symmetrical division you see here. And here I could see a whole chain. See, in a section how the clonal expansion looks, you get a chain there. And we looked at markers by in different testicular sections. There was OCT4, NANOG, etc. When you do qRT-PCR, so basically what I'm showing here, that there are 2 alternatively spliced isoforms of OCT4. This is called OCT4A, which is nuclear, which is specific for pluripotent state, and there is cytoplasmic. So majority of cells have OCT4 in the cytoplasm. Why? Because the progenitors are much larger in numbers. So the cytoplasmic OCT4 is much more, whereas the pluripotent OCT4A is very, very minimal. But it is this OCT4 which is responsible for pluripotency and for VSCLs. Then we tested the presence of OCT4A isoforms by different antibodies, and this paper got published. So this was the basic first paper. So then I have told you that 2013, there was a big controversy regarding these stem cells and flow cytometry. So this year we now published a very simple protocol, very robust protocol for people to believe in VSCLs. So you take the testis, do enzymatic digestion, then you filter the cells, total cells. Okay, then you spin at 1000 RPM. So you'll get the pellet here. These are the big cells, mature cells, etc. Take the supernatant and spin at 3000 RPM. You get a pellet. These are the VSCLs. This pellet here, you can also further filter overnight on a 5 micron cell insert so that you get pure population, not pure, but cells smaller than 5 microns, and which are most of them are VSCLs. A simple and robust protocol to enrich VSCLs from adult mouse testis. So this is how the stem cells look under a H&E in a smear. And then you can do RT-PCR for different pluripotent markers. This is how. So see these small cells, which are the VSCLs, and bigger ones are the SSCs, spermatogonial stem cells, higher nitrogen. So if you look at this VSCL, it has minimal cytoplasm. That is the property of a stem cell. Minimal cytoplasm and quiescent in nature. And because the chromatin is totally euchromatin of a pluripotent stem cell, that is why it stains so dark. So now I had discussed that we'll discuss the flow cytometry for this, how to study these stem cells by flow cytometry. So earlier on, my last set of PhD students, when we were trying to study VSCLs by flow, what we used to do, we used to study by flow in the whole testicular cells. You take testis, I'll show you. Here, you take these cells, filter, so you get total cells. So we used to do flow cytometry on this population. So what was happening, that the number of cells was very, very less, 0.001%. Yeah, so when we used to do flow, we used to get 1 event, 5 events, 6 events. So when this validation committee people came, they were not convinced by this data, seeing only 5 or 6 events. They said, show us 50 events. So now what we did this year, the data that we published, we separated this pellet here and this pellet here. And now we subject these two pellets to flow cytometry. And it has made a lot of difference for us. So we take cells, we take singlets, cells in the size range of 2 to 6 microns, and we work on viable cells, which are 7-AAT negative. They have to be Lin negative, CD45 negative, SCA1 positive. 0.03% in pellet A, the one that we spin at 1000 RPM. And see in pellet B, we get 0.3%. And look at the numbers. They have increased 10 folds. And for testis especially, we study 5 lakh events, and we get large number of events which are VSCL specific, and we have become quite strong with our data. So these stem cells can be then characterized for different markers. Yeah, like pluripotent markers, whatever marker. Once you have a very enriched population of these cells, you can do all kinds of studies. You can do RT-PCR, you can do immunolocalization, you can do flow cytometry. And something very interesting that we found, that these stem cells express estrogen receptors. They express estrogen receptors alpha and beta. So what happens? Endocrine disruption. There are so much talk now about endocrine disruption affecting reproductive health. So we think that it is the stem cells. I'll show you. Yeah. This paper we have published this year. This is all the work done on mice. So if you take a mice and treat with, let's say, diethylstilbestrol when the mouse pups are there at this small stage. So day 5, we treat with diethylstilbestrol. And when they become aged, there is infertility, spermatogenesis is affected. So they are infertile, sperm count is reduced. And something surprising, that almost 9 out of 10 mice developed cancer. So we had a wonderful study model where we could study stem cell biology. So what we found, that if you treat with endocrine disruption, there is block differentiation resulting in infertility, and there is excessive self-renewal causing testicular cancer. So this work we have published this year, and it was appreciated, and it has also come as the cover page for the journal, that it is the VSCLs which are involved in maintaining normal fertility, and their block differentiation causes infertility, etc. And excessive self-renewal of VSCLs initiates testicular cancer. And then my student now, she's in the process of doing OCT4 expression and SSCA1 in the testicular tumor sections. This data is not yet published. Similarly, in the pancreas, we have found like Nature, there's a paper in Nature by the most expert in the field of pancreas that there are no stem cells. But we show that if you spin at 3000 RPM, there are so many stem cells in the pancreas, and this is at 1000 RPM. And you take those pancreatic stem cells, you can characterize them, You can study for pluripotent markers, and you can do all kinds of work. Study them by flow cytometry. So I'm coming towards the end of my talk. So what is the potential of these VSCRs? So I have told you that they spontaneously differentiate into gametes because they are equivalent to PGCs. My papers are there. The other thing is that being quiescent in nature, you know what we have found, that they survive in the testis. They are quiescent. So whenever you treat with any cancer drug, being quiescent, they survive oncotherapy, both in the ovary and the testis. This data we have in mice as well as in humans, that VSCRs in the gonad survive oncotherapy. So if they survive oncotherapy and they exist, later on we can manipulate them to regenerate the damaged gonads, restoration of fertility in cancer survivors. Also, they can be targeted to treat diabetes because these endogenous VSCRs can regenerate the pancreas, and cancer initiation, etc. we need to study. So since morning, you all had 2 lectures on mesenchymal stem cells. So firstly, I would like to share that they are not stem cells. The mesenchymal stem cells are actually the stromal cells. There has been a lot of debate. They are stromal cells. They are multipotent in nature because they can differentiate into 3 types of like adipose cells, etc. But they are not pluripotent. They can't give rise. They don't have that plasticity. But I understand that almost 1,000 clinical studies have been done using mesenchymal cells. They are basically paracrine providers. They provide growth factor and cytokines. To whom? To the endogenous tissue resident VSCRs. VSCRs and the tissue specific progenitors are sitting in the damaged tissue. You transplant mesenchymal cells, they restore the niche and regeneration occurs. The regeneration occurs from the VSCRs. They will regenerate. And what is the function of mesenchymal cells? They only rejuvenate. They rejuvenate, whereas VSCRs regenerate. So MSCs are paracrine providers, whereas VSCRs bring about regeneration. So you have to think of both of these cells together because the VSCRs are the stem cells and MSCs provide the niche for the stem cells to work. So the stem cells proliferation, differentiation, everything is controlled by the niche. That's why I put here, you know, the shakand here is between VSCRs and the mesenchymal cells. So all the trials with mesenchymal cells have succeeded. There is a lot of potential. Yeah, you heard 2 lectures since morning. But why the mesenchymal cells, whether being transplanted as such or on sheets, if you transplant, or even their secretory products like exosomes or microvesicles of mesenchymal cells also provide benefit. Why? Because they go and act on the endogenous VSCRs, which bring about regeneration. So my group thinks about COVID in a very different way. What is COVID infection actually? What is happening in the lungs? The virus is going and destroying the lung epithelial cells. So in young individuals, the destroyed epithelial cells are immediately restored from the VSCRs differentiate rapidly and they replace the damaged epithelial cells. In young people, that potential to regenerate is very good. But in adult people, especially who are also having diabetes, etc., this function is compromised. Cells are not able to differentiate as efficiently as in young individuals. That is why mesenchymal cell trials are showing promise. Because if you transplant mesenchymal cells, the endogenous VSCRs get activated and ensure regeneration. Yes? So this is what we think, that actually it is all to do with regeneration and stem cells. So I like to stop here and thank you all. Thanks so much. I'll take questions.
Thank you, Dr. Deepa Bhartiya, for the fantastic talk and the new insights into stem cell biology, particularly in the pluripotent stem cells. And I must congratulate you for the kind of work you have been doing since 10 years and then showing, being the torchbearer in this field in the country and in the whole world. So are there any questions from the audience, from the students and the participants?
Now I would request Dr. Godwin to present.
Yes, sir.
A lot of students are excited about the findings.
Yeah, certainly. Amazing.
Because so much basic research can be done now with this understanding. How cancer initiates, what are cancer stem cells.
You have shown us a new cave, a new Atal tunnel. We have to explore that.
Yes. And I am open to collaborations with anybody, whoever wants to work with me.
Thank you, man. Thanks. Yes.
Okay, sir. I'm just going through a few questions. The first question, I am Prashant Rajan, studying second year BTech Biotechnology at Karunya University, Coimbatore. I was very interested. It was a very interesting talk. We learned a lot new about this new information. I want to know, can we collect VSELS from blood? Can we donate VSELS instead of blood donation?
So basically we can collect VSELs from bone marrow, from peripheral blood, even from cord blood. But what I basically think that there is no need to transplant VSELs because they are there endogenously. What you need to transplant is the mesenchymal cells, the niche, because the niche is damaged. If you improve the niche, you can restore fertility in cancer survivors and so many different, even hematopoiesis after bone marrow, after you treat with radiotherapy and oncotherapy to deplete the earlier ones. You transplant mesenchymal cells and you will get restored hematopoiesis and even testicular function, ovarian function, everything can be restored by MSCs. But you know what is happening? The scientific community, MSC is doing all this. How is it possible MSC can do so much? So there is a lot of sarcasm in the field. People don't believe in this potential of MSCs. So basically now it is time that we think of MSCs and VSELs together. MSCs are rejuvenating, VSELs are regenerating, but they work hand in hand. So that is what that is how we can explain and take the field forward.
Thank you, madam. I hope that Mr. Prashant has received the answer for his question. Next question from researchers have created from a body cell. How these experiments are done. The second part is image of mice with the human ear is very common image on magazines. As you have done a lot of research, we want to know how these organs were grown in mice. Can we grow organs in or on mice in India using the present ELITE facilities?
No. Those figures also, they are more of hypothetical. Whatever figures are there, that it is possible to do that, grow a human ear in mice, and it's all hypothetical. But actually, nobody has grown a mature ear on a mouse, or at least that is what I think. But the concept is there. Embryonic stem cells have the potential to make an ear, to make gametes, to do everything, but their potential is falling short, because they only give rise to their fetal counterparts.
Thank you, madam. Next question by Anand Rajan. What are paracrine providers? Can you shed little more light on this area?
Sure. So paracrine providers, basically, for a stem cell to divide and to differentiate, it needs a particular set of growth factors and cytokines. So the MSCs are the best as a source of growth factors and cytokines like VEGF and so many different kinds of cytokines, miRNAs. They are a very rich source of all these, which are very important for stem cells to proliferate and differentiate. Like you see, when you do embryonic stem cell culture, in order to differentiate them, you put in a lot of growth factors, etc., isn't it? Rather than putting in growth factors, you can grow them on a mesenchymal cell bed, because those mesenchymal cells will provide all those growth factors and cytokines, which are crucial for the stem cells. So paracrine providers means they secrete the growth factors and cytokines, which work in a paracrine manner on the VSCLs to support their proliferation and differentiation. And rather than mesenchymal cells as such, these are secreted as small microvesicles from the MSCs. So rather than transplanting mesenchymal cells, you can also just transplant these microvesicles or the exosomes produced by the mesenchymal cells, and you will see similar benefit.
Thank you, madam. Next, a small piece of appreciation from Rajan AR. Thank you so much for the talk, madam. I'm doing my project on hematopoietic stem cells, and this lecture was really informative and shined light into my new research area. I would be really missing if I would have missed your lecture. Thank you, madam. The next question is from Krushi Srinivas. As you have said earlier that if we abrupt the hormone, it might cause cancer due to increased growth of VSELS. So if they survive in cancer treatment, is it possible that they again cause cancer if they get similar conditions?
Yeah, that is
yes, why. Then why are not they dangerous?
So that is why the whole problem of recurrence. Why so much of recurrence is there? Because whatever treatments that are available right now, they are targeting the actively dividing cells in the cancer. So all the actively dividing cancer cells get destroyed by treatment, but the quiescent stem cells, they escape. The quiescent cancer stem cells, they escape. And since we have not rectified the niche, they are in the same inflammatory niche and they cause recurrence. So in the actual way to treat cancer is to just improve the niche of the tissue, to transplant mesenchymal cells and improve the niche. And you will be telling the VSCs, behave yourself, don't divide more, do your normal function of differentiation and behave normally, maintain homeostasis. And that is how we should treat cancer. There will be no recurrence if you improve the niche to the VSCs. But right now, everybody targets the actively dividing cancer cells.
Okay, thank you, madam. Next question from Surya. In regard to VSELS density gradient separation from cord blood, was it from lymphopreparation? What happens when we use hetastarch or SPAN for RBC sedimentation? Does it have an impact on VSELS separation during first spin?
Yeah.
So madam, the question is continuing. If so, what should be the ratio cord blood? Is it 1:5 or 1:6? And molecular weight of HESPAN?
Yeah, actually, we had done using HiPAC to prove our point. But with these other separation methods, we still need to see where exactly the VSCs go and separate, whether they get discarded or they get collected. But mostly, I think they get discarded along with the RBCs. But I have not used these different all these. I don't have any experience with these.
Thank you, madam. Next question from Adish. Good afternoon, madam. It was a really good lecture and I learned a lot. I just want to know that does it mean VSELS once isolated separatively from MSC differ in their efficacy than when MSCs and VSELS are used for treatment potential study?
I didn't get the second part of the study. Second part of the question.
Second part. Once isolated separatively from MSCs differ in their efficacy than when MSCs and VSELS are used together for a treatment potential study.
So actually VSCs from different tissues in our body, they all are the same. VSCL isolated from any source, including mesenchymal cells, they will be the same stem cells. But the tissue-specific progenitors, they vary from tissue to tissue. So if you transplant pure MSCs or you transplant MSCs plus VSCLs, I think transplanting MSCs plus VSCLs will be much more useful. But actually VSCLs are not required because whatever the disease tissue, like even if you have infarct in the heart or etc., the VSELs would have increased in numbers. It's only the niche they require. They only require the mesenchymal cells, the niche, in order to regenerate. But if you transplant MSCs along with VSELs also, both the endogenous VSELs and the transplanted VSELs will differentiate.
Thank you so much, madam. You have clarified all the questions, and
Okay.
it was
Yeah. Before.
Can I ask a question, Dr. Bhatia?
Yes.
Yeah. A very general question. Would you like to throw some light on the relative quiescence of a cancer stem cell versus a VSEL to be seen in the context of stress causing the mobilization of VSELs in case of an infarct or any condition, pathological condition? So on the one hand, stress is mobilizing these VSELs versus
versus the cancer tissue where they are actively dividing. So these are questions that need to be studied. But Shin, the Radzei's group has studied on the molecular basis what are the reasons for this quiescence in normal tissues. But how their quiescence varies and how they become active in order to initiate cancer, these are questions that remain to be answered. But in case of testicular cancer, we have a study model. If you treat the pups with diethylstilbestrol, when they become 100 days old, they become cancerous. The testicular cancer is seen. And the VSELs have increased in number 7-fold. They have become 7-fold more. But how these VSELs from normal tissue and those in the cancer, testicular cancer, how they are different from each other, you won't believe. Today only I had an animal ethics approval project in the morning. Now I want to transplant the VSELs from DES treated mouse in a SCID mice to see if they develop teratoma. Because if you isolate VSELs from normal tissue, they don't make teratoma in SCID mice. But whether these VSELs from a testicular cancer, whether they make teratoma in mice and how they behave differently in culture, they get epigenetically altered. But what are the epigenetic differences that nobody has studied till now? There is no information. There are hypothetical. Dr. Radzei's group has written that all the cancers initiate from VSELs.
This is Dr. Devashi speaking.
Lot more research can be done in the area.
So, would you like to join the session now, sir? Because the Dr. Bharatiya session is over.
But I can't find you in the list. What is your...
Thank you, madam.
Yeah, thank you.
I also have a small question. So since you said that they are present in the adult tissue and they are for their renewal and then injury and all these things, homeostasis. So any study has been correlated with the aging or longevity of the animal with the presence and the amount of the number of VSELs. Is there any study like that?
Yeah, we published this year only. Mouse bone marrow cells. In the mouse bone marrow, how the VSELs and HSCs, they change in numbers with age. And how they differ, and also how they respond to a stress in a young mice versus an aged mice. So that whole paper is there in literature, I can share with you. But what we have found, that the numbers don't change much with age. But if there is any kind of stress, these cells have a lot of potential to proliferate. In young, once the homeostasis is achieved, they come back to normal. But in aged people, they remain elevated in numbers. They don't have that potential to come back to normal. So they remain elevated. And that's why the tendency to cause more of leukemia, etc. with age. I can share the article with you.
So we can implicate that. Definitely we can research. Aging, we can do something about it.
Yes.
Fantastic, ma'am. Thank you so much. So in the interest of time.
Amit, that I want
Sure. Go ahead, please.
Professor Kora, Madam, just you mentioned that there is no such adult stem cells, only this USEL cells in every tissue?
No, there are adults. I am saying hematopoietic stem cells are there, spermatogonial stem cells are there, ovarian stem cells, but those are all progenitors. They are tissue specific. But actually, I think personally that we need to define the definition. VSELs are the tissue specific stem cells, other stem cells. Whereas the hematopoietic, spermatogonial, those are all actually, they are called adult stem cells right now, but they should actually be called tissue-specific progenitors. Because a hematopoietic stem cell will only make blood cells.
Because I used to teach students. Now I have to refine my brain about this, your findings and other things. That will be good idea for things. But what about this Yamanaka factor for this your USEL cells? Any accept of 4?
So when this Yamanaka's group published first time, I was there in that meeting when he presented. So people were asking, people asked him, is it really the skin fibroblasts that get reprogrammed? Is it really the adult stem cell, adult mature cell that gets reprogrammed to pluripotent state? Or is there some stem cell in your initial culture that starts dividing? So I still today, I believe that there were VSCs in his culture, which actually started dividing and made colonies rather than reprogramming of adult somatic cells. But there is a paper also in PNAS that if you remove the Muse cells from the from the skin fibroblasts, you will never get IPS. If you have the Muse cells, then only you get IPS. So actually it is the Muse cells which divide.
So you, in disguise of ESELs, that induced pluripotency has been developed.
Yes.
Maybe.
Yes.
Okay. Thank you, ma'am. Because I have to change my mind to how to teach the students about this stem cell technology.
Thank you, Gora sir, for this wonderful question. For the want of time, we may have to wrap up the session. I would request Dr. Amit to acknowledge for the session.
Thank you so much, Dr. Deepa Bhartiya, for your fantastic and inciting lecture. And then I thank Dr. Godwin Christopher, Dr. Debashish for helping us, and all the participants for very fantastic discussion. And on behalf of whole VIT family, including organizers, I extend my thankfulness to Dr. Deepa Bhartiya, and we hope to hear more from you in coming days. And then we would like to host you in person whenever the situation gets better. So that, and then best wishes from our side for your upcoming research towards this direction.
Anybody can write to me, contact on email. I'll be open to answer.
Thank you, ma'am. I'll be writing to you definitely.
Sure. Definitely.
Thank you, Dr. Bhartiya.
We have Dr. Biju George. He has already joined for the next session. Unfortunately, for the next session, we do not have the chair is not available now. So I would request Professor Amit to continue as a chair. I'm going to share the introductory slide with him. So for this session, I should thank Dr. Amit and Dr. Christopher, Godwin Christopher, for doing this wonderful job of coordinating this session. Thank you very much. Thanks for being with us. Thanks for all your help. Thank you very much, sir. I would request Dr. George, I would first welcome him. I greet him with a warm good afternoon. And I would request him to put his slide if it is okay, just for a testing purpose. Yeah, it's
the slide seen? Yeah.
Yeah, it's working. Yes. So you can put it in the curve view mode. You can scroll a couple of slides so that we can see. Yeah. That's really working good, sir. We can you can stop sharing now. We will put your introductory slide, slide which has got your CV. Yeah. I'm going to put this on.
Dr. Biju George, welcome. Thank you for taking time out from your busy clinical work. Thank you, sir.
Dr. Amit, is it visible?
Yes, it is clearly visible.
Okay. For this session, we have Dr. Amit to give, take the position for Dr. Everett Jacob Remington. He just got busy with with our dean, some official requirements. Also, I should welcome Dr. M.A. Jayashree. She would be the moderator for this session. Welcome, madam.
Thank you, sir.
I have given you the co-host credit, so that all the students will be shooting questions to you, whatever they get in this session, whatever queries they get in this session of Dr. Biju George. Now over to Dr. Amit. I request him to take a bit more pain and introduce our guest. Thank you, sir.
Thank you, Dr. Devashish. And on behalf of organizers as well as VIT family, I welcome Dr. Biju George, and it is my pleasure to introduce him. Dr. Biju George is a professor of hematology at prestigious Christian Medical College, Vellore. He did his MBBS from the Madras University, followed by MD in internal medicine and DM in hematology from MGR University. He received his subspecialty training in clinical hematology under the guidance of respected Dr. Mamen Chandy, who is also a pioneer in the field of the bone marrow transplantation in India. His research area of clinical and laboratory research lies in aplastic anemia, Fanconi anemia, myelodysplastic syndromes, post-transplant infections, alternate donor transplants, supportive care, and cellular therapy. He has accomplished more than 15 clinical studies. He has discussed his findings via nearly 180 invited talks in various national and international platforms. He also has contributed to several book chapters related to his research. He has more than 200 publications in peer-reviewed journals and clinically revered journals. This includes review articles in some of the most prestigious clinical journals of this field, like British Journal of Hematology, Biology of Blood, and marrow transplantations, bone marrow transplantations, pediatric transplantation, and Annals of Transplantation to his name. He has staggering citations of more than 5,000 and H-index of 38. His concurrent mission is on developing effective cellular therapies for the management of infections and treatment of refractory malignancies. So, Dr. Biju George, I welcome you once again. And then I would like you to share the screen so that we can hear your lecture, and we are looking forward towards it. Thank you so much.
I'd like to thank the organizers for inviting me to this meeting. Thank you, VIT. And I'd like to thank the chairperson for introducing. So what I would like to do over the next 30 to 40 minutes is talk about the role of stem cells in the field of bone marrow transplantation, as well as talk about something more in hematology. So what is a bone marrow, or what we popularly now call as a stem cell transplant? This is a process in which an abnormal bone marrow, and this abnormality could either be genetic or acquired, is destroyed and replaced with stem cells that will produce healthy blood cells. Now we know that the hematopoietic stem cells within the marrow, these are adult stem cells, have the propensity to form all the lineages of the blood, which includes the red cells, the white cells, which include the myeloid and the lymphoid cells, as well as the platelets. So any disease which involves the hematopoietic system can potentially be cured with the stem cell transplant. Now, these diseases could be any cancers, and the general cancers for which you would do a transplant include the acute leukemias, AML and acute lymphoblastic leukemia, the chronic leukemia, CML, and of course myelofibrosis, and other diseases such as myelodysplastic syndromes, lymphoma, and myeloma. Now, we need to remember that each disease per se may not need a transplant, and for each disease, there are specific indications where you would do a transplant and where you would manage with just medical treatment alone. If you look at the non-malignant hematological conditions, there are a lot of conditions which are curable only with a transplant. For example, genetic disorders such as thalassemia and sickle cell anemia, bone marrow failure diseases like aplastic anemia and Fanconi anemia, and a disease that we commonly see in childhood, which are the primary immunodeficiency disorders, usually severe combined immunodeficiency and HLH. Now, any of these diseases can potentially be cured with a stem cell transplant. Now, when we look at the process of how a stem cell transplant occurs, the first step is to eradicate the host bone marrow, that is the bone marrow of the patient, in order to allow the stem cells to come and settle down. Then there is the infusion of the stem cells that occurs, and subsequently these stem cells home into the bone marrow of the host, settle down, and then start engrafting, and that finally shows up in the blood counts recovery. The eradication of the host bone marrow is done usually by either chemotherapy alone or in combination with radiotherapy. Now, we do not try to kill the entire bone marrow as such, but it should be just enough to create space in the marrow, as well as ensuring that it does not damage the other normal living tissues in the body, mainly the liver, the heart, the kidneys, and the lungs. Once that is done, then the next step is the infusion of stem cells, and for that, we need a donor. So what are the kinds of transplants that are available? In general, there are two broad transplants. One, what is called as autologous or self, and it essentially means that the donor, the patient acts as his own donor. So the first step is to actually collect stem cells from the patient, cryopreserve it, then give chemotherapy, and then post chemotherapy, the cells that are already preserved are infused back into the patient, and like I mentioned previously, engraftment will occur. Now, these are generally done for diseases such as lymphoma and multiple myeloma, which are the common indications for an autologous transplant. On the other hand, an allogenic transplant is where you have a patient, you have a donor who's separate, and you give chemotherapy to this patient, and then you collect the stem cells from the donor, infuse it, and then you expect the patient to recover. So this means that this is useful in diseases where there is a primary problem. So if you have a genetic disorder, for example, like thalassemia, there is no point taking the patient's own stem cells and trying to transplant it. You need to get stem cells from somebody else. Similarly, if you have a marrow failure, you're not going to get enough cells to transplant. So most of the diseases where the marrow is primarily involved either by genetic or by a failure, you will need an allogenic transplant. Now, when you want an allogenic transplant and you need a donor, the question is, who can be the donor? Now, to find a donor, we need to do what is called as HLA or histocompatibility antigen typing. And essentially, we look at 6 major antigens which need to be identical. These antigens are important because they are the ones which help in determining self from foreign. There is MHC class 1 and class 2, and these are the ones which help our innate and our humoral immune system in recognizing proteins within the body, which are either your self proteins or foreign proteins, and attacking foreign proteins. Now, if they are matched, you reduce the risk of both graft failure and graft versus host disease. We look for 2 antigens on A, 2 on B, and 2 on DR, and we generally need a full 6 out of 6 match for a donor to be available. So looking at this, who would you get as a donor? Now we need to remember, for each of us, the HLA comes half from the father and half from the mother. So the best chance of finding a match would be a sibling, either a brother or sister. And there is probably a 30 to 35% match that you can find a sibling who is matched. But remember, out of 100 people who need a transplant, only about 30 to 35% will find a donor. Now, in certain scenarios, for example, if you take Tamil Nadu, they have a system of consanguineous marriages. In other places in North India, they marry within the community. Now, there are only a certain amount of HLA haplotypes that are available throughout the world, and therefore, it is, if you are marrying within relatives, it is possible that the father and the mother may share similar haplotypes, and so there is a chance that the children may actually match with parents or with the extended family. The third would be to look for unrelated donor around the world, and because there is a limit to the amount of HLA haplotypes available, it is understood that 1 in million persons will be matching with you completely identical. So around the world, there are worldwide bone marrow registries that do HLA typing and save these information on a computer, and you can search through the database and find out if your patient who doesn't have a sibling donor has a fully matched unrelated donor. Now, what is coming up, and I'll talk about it later in a big way, is a mismatched sibling. That means we know that because half the HLA comes from the father and half from the mother, we know that for every child, the parents will be haplo-identical or half-matched. So now there's an increased drive to look and see whether they can be used as donors for stem cells. And finally, you can use cord blood. So where do we get these stem cells from? Now, these stem cells we can obtain from the bone marrow or from the peripheral blood, or as mentioned, through the cord blood. Now, this, if we are obtaining stem cells from the bone marrow, we generally take them into a theater, and it's done under general anesthesia. It's done under sterile circumstances. You use small needles to aspirate along the iliac crest, and once you aspirate them, you put them into a large bag, and once it is counted, and then this bag containing blood and the cells are infused back into the patient. Now, if you're going to collect it from the peripheral blood, you can do it as an outpatient procedure, and this is done more often now. You just connect them to an apheresis machine. It basically draws the blood into the machine. It is a centrifugal force is applied to the blood product. The blood separates into layers depending upon what component you want to collect. You feed in that program, it will collect those cells alone, and the rest of the blood comes back into the patient. So if you see this bag on top, this is the component. So finally, what happens is this procedure goes on for 3-4 hours. You would end up processing 10 to 15 liters of blood, but what you finally would take out is roughly about 200 to 250 ml of a harvest product, which is actually even less than a unit of blood that you would donate if you're going to do a blood donation. The transfusion, or what we call as the transplant process, is actually a simple process. It is not like an organ transplant, and it is essentially transfused similar to how you would transfuse a routine blood product. Now, these have the they go through a first bypass through the lung, and then they home into the marrow, and then they produce good cells. So if you look at a harvest product, you need to remember that it would contain stem cells, it will contain your T lymphocytes, it will contain some natural killer cells, and it will contain some B lymphocytes. So the harvest that we actually give to the patient is a mixture of all of these cells that are there inside the harvest bag, whether it is from the bone marrow or from the peripheral blood. And where are these stem cells? These stem cells are in, most of them are in quiescence and retained within the hematopoietic niche. And so when you do a puncture to try and aspirate, you are trying to aspirate as much of cells that you can from the hematopoietic niche. Now, how do we do when we want to get it from the peripheral blood? We give something called as growth factors or colony stimulating factors, and these help in mobilizing. So the CXCR4 and the CXCL12, which help in retention of the hematopoietic stem cells within the marrow, the bonds are broken, and these hematopoietic cells escape through these endothelial sinuses into the blood, and within 4 to 5 days they are present in the peripheral blood. So from a normal WBC count of about 4 to 5,000, once you give these growth factors, by the 5th day the white cell counts will be sitting at about 40 to 50,000 with an adequate amount of stem cells, and then you put them on the machine and collect as much of stem cells as you want for the transplant. So there are disadvantages, advantages of doing one after the other. So the bone marrow has got a lower number of stem cells, while the PBSC has got higher number because you're stimulating it for 4 to 5 days. Bone marrow has also a lower number of T lymphocytes as compared to PBSC, but these higher number of T lymphocytes contribute to one of the problems that occurs after a transplant, which is called as graft versus host disease. The graft versus host disease is essentially the donor T lymphocytes attacking the patient's body, and because it is foreign to them. Now, there are some advantages. So you don't have a risk of anesthesia when you do a PBSC. You can do it across blood groups. So unlike, say, a renal transplant where you like to get somebody who's got the same blood group, here you can do it across blood groups. You just need to deplete out the red cells, because what we are finally interested in is the white cell product, because it's within that the stem cells are actually present. So what happens normally is, and this is a pictographical representation, you give this chemotherapy here for about 5 to 7 days. You infuse these stem cells, so your blood counts will drop. The WBC counts remain at about 100 for about 2 weeks, and then it slowly starts recovering, so that between day 14 to 21, it comes back to normal levels. Generally, by day 28, the natural killer cells come back to normal levels. But we need to remember that the B cells will take and the T cells both will take between 1 to 2 years for them to come back to normal levels. So because of the neutropenia and because of the late immune reconstitution of the T and B lymphocytes, we have a host of infections that can occur during this period. These infection is usually related to the fact that the immune reconstitution is occurring very slowly. So because they go through a period of neutropenia where they are at a high risk of infection, all of them kept in isolated rooms which are HEPA filtered, and so that helps in reducing the risk of infection. Everything that is needed for the patient, including stethoscopes, whatever is needed in terms of IV fluids, everything is kept within the room so that there is no cross-contamination across patients. Food, if they can eat, is terminally pressure cooked. Only boiled water is used, and we try to minimize relatives so as to reduce the risk of infection. So in general, you go through this period where the counts are very low, the peripheral blood is very clear without any cells, and within 2 weeks, as the counts start to come up, you find these neutrophils coming out in the peripheral blood, which means engraftment has occurred. During this period, patients do need blood and platelet support as well as antibiotic support during this period till the counts recover. There are complications that occur during a transplant. One would be toxicity of the chemotherapy drugs that we give. I have mentioned infections. The third dreaded complication is graft versus host disease, where I said that the donor lymphocytes will react against the body. Now, some GVHD is good because along with the graft versus host disease, the donor lymphocytes will also recognize any leukemic cells which are persisting within the body as foreign and will attack it. And so you also get an additional graft versus leukemia effect. So little bit of GVHD will lead to GVL, but bad GVHD can actually kill. And as was mentioned by the previous speaker also, one of the biggest banes of any cancer treatment is that despite what we do, you can have relapse of the disease even after a bone marrow transplant. Now, briefly, I just talk about our department. So we started bone marrow transplants in 1986. We are the largest transplant center in the country, and we do roughly about 250 transplants a year, including 180 allogenics and 60 autologous. Roughly 40 to 50% of transplants are in children, and we do between 3 to 4 alternate donor transplants. And this is the kind of breakup. So this is data on about 2,500 transplants that occurred till June 2020.
Sir, so I'm sorry to interrupt you, sir.
Yeah.
We are unable to see your slide moving.
Okay. Now?
No, sir.
I am seeing it move. Shall I share it again and see?
Yeah, please.
Yes, please. Yes. Yeah.
No.
Now can you see?
Now it is okay.
Okay. So this is about 2,500 transplants till June 2020. So genetic disorders, thalassemia form a major group, followed by acute myeloid leukemia and aplastic anemia. The rough cost is about 15 lakhs, but we are much lower than most of the Western countries in terms of the cost. This is for a sibling donor. So if you have a matched sibling donor, and we are looking at between 50 to 80% cure rate. So it will differ based upon whether you're dealing with a malignant disorder or a non-malignant disorder. But these rates are very similar to what you would get at most of the major institutions around the world. Now, that is the use of stem cells in bone marrow transplantation. So the question is, can we use these stem cells in some different ways also? So like I had mentioned, only about 30% of the patients who need a transplant will find a matched sibling donor. So what do we do for the rest who need a transplant but do not have a sibling donor? Their haploidentical parents or siblings have become universal donors. So in haploidentical transplants, there is a fine line between graft failure because the HLA is half matched. There is a high potential for the patient's body to reject the donor cells, which leads to high graft failure. There is also high potential for the donor cells to react against the patient's body, which is graft versus host disease. So both the risk of graft failure and graft versus host disease is high. So what can you do for this? So we need to remember that the CD34 positive stem cells are the cells which are most important in terms of engraftment and reducing graft failure, while the CD3 positive lymphocytes are the ones which cause graft versus host disease. So the question is, can we increase the CD34 positive stem cells and reduce the T lymphocytes, which will help in improving engraftment and reducing graft versus host disease? And so there is a method of doing it. So you do a large harvest, you take the harvest product from the apheresis, which is what we collect, and then we can add antibodies against CD34. This is called as a CD34 positive selection. That means you add an antibody which is mixed with iron against CD34, you put it into this bag and allow it to rest, and then you run it through this magnetic column. This is the Clinimax, which is used for clinical separation. As you run it to the magnetic column, all the CD34 cells which have got iron sticking to it will get collected into a different bag, and all the rest of the products will get collected into a different bag. So you will end up having a product which will have mainly CD34 with hardly any CD3 positive T lymphocytes. You will also have hardly any B and NK cell lymphocytes. So using this approach, a donor is available for all patients and roughly about 50% can be cured. But the big problem is that you can get recurrent infections because there is hardly any T cells that you're putting into the graft. And so the T cell reconstitution after transplant becomes much more slower. And so there is a high risk of viral infections occurring in this group of patients. So what else can we do? So what we can do is, and sometimes what we do is, instead of positively collecting the CD34 cells, you can actually negatively select out the CD3 cells and remove that out. So again, here you use an antibody against CD3, which is coated with iron, so that just that alone is collected and separated out. And here now you have a mixed bag of stem cells with B lymphocytes and natural killer cells which are there, which can be given. Still, because there are no T cells, there's a higher risk. And so newer machines have come out, which makes this processing much faster. And so people are looking to see, can we remove only the activated lymphocytes that cause graft versus host disease, while the rest of it, which is useful for a transplant, can actually be infused back to the patient. Now, the other cells which is of interest to a transplanter are these mesenchymal stromal cells, which are present in the hematopoietic niche along with these quiescent hematopoietic stem cells. Now, these mesenchymal stem cells or stromal cells have the ability to differentiate and commit themselves to one of these lineages, including the lineages of the blood cells. So what has been shown, at least in the field of transplantation, is that the MSCs play an important role in modulation of immune responses against allo and autoantigens. And it can sort of dampen the cells which generally cause an inflammatory process. So it inhibits natural killer cells, activated immature dendritic cells, and the T lymphocytes, all of which form a part of the process of causing graft versus host disease. Importantly, it activates T regulatory cells, which is known to be important in reducing graft versus host disease. So now studies are ongoing whether you can actually co-infuse MSCs along with stem cells to a patient who's undergoing transplant to help reduce the risk of graft failure and reduce the risk of graft versus host disease. I should also mention that MSCs are now increasingly also being used because of the good differentiation potential of being turned into multiple tissues, as was mentioned also by the previous speaker, in terms of making cardiac myocytes, liver, islet cells to help and regenerate other tissues. The other area where stem cells are coming into a big being, which is similar, akin to stem cell transplantation, is the whole field of gene therapy. Now, if you have a patient who's got a stem cell which has got a genetic defect, which is monogenic, then you can isolate out the stem cells. You have a vector and then you put it in, and then it converts and corrects the stem cell. You grow it in culture and then you put it back into the patient, and then hopefully, because you've corrected the defect, genetic defect, you can actually cure the disease. Now, there are advantages and disadvantages. The big advantage is that these are autologous cells because you're taking the patient's own stem cells and then reinfusing it back. So the risk of rejection is very low. And the risk of getting graft versus host disease is very low. Importantly, you need very minimal chemotherapy to allow these genetically engineered cells to engraft back, so the side effects are less. But the problem is it's not easy to produce. It has to be individualized. It is individually based. You need monogenic diseases. So most of the acquired diseases, which are mainly cancers and bone marrow failures, cannot be treated this way. And one of the biggest worries has been that you are using a viral integration, and so you can get random viral integration. There have been some studies, especially when looking at immunodeficiencies, where there was a slightly increased risk of them developing leukemia. The other problem is that the viral integration and the production of good cells may not be complete. And so you may get a transient phase where you get a good improvement, but it may not last long. But there are clinical trials ongoing in severe combined immunodeficiency, thalassemia, sickle cell, and hemophilia. And that is something we need to look forward to. So the other question is, can you create stem cells if you're not able to do that? So I think this was again talked about the fact that Yamanaka showed how you can produce pluripotent stem cells from human fibroblasts and create iPSCs. And the important thing about the iPSCs is that these iPS cells can again be differentiated back into mesodermal, endodermal, or ectodermal cells, depending upon what programming factors you use. Now, why is it important? It's now becoming very important because one, you can actually study disease biology. So you can create these cells, study the disease biology. You can actually knock out a gene and see what is the effect of that knocking out of that gene and how much it plays a part in the disease biology. People are looking to see whether you can use those to screen compounds and find out which are the compounds which are likely to be active against specific diseases. People are looking to see whether you can do gene editing and gene targeting to repair specific disease mutations. And once you do that, whether these health cells can then be redifferentiated and transplanted back. And so you look at stem cell transplantation of genetically matched iPSC. So there is a lot of work going on into looking at these factors in various roles in the field of hematology and stem cell transplantation. And of course, not to forget the fact that the use of iPS cells is also playing an important role in regenerative medicine in the formation of looking at the different cells, the neural cells, muscle cells, bone, and seeing whether it can be used in other diseases. For example, osteogenesis imperfecta. There are many diseases where trials are going on in terms of producing these cells. In the end, I would just like to talk about two associated things. One is we know that there are complications of infections that occur after a stem cell transplantation. So the question is, because there is delayed immune reconstitution, can you actually manufacture cells which will act against viruses? So, and people are looking to see, can we develop virus-specific cytotoxic T cells? So when you do a harvest for a transplant, you take a bit of these cells, keep it, and separate out the T lymphocytes, then induce it against viral antigens, and then try and get cytotoxic T cells. If you're able to do that, and now with the newer devices, you're able to do that within 24 hours, you are able to. So if you have a patient who's got a drug-resistant infection, you're actually able to induce these cells and infuse that in the patient. And there are clinical trials going on using it in CMV, Epstein-Barr virus, adenovirus, BK virus, showing that these are useful in controlling infection. People are also looking to see whether you can use natural killer cells instead of cytotoxic T cells. And importantly, the other big problem in India, because of the massive construction that goes on, is fungal infection. So people are also looking to see whether cytotoxic T cells can be generated against Aspergillus antigens, which will again be infused back into the patient and can help in fighting infection. And the last is the whole promise of chimeric antigen receptor T cells. Now we need to remember chemotherapy is limited in terms of how it can kill cells. And so you need to try and harness the immunity of the individual. So these chimeric antigen receptor cells are T cells, which are autologous cells, which express a receptor antigen against the tumor surface. So if you have a B cell leukemia, which expresses CD19, then you get these CAR T cells, which also express CD19. It will go and bind to these tumor cells and then will cause lysis and kill. So you get these T cells out from the blood, you induce the gene for CAR, you get a chimeric antigen receptor, you grow millions of them and infuse it back into the body. And there are, depending upon what kind of antigen receptor you're looking for. So you're looking at CD19 for leukemias and the B-cell non-Hodgkin's lymphomas. You are looking at BCMA for myeloma. The studies going on CD33 expression in AML and 30 expression in Hodgkin's. But it has been very impressive that people who have failed 4-5 lines of therapy and have even failed a transplant actually show response to these kind of therapies. And so if you look at FDA approvals, there are CAR T cells which are now approved for its use in refractory B-cell ALL, diffuse large B-cell lymphoma, and mantle cell lymphoma. Problem is the cost is prohibitive. In the US, it's about $500,000 to produce these CARs, and it's another $500,000 post-infusion to support the patient because of the complications that occur. So it's roughly about $1 million to actually treat a patient with CAR T-cell therapy. There are multiple institutions within the country which are now started to look at CAR T-cell production, and we hope it's available to the Indian population at a much lower rate. So I would conclude by saying that stem cells form an integral part of the process of stem cell transplantation, and there's an increasing role for manipulation of stem cells in alternate donor transplant. Mesenchymal stem cells are increasingly studied in stem cell transplantation and how it can be used along with the stem cells in helping have a successful transplant. The use of gene therapy and the iPS cells needs to be explored further, and the use of cytotoxic T cells and CAR T cells are new forms of treatment that is slowly starting in India. With this, I'd like to stop. Thank you for your attention, and I will be happy to take questions.
Thank you, Dr. Viju George, for the fantastic presentation. And then I'm confident that the audience must have learned lots of things, and then they have few questions in their mind. So if audiences have questions, they can put it forward, and Dr. Jayashree can.
Yes, sir. Yeah. Thank you. There is one question from the participant, Kushi. The question is, what are the reasons that stem cell therapy for thalassemia is cheap in India but expensive in the USA?
I think that has got to do with just the basic insurance system. So because of the insurance system, we need to remember that there is a different kind of payment that is done for every person who works. So because of that, the costs of transplantation are much higher in the Western countries as compared to India. But I think that is true not just for transplantation. Even if you take, say, an angioplasty, it is definitely much more costlier in the West as compared to India. And that is because a lot of hospital costs, overheads, other associated costs all come in. And that is because it's all covered by insurance. While here, most of the time, patients are paying out of their pockets. And so it's important for us to realize that and try and keep the costs as low as possible, but making sure that we are not compromising on efficacy.
Okay. Thank you, sir. So the next question is from Kshana Surya. The question is, what is the dose of MCs to counter GVHD? Is it 1 million cells per kg body weight?
Yeah, generally it is between 1 to 2 million cells per kg body weight. The question is, it is not more than the doses. The question is the number of infusions that you need to give. So some people would consider giving 1 to 3 infusions at weekly intervals. Some people would give just 1 infusion and see, but it's generally 1 to 2 million per kg.
Yeah. The second part of the question is, what's your view on first pass effect after MCS infusion to counter GVHD?
So we need to remember that when we are transfusing stem cells, we are transfusing a lot of T lymphocytes, and these T lymphocytes naturally will cause GVHD. Now the question is, as a transplanter, do you want GVHD or not? In a non-malignant situation, you don't want any GVHD. So there, co-infusion of MSCs along with the stem cells will help in reducing GVHD. While in a malignant transplant, you want some GVHD because it has got a graft versus leukemia effect. So you don't want to completely stop GVHD. So what you would probably do in that situation is to watch and see what happens. When you naturally develop GVHD, is it severe or not? If it is severe, you can treat it with MSC infusion and don't use it as a preventive measure.
Okay. Thank you, sir. There is one more question from Anand Rajan. How much time it takes to complete this transplantation in your lab?
So the transplantation doesn't occur in the lab. It is the patient is in the ward. So basically it is 1 week patient is in the ward getting chemotherapy. Then the stem cell infusion is like a blood infusion over 1 to 2 hours. And then it takes about 3 weeks for the patient to recover back to become normal health. So it's roughly about a month of time that they will have to be in the hospital. But for them to become well enough to get back to work would generally take between 6 to 12 months.
Okay. Thank you, sir, for clarifying the questions. That's all from the participant side. Over to you, Dr. Debashish.
I would request Dr. Amit to conclude the session. First of all, I should thank Dr. George for his stimulating talk, and now I should thank also Dr. Jayashree for efficiently managing the session. Over to Dr.
Thank you, Dr. Debashish.
Dr. Amit,
Yeah, sure.
Dr. B. George, I want to thank you for having taken time out. And also, you know that Dr. Shaji and Dr. Sanjay Kumar are also presenting.
Yeah, I saw that. I saw the agenda.
In fact, Dr. Shaji helped me to connect, connect to you.
Okay.
And, I'm very thankful, and I'm sure we'll have more discussions later on.
Sure.
Thank you.
Dr. Khora, do you have any questions?
Just for curiosity, this is for Biju George.
Yes, sir.
This Professor Khora. I think, as you mentioned that your bone marrow transplantation started in 1986 onwards, right?
Correct.
So before that also, was there any notion about stem cell transplant, or only in the sake of regeneration? I think stem cell therapy has new phenomena, right?
So
how to explain that before that transplantation happened?
So we when we talk about stem cell, when we say 1986 is where we started in India in CMC. But if you look at the data, the stem cell transplantation started actually in the 1950s and 60s in the US. So it mainly started because they started looking at people who had leukemia or bone marrow failure after the nuclear explosions or after a nuclear holocaust. And they were trying to see how they can rescue these people. So initially they found that by collecting stem cells, bone marrow, actually collecting just bone marrow from other people, it could be infused. And then some people improved, but while some actually developed bad graft versus host disease, they realized there is something beyond just routine infusion of bone marrow cells. That is where HLA came into being, and the fact that you need HLA identity. So that work started in the 60s in the US. Here we started only in the 80s.
No, my another further question is that in the 1960s or 50s, where it was started, that concept of stem cell therapy or stem cell therapy was not there, I feel, in medical science. So how we conceptualize this regeneration medicine, stem cell therapy?
Correct. And I think that happened when people started to understand the biology of stem cells better. Initially, the understanding was that stem cells, which are derived from the bone marrow, are destined only to produce hematopoietic cells. And that is how it was used for stem cell transplantation. Subsequently, understanding of the biology meant that, one, we could find other cells, for example, the mesenchymal stem cells in the bone marrow. Two is the whole question of programming. And that is where people realized that you could actually take one set of cells and you could try and program it to becoming other cells. And that is where the whole process of regenerative medicine has come in.
Oh, thank you. Thank you.
Thank you,
Now,
Dr.
Amit,
Yeah.
thank you very much.
So on behalf of organizers and whole VIT family, I thank Dr. Biju George for taking his time out of his OPDs and busy schedule in the clinic. Thank you so much once again. Over to you, Dr. Devashish.
I thank Dr. George again for this time. Stimulating talk. For the time being, we are closing the session. We will rejoin the session at 2 p.m. To be precise, 2:15 p.m. Our next session starts. That session coordinator will be Dr. Manjuwala. She'll be coordinating the whole session.
Dr. Devashish,
Yes.
Tell them 2 o'clock. 2 o'clock will be okay.
Okay. So all of you try to have a quick lunch and please join at 2 o'clock as our conveners, Professor Suresh corrected me. So we will have Dr. Ashwini Sharma from Sweden. He'll be joining us at 2 p.m. He will be also talking about on induced pluripotent stem cells. Okay, so it's nice coordinating the session. I thank Dr. Suresh for giving me the opportunity. I thank all the session chairs and moderators really did a very great job in coordinating the sessions. Thank you very much.
Dr. Devashish, on behalf of everyone, I wanted to thank you once again. It is a wonderful job and your job is not over. I hope you continue to support us for the remaining 1.5 days. I know you have other academic commitments and today I'm very glad to say that you did a wonderful job and I need your support for the remaining 1.5 days. Thank you, sir.
I am there, sir. I'll be there. At 2, I have a class, so maybe I'll join at 2:15 or so, 2:45 or so. I would request Dr. Manjuwala to go ahead, start the sessions. I'll be logging off, so the host credentials, we have to again get it back from Mr. Gopi. Dr. Suresh, I will request you to communicate with Mr. Gopi again.
All right. All right.
And we are closing the recording also. So at
Sir,
2,
can we leave the session on itself over here? Just close the recording and let the Zoom be here meeting.
That also possible, but let me make you the host now. Okay, madam?
Yeah. Okay. Okay. That's not a problem, sir.
Yes. I'm closing the recording and stopping streaming. Okay.
Yeah.
For streaming, you have to go to the more and go to YouTube Live. Or I'm not stopping the streaming then. Okay, I'm just making you the host now.
I suggested we will leave the YouTube Live on as well.
I'll just leave it on. I'm just keep making Dr. Manjuwala host so that she will be able to manage the session.
To turn the YouTube back on.
No, I haven't turned off at all. I have just turned off the recording. It's still going live on YouTube.
Yes, sir. Yes.
Yeah. So, madam, you are the host now. I am neither host nor co-host. I can easily, leave now. Thank you, ma'am.
Thank you, sir. Thank you. So, Khora sir, We will meet by 2 o'clock, before 5 minutes before 2.
Or by 2 o'clock, at 2:15, no, our session starts?
No, sir,
2:15?
we starting at 2.
Okay. Okay.
Yeah, we'll start at 2.
Okay. Thank you. Okay, madam.
Okay, sir. Thank you.
Good afternoon, madam.
Good afternoon, sir.
Yeah. So you are going to be the moderator in the first session, isn't it?
Yes, sir. Yes, sir.
All right.
Sir, I'm waiting for Dr. Ashwini Sharma, and I'm not sure about his login name.
Aha. Okay. I will send him a mail.
Yeah. And also, Dr. Bhavna Muralidharan has not joined the meeting yet.
Okay. Okay. I'll send a mail.
Yes.
Oh, no, I'm here.
Okay. Hello. Sir, I had a doubt with your ID, so I was just got confused.
Yeah, it took my, I think so, computer name, ASH, like initial.
Yes, yes, sir.
I was there.
Ashwini Sharma, good afternoon.
Like around 4:30. It was very interesting inaugural lectures and something. It was very interesting to listen.
Dr. Ashwini Sharma, this is Suresh here. Suresh here, PK Suresh.
Yeah.
I communicated with you earlier. In fact, you're the one who put us in touch with Dr. Kannan Krishnamurthy.
Yeah, thank you. Yeah, he was very great to speak. And then plus also, I know VIT team. I don't know, I never visited, but I know some fellows who are working there, and then plus actively communicating with them the work. And it was really interesting to listen lectures of Professor Kannan, and plus also the inaugural lectures. It was really interesting. So that make me awake in the morning itself to listen all those things.
Yeah, the time difference now it's about
4.5 hours.
4.5 hours. About 10:30 or
now it's like 9:30.
9:30. Okay. Dr. Manjubala would be
yes, sir.
She's the organizing secretary here, and she'll be running the show with the support of the chairpersons and the moderators.
Okay.
Madam,
Yeah. Yes, sir.
Recording has started.
I just started the recording, sir.
Thank you.
So, good afternoon. A warm welcome to all the participants, the speakers in the afternoon session. Today morning, we had very good sessions on stem cells and also the induced pluripotent stem cells and so on. I take privilege to coordinate this session for 4 speakers of the conference today: Dr. Ashwini Sharma, Dr. Bhavna Muralidharan, Dr. Krishnamurthy Kannan, and Dr. Sarjan George. They will be the speakers of this afternoon session. And I would like to introduce the chairperson, Dr. Samantha Shekhar Khora, who will be chairing this session for the first 2 speakers. And in fact, I will be moderating the sessions. In case if anyone is having any questions, please post it to me. I'll be there in the chat box all the time. And I hand over the session to Dr. Khora, sir. Please.
Thank you, ma'am. Thank you.
Yeah.
I think if I'm correct, that stop recording, it has come, not recording. Stop recording. It is showing stop recording.
No, it is recording.
It is recording, sir. It is recording, sir.
Mentioning the stop recording.
No.
Please see that.
No. That is to stop the recording. If you hit that, the recording will stop. You're okay.
Okay. Thank you, ma'am, for giving this opportunity. Can you?
Yes, sir. Yes, sir. I'm just sharing the screen, sir. 1 second.
Please.
Is that okay, sir?
Okay, madam. That I think make it enlarge. It is a previous mode. Madam, okay. So, thank you for giving me this opportunity to chair this session. Now, I welcome Dr. Ashwin George, Ashwin Sharma for this talk of this induced pluripotent stem cells in this international virtual conference on update on stem cell academic and industrial perspective future challenges. Dr. Ashwin Sharma has more than 10 years of working experience in domains of business development and industrial research. He has PhD in bioinformatics from Indian Institute of Technology, Bombay in 2011. Worked as a research scientist, position in one of the prestigious engineering institute of France, École Polytechnique. Worked as a senior research scientist and marketed research manager with Sanofi Aventis, France, Frankfurt, and University of Nuremberg, Elbergen, Germany, in the field of drug discovery and molecular modeling between 2011 to 2013. Later, he joined École Polytechnique and ENSCP Paris as a senior scientist in the field of computational chemistry with application in drug discovery and DNA diagnostic. Since 2017, he is commercial director, business development manager, and sales manager in the different biotech industries of international repute, which provides applications of cells in the field of drug metabolism and drug toxicity prediction. Also, he serves a board of members for France Pharmacokinetic Society and European Society for Pharmaceutical Sciences. With this, I welcome Dr. Ashwin Sharma to start his presentation with a limited time. Dr. Sharma, please.
Okay, thank you very much. So I will share my screen. Okay, now everybody can see my screen?
Yes.
Yes, visible, sir. Yes, sir, go ahead.
Yeah. So first of all, thank you very much, VIT, and then Dr. Suresh and Dr. Manjubala and Dr. Kora to give us the opportunity to explain a diverse field and to learn also. It's not only to explain, but also to learn from different eminent scientists of India. I was always communicating with different people in different fields, but I was really amazed. The lectures, even since organized in the morning, and that's why I was saying like it was very opportunistic to learn new techniques. Plus also I can see like in next 2 days, it's going to be very eminent lectures. So I'm very much happy to exchange our thoughts and my experience from the company and what are the new tools can be useful for upcoming fields. So today I'm going to present one lecture, which is on induced pluripotent stem cells, which is a very good in vitro model for drug discovery and therapy. So as already explained by Dr. Kora, so I will not explain about myself. Currently, I'm working as a business development manager and the sales manager in the company Sensagene, which is located in Sweden. So where exactly these kind of cells are useful? Just to have a little introduction, maybe most of the people are aware about drug discovery process because my half of the life was spent on drug discovery, working with pharmaceutical industry, and also provide some information of drug discovery for cosmetic industry. Just to take a quick glimpse of drug discovery process, when you know very well, like the drug discovery is not an easy task. It goes for the 10 years. And then that's why there are two different ways to treat the particular disease. Either you go for the therapy process or you for the drug development. When you will go for the drug development, the initial phase, which will start for exactly for target discovery at hit generation and further on lead identification. These processes are now adapted by different tools where you can use either high throughput screening tools or in silico tools. But the major roadmap which will start after the lead optimization is your preclinical screening. And preclinical screening, which cover lead optimization and animal models. So what you are exactly looking for at this preclinical screening. You are looking for ADME. So ADME is absorption, distribution, metabolism, and elimination. So whenever you will take a drug, you have to carry out all those assays which are providing ADME analysis. Further on, you have to analyze efficacy and then safety of the drug. When you will try to escape this particular door to enter into clinical science and the clinical stage of the drug discovery program, you always get lots of problems. Like you can see a recent example when the COVID-19, which passed this preclinical screening, at the later stage, like you are start getting response from the patient that you are developing some toxicity, some patients are developing some particular, and the same medicine we consider if it is not tested at the preclinical stage, it will get rejection based on the population. And according to my experience, like the same drug cannot be applied for different population. Why? You have to understand preclinical screening based on those population. So preclinical screening is one of the major steps for drug discovery process. Where and how you can apply for preclinical screening. As you can see in this stage, when you will consider pharmaceutical industry, you have two different assay models where you can use. Either you will use the in vivo assay, which is also widely applied till now, but there are some restrictions are coming in European Commissions about that. So when you will look about in vivo assay, usually it's simple to implement. You inject your drug, depends upon where exactly you are looking for. You select the animal model and further on you collect the parameters. So it's quite straightforward. But when you will go for in vitro assay, you have to select which particular in vitro assay you are selecting, which particular in vitro model, which is based on those assay. So here you can see based on those in vitro models, there are several cell lines are developed where you are looking. For example, if you are looking for liver disease, if you are looking for kidney disease, so you have different cell lines are also there. In the same way, you are also looking for primary cells. So you can see preclinical screening are divided into two stages, where you can use animal model or in vitro model. Now, when you are going towards European Union, so in European Union, they start banning animal testing. And they said, okay, for cosmetic industry, we completely ban the animal testing. If we want to compensate in pharma industry, how we can reduce these animal use and to put more towards in vitro models. That's why the FDA and EMA, they aligned together and they decided, okay, we have to force pharmaceutical industry to use as much as in vitro model to replace animal testing. That's why they generate this concept, which is very famous in Europe. And also I attended several conferences in India as well as with FDA regulatory agencies, their recommendations, lots of discussions. So they decided to implement same, which is going on in Europe. And we call this is like a 3R principle. Several societies now established in different countries, like I'm located in France and also worked in Sweden and in Germany. They are implemented this 3R principle. What exactly 3R principle is? Replace, reduce, and refinement. When you will talk about replace, when I talk with different pharmaceutical industry, okay, you want to use the stem cells, you want to use some primary cell line, you want to use cell line, or you still want to use animals. The first and foremost answer, we want to use animals. So why? Because the data is quite relevant because it's in vivo, but they want to reduce, and currently because of this strict forces, the government has to provide some solution, but the government is not able to provide those solutions directly. So they decided to communicate with different agencies, like we follow the FDA, and EMA is less implemented in India. But now all the agencies are together to understand how we can implement 3R principle. So in replacement, you try to put some compensating in vitro models which can replace the animals. Yes, it is very difficult, but yes, this is the way for the future. Reduce. If you can say, okay, you can produce some assay from animals, but can we implement like some in vitro assay where we can have reduction in number of animal use for the experiment at preclinical screening? Another one, refinement. In this stage of refinement, we can we do something, means we can provide that particular dose which is not so stressful for animal, and also which will not so much of painful for animal. So based on these three concepts, the government decided to implement 3R principle, and all the agencies are aligned together to implement 3R. So now several society come up, and FDA and EMA, they implemented this directive that we call 3R principle. Where exactly these 3R principle move it towards? They said, okay, we have ethics in using the animal for all in vitro assay, for drug metabolism, for toxicity prediction, for analyzing therapy. But if we say, okay, all those animals are replaced by some in vitro models to understand the toxicity, so then what we could do? So they start deciding, okay, let's take, if you want to take for skin, for eye models, for kidney, for brain, for liver, then they implemented several in vitro models where you have explant culture. Either you can take organ slices, you can take cell lines. Then the government said, and plus also all the scientists said, okay, if we take all those in vitro models, how we can carry out these experiments? Because you have to optimize the cell culture. Then you should know your cell culture is coming from which population. If you are taking care of, okay, drug discovery for Caucasian population, so you need a cell culture from Caucasian population. In the same manner, when you are looking for Chinese, Japanese, and Indian, so you need different population of cell culture. Then you need further selection of assay for toxicity evaluation. Then further on, you have to analyze whether your particular biomarker are expressed or not. Then those response relationship, and later on you have data interpretation and validation of that cell model. So you have to carry out several steps to understand your cell model can be used for drug metabolism and toxicity or not. When you will consider, okay, now have a selection of cell models. So what are the different steps you have to perform to understand that my data, which is responsible for providing toxicity assessment, is equivalent to animal model or not? Straight away, when the people wants to use first in vitro model for analyzing drug toxicity and metabolism, they directly go for first liver model. Liver as is the main organ for detoxification of drug, and it is one of the central core for a whole pathway we call like the drug which is going from your mouth, and after that it passed through the gut. Then further, there are major metabolism of drugs are going on in the liver, which we call as an exact location for the drug metabolism. Then the drug will pass for hepatobiliary cycle and then go to the kidney and then for elimination. So when you will consider the absorption, then further on distribution and metabolism and elimination. Look into inside when you will having drug which is in the liver, it's very quick reaction. So there are several reactive metabolites are generated where you have inactive drug converted into active drug. In the same manner, the active drug converted into inactive drug. So elimination. Once you have in drug metabolism formation of reactive metabolites, it generates toxicity. So that's why liver is the major organ which is responsible for drug metabolism. And then people start concentrating, okay, if we need to get more in vitro models, we should concentrate on liver because it's a central core for the drug metabolism and understanding toxicity. Then when you will look about different organ, then we understand, okay, we need then different biomarker to understand all those in vitro models. So selection of in vitro models became very important. then selection of which enzymatic expression, which biomarker in those in vitro model also became very important. So that's why we need good in vitro model for performing drug metabolism and toxicity of the drug. Look about all those enzymatic markers. When you will look about drug metabolism, you have a toxin, can be a drug, insecticides, and any other external molecule. We call either API or we consider like suppose it's a full formulation. And when it will go to the enzymatic metabolism, which is located in the drug, and it's dominated by CYP enzyme, cytochrome P450 enzyme. So when you are looking for in vitro model, you need information about what is the expression level of cytochrome P450 enzyme. When you will look into the roadmap, so you have a phase 1. So first the drugs goes to phase 1 metabolism, where you can see different CYP enzyme playing the major role. Here, the most important and dominant factor in CYP enzyme is CYP3A4 and CYP3A5, which compensate with the CYP3A4. So when you will look about marker expression in different in vitro model, so first and foremost is the CYP enzyme. Then you have other contribution. After the phase 1 metabolism, the drug goes to phase 2 metabolism, which is dominated by UGT. So you have a combination of phase 1 and phase 2 metabolism in liver, and then selection of in vitro model is based on these expression of enzymes. Now, when you will look on different in vitro model, it just a glimpse like how different strategy are implied. So when you will look about liver model, people concentrated on primary cells of liver, which is known as a hepatocyte. They said, okay, if we cannot take like animal model, so can we use externally use like B cell model like hepatocyte, where they can use from the human, they can use from the animal, which are different species are located. So people tried all the in vitro model. In the future, and also which is going on from last 5 years, iPSC growing very rapidly in the domain of drug metabolism and toxicity assessment. Another, as I said, like you can take a slices culture from the organ, little complicated because of ethics, and then plus also because of quality of tissues. So this is also one of the way. Another one, when we say, okay, cell line. So all the models have different advantage and disadvantage for using as a drug metabolism and drug toxicity assessment. What are those little information about advantage and disadvantage? I will use more probably information from primary human hepatocyte because this is coming directly from liver, and then also iPSC cells. And then later on some information about cell lines. What exactly the primary human hepatocyte? So here I will give more information. What exactly primary human hepatocyte? Primary human hepatocyte is the cell which are responsible for morphology of the liver. These are the major cells which are connected with each other and then forming a chain of reaction and connected with different portals, veins, arteries. When the drug which is entering into the liver, they further metabolized here and then in the cytoplasmic location. There they generated some particular metabolites, and these metabolites, which will be either uptake by different transporters here, then they are eliminated from the efflux transporter in the hepatobiliary cycle. So you can see when the drug is entering in the cytoplasmic location, it has to be go outside through hepatobiliary cycle, which is also goes through the bile. So when you have hepatocyte model, the hepatocyte model should express all those transporters, which are uptake transporters and the efflux transporters. And when you have any problem related to expression of those transporters, then you have a problem on understanding hepatotoxicity. Similarly, when you have fluctuation into the transporter expression based on the drug dose, you can evaluate, okay, this drug is toxic to the liver or not. So this is the way where primary human hepatocyte play major role for analyzing hepatotoxicity, for analyzing drug transporters and other activities. When you will look, okay, if we see, okay, we have a primary human hepatocyte and different cell lines, So what are the expression of enzymes you can look about for different transporters, phase 1 and phase 2, and here I giving example. So there is a HepG2. So HepG2 is one of the major cell line which used since long. And then after that, there is another cell line, HEPA-RG, which is also very good cell line for providing liver-based facility to understand drug metabolism and toxicity. Here you have primary human hepatocyte, and you can see liver slices. So it is major comparison for the primary human hepatocyte, where you can see there is a very good expression of different CYP enzyme and plus also expression of phase 1 and phase 2 enzyme. So this is, you can see here, very good expression of phase 1 CYP enzyme. Then you will look about like a phase 2 enzyme. So phase 2 enzymes are also fluctuating, but the expression is going there. Then you have an expression of transporters. So when you will look about primary human hepatocyte with the liver slices, which is directly from organ, and plus different cell lines, there is a constant expression of cytochrome P450 enzyme. And then after that, phase 2 enzymes and transporters. So this is the first way to look, okay, whether your in vitro model is able to express those enzymes which is required for drug metabolism and hepatotoxicity assessment. Now, where exactly iPSC play the role? So iPSC are now growing very widely since almost like 10 years, and then now they are very much implicated in drug metabolism and pharmacokinetics, and plus also hepatotoxicity assessment. So iPSC cells is known as induced pluripotent stem cells. Maybe most of the people are aware more. So like you can extract these iPSC cells directly from the patient, and you can generate somatic cells. Now, the major role come here about external factors, which we call environment. Based on those environment, you can say differentiation of those iPSC cells. Then another technology which will imply for the gene therapy. So either you can use when you have this patient-derived iPSC cells with the help of replacement of specific genes. So there are different technologies are available, like a CRISPR is majorly used. Then you can genetically correct those iPSC cells, and after the correction, you can use for the cell therapy. Now, when you will consider for drug metabolism assessment, same iPSC cells can go for in vitro differentiation, and that's here is the major things are coming, that your in vitro differentiation is very dependent on your external environment. And once you have this, then you can use for target identification and validation and the drug development. What is the benefit is coming by using iPSC as compared to when you will look about primary human hepatocyte, and then plus how they are becoming major power. So first and foremost, when you will look the production, so you can see that Yamanaka is one of the major like breakthrough about the development of iPSC cells, which can go for drug metabolism and which can go for therapies. And because of the power of iPSC cells, like you can have different facility which is not available with your primary human hepatocyte. When you will look about the iPSC cells, you can see when you will have a somatic cells, and then further on, you can reprogram iPSC, and then you will have a pluripotent stem cells, which can either self-renew also, and also produce different factors. Like for example, based on the transcription factors, it can differentiate into mesoderm, endoderm, and ectoderm. And this can migrate to your spatial cellular fate. So everything is dependent upon your external environment, because any fluctuation in external environment can divert the fate of your iPSC cell. That is the only drawback available in iPSC cells. Otherwise, like these iPSC cells are self-renewable, so it's a very good factory based on your optimized external environment. Plus also, when you will look about iPSC cells based on your primary human hepatocyte, they have very stable fate of the enzymatic expression. Now how look, so it's just informations like how the people in drug metabolism industry are producing primary human hepatocyte, considering so you have a donor. Now, as I said, like the major problem associated, how to obtain the donor, because the donor ethics is very important, and plus also here which population you are targeting. So this is the first important step here. Then after that, you will get several growth factors. You obtain your iPSC cells. Further on, with the external environment, you generate your induced primary derived human hepatocyte. These hepatocyte then you can use for drug metabolism and toxicity assessment. So always it depends on which donor you are using. I'm not going more into details about how drug industry are carried out different parameter, because it's a long series of parameters they have to understand at the premier stage for selection of donor. And then what is the environment you are providing, because if the environment is not good, then you will not get particular cell type. If you are looking for hepatocyte, if you are looking for any other cell type. So everything is dependent upon which donor you are using, which environmental factors you are providing to your iPSC cells. Here, when you look information about with different, so here this is a very good example. Now we compare, okay, with primary human hepatocyte, and then one of the cell line HEPA-RG, and then human induced primary human hepatocyte. You will look here into the expression level. Usually, like when you will look about primary human hepatocyte, in almost all the examples, when you are looking for drug metabolism, is usually you want to evaluate this for 7 days to 14 days. So when you will look about expression of different enzymes based on the different in vitro model, the primary human hepatocyte is widely used for metabolic pathway, xenobiotic metabolisms, which is about using the drug metabolism, and then plus carried out with some other metabolisms. So primary human hepatocyte is widely used for the xenobiotic metabolism. When you will look expression of cell line, so here you have different DNA replication enzymes are available, cell cycles enzymes are available, and they are widely good expressed here. They have some expression of the xenobiotic enzyme as well as, and depends upon like which enzymes you are looking for, like CYP3A4 is very widely expressed here. When you will look about human induced pluripotent stem cells based hepatocyte, so they are very good expression of some xenobiotic enzymes, but also they have very good expression of cancer related pathways. So there are different enzymatic expression are there, and then based on this expression, the drugs are selected to test on which in vitro model we can use for our drug metabolism and toxicity assessment. So this is a glimpse information like when we compare with primary human hepatocyte and cell lines. Now, one more important precautions here, like primary human hepatocyte, as we said, it depends on the donor. And similarly, iPSC cells is also depends on the donor. HEPA-RG cells is like donor independent. It's already selected by one donor, which was the female donor. So it's stable based on the donor variability, but primary human hepatocyte and human induced pluripotent stem cells hepatocyte is always depend on the donor. Here another example. So as we said, like people can pick up and then verify by different donors. So there is another example where primary human hepatocyte for expression of phase 1 enzymes, phase 2 enzymes, and all the transporters. So here when you will look about the primary human hepatocyte, they are able to express most of the CYP enzyme. In HEPA-RG, so there is also expression of other enzymes, and then there is an expression of CYP3A5 in case of iPSC cells, and there is an expression of CYP3A4. So you can see that primary human hepatocyte is always a very good gold standard and also recommended by different agencies. But then people are selecting different in vitro models. Why? Because of problem associated with the in vitro models, because of the problem associated always with the stability of enzymes, and that is very important to understand here. In case of phase 2 enzymes, you can see iPSC are expressing most of the important enzymes for the phase 2, and which are not good expressed in primary human hepatocyte, but are good expressed in the human pluripotent stem cells hepatocyte. So in the same other way for the transporter, now you have to select which transporter you are looking, and based on the expression of those transporter, you have to select in vitro model. And that's why I was talking about, like, when the people we are talking with the pharmaceutical industry to implement 3R principle, they said it's a little long way. It's still controversial, because when we want to replace the animal models, then we have to select different in vitro models. And that's why we say they need battery of in vitro models. Now you cannot select only one models, but you need battery of in vitro models to perform your drug metabolism and toxicity assessment. Now, when you will look about another donor, which is providing iPSC for enzyme metabolic rate, and we discovered, like, okay, that in iPSC, based on number of days, they have a very good metabolic rate for CYP3A4 enzyme, which is providing 50 to 60% drug metabolism. And so CYP3A4 is very stable in case of iPSC coming from donor 1 and then donor 2, which is not quite relevant with other cell models. So here you can see that selection of donor became very important for understanding drug metabolism. These iPSC cells, when you will look and compare whether they are relevant to understand toxicity assessment, because when you are looking for the cell model, it should give your directly how the drug toxicity is also implemented. Here you can see one of the, there's just one biomarker I am giving, like albumin secretion. When you are selecting the cell model, you are selecting also different biomarkers which should express to consider, okay, this cell model, and the result can be extrapolate at the in vivo level. So one of the biomarker we consider albumin secretion. And when you have a drug toxicity assessment, so you should understand, okay, these biomarker will fluctuate. As I said, like a treatment of the drug is also responsible for fluctuation if the phase 1, phase 2, and other transporter plus also biomarkers. So here, this is giving good example here that how iPSC beautifully giving information about drug toxicity. So in this example, the aspirin, which is non-toxic, some particular dose, and diclofenac, which is very toxic. So they tested these two drugs on iPSC human hepatocyte, and then they found that aspirin treatment is providing continuous expression of albumin. But when they used diclofenac and they increased the dose, and they found their secretion of albumin decreased. It means the iPSC cells are beautifully expressing the dose-based toxicity assessment of different drugs. In the second example, this is the 4 hours of iPSC seeding. So you can see it's a beautiful expression, a beautiful representation of iPSC hepatocyte. And you can see it's more deep inside at 1 week of culture. When you will look about the treatment of aspirin, as you can see, this is non-toxic and some dose. So you can see it's like a maintaining like a morphology as compared to when you will use the diclofenac, where you can see this kind of fainting stuff. So means the toxicity is very easily able to predict by iPSC cells. Here it's an example of approximately 50 drugs, which is to understand whether iPSC cells are very sensitive as compared to primary human hepatocyte or not. Now the cells, when they grow in a co-culture mode, so co-culture sometimes is useful to increase the expression of enzymes. So here they use like a normal primary human hepatocyte with the co-culture of 3T3 J-tumorin embryonic fibroblast. When you will look about these three, you can see here, this is the somatic cells hepatocyte, and then primary human hepatocyte with the co-culture, and this is induced primary human hepatocyte with from iPSC. When you will look about the sensitivity, so in somatic cells hepatocyte, you can see here that sensitivity is quite low. When you will compare the primary human hepatocyte and iPSC, the sensitivity is quite high. And then specificity is also quite relevant. So means iPSC can be a very good model for understanding drug toxicity. it's given an example which we worked together with one industry before. So it's a molecular device, and they implemented dedicatedly iPSC based method. So you can use either in different plating methods, which is for high throughput, low throughput, 24 well plate, 96 well plate, or 384 well plate. And this is to measure hepatotoxicity. When you can use iPSC cells for these hepatotoxicity assessment, and then after that they analyze like, okay, how different modules of toxicity can be useful. I'm just giving an example how they beautifully used this for just straight away when people are using viability analysis. So here you can see when the iPSC untreated control. So here you are giving treatment of drug amiodarone at 72 hours. When you will look, this is the untreated control using the dye calcium AM. Live cells, they will pick up the dye. And then when you have a dead cell, so then dye is not there. So when you will look about here in the untreated control of iPSC cells, that you can see that beautifully representing the live cells. And when you will provide the dye, then you can see that most of the cells are retaining the dye. When you will have a treatment of drugs, you can see the cells are dead cells are prone most as compared to live cells, and you can easily see the dye is not there. So this kind of viability assay is very beautifully presented in iPSC cells. The another factor which you can look about the drug toxicity assessment is mitochondrial toxicity. So mitochondrial is the area where the lots of oxygen consumption is there, ATP production is there. And then when you will look about the drug treatment, you are majorly looking how this cell membrane depolarization is there. Because when you will have a disruption of mitochondria, it's providing the fact that the drug is toxic to your cell, and then the cell will not survive longer. So mitochondrial toxicity is also one of the factor for understanding drug toxicity. Here in this example, there is no treatment of any drug. So here giving example of valinomycin. This is very beautiful antibiotic to understand the toxicity among different in vitro models. So valinomycin, which is also toxic, and in 60 minutes it produced the toxicity by using the dye JC10 dye. So when you will have using this dye and the live very good mitochondria, they retain the dye and they become like a red and orange color. When you will have treatment of this drug based on the dose, this mitochondrial membrane become depolarized, and because of this depolarization, the dye which is inside in the live and intact mitochondria then start leaking into the cytoplasm. And because of this leaking, you can understand, okay, which is the dose which is providing major depolarization of mitochondria. So the first and foremost was straight away cell viability. The another factor for drug metabolism and toxicity assessment is enzymatic expression. The one of the endpoint could be for mitochondrial toxicity. So here you can see that how iPSC cells are beautifully presented the mitochondrial toxicity. When you will combine either all the toxicity together, in drug industry measurement, we call this as a multi-parametric hepatotoxicity, where now you are not looking for one endpoint. You are looking for different endpoints, means you will combine your cell viability, you can combine how many number of viability cells are there, what are the membrane depolarization of mitochondria, plus also how the cell surface area will change based on the treatment of the drug. So this kind of endpoint measurement, multiple endpoint measurement, is known as a multi-parametric hepatotoxicity evaluation. And iPSC cells become very widely used in high throughput manner, where all the different toxicity can be assessed at the same level. So here you can see, like when you will look calcinemi, it's for the live cells, it's for nuclear count, how many live and dead cells are there, cell surface area measurement, how the drug is changing the cell surface area, and then to understand apoptosis. So this was the claims like how iPSC cells can be used in the drug industry. The major important part in that case about how is the stability of your enzymatic expression in the iPSC? How is the stability of your cell model? And then how beautifully your cell model is representing your endpoints. So that is a very important part for selection of in vitro model. And according to analysis, like primary human hepatocyte is always straight away is a gold standard, but it's very difficult to obtain measurement, to obtain ethics, to obtain donor, to obtain the good quality of cells. It's a little complicated process. So based on that, iPSC is becoming more prone for analyzing those toxicity. Now from almost now most of the people are more aware here, and then we are expert here as compared to me, which is about iPSC are now used for the cell therapy as well as. So where exactly iPSC can be used for the cell therapy? When we look about different examples about how iPSC can be used, so we look about like a cardiovascular disease, neurological disease, metabolic disorders. Apart from that, like I looked into the lecture also, and I'm really happy to learn about that bone replacement could be useful. So iPSC cells are nowadays not only used for the drug metabolism or for drug toxicity prediction, but also for directly cell therapy. Yes, depends upon the ethics of different countries. So you have a ban to use those for therapy, but is like beautifully used in India, which is also very good example. In European commissions, when we work with regulatory authorities, so they have a partially, okay, some particular therapies you can use, but some particular therapies are completely banned. So you have ethics associated at different location of territory. When you will look about what are these ethics associated, so when you will are selecting the somatic cells, so I'm not talking about here now only for India, but I'm talking about the globally, like when you will look the iPSC cells for the cell therapy, where you have to obtain consent from the cell donor and donor privacy. And this is also implemented when you are selecting iPSC cells as an in vitro model. You have to take a donor consent and the donor privacy. Then the problem is coming for the intellectual property and the manufacturing. So ethical status is very important for the country for related to cell therapy. Then after that, when you are looking for, okay, differentiation of iPSC, then it's become very important which environment you are providing, because once you are, you have to optimize this very carefully, because if your environment is very eagerly fluctuating, then this is creating a problem, because then the iPSC cells can create cancers, because you can see in the enzymatic expression, so they are used in cell cancer pathway, but also more prone to the canceric origin into the patient. So that's why you need high quality of iPSC clones, and plus also safety of reprogramming. So you should optimize these steps very carefully. Then later on, other ethical donor privacy and intellectual property. Similarly, iPSC differentiation. So same quality is very important in case of iPSC. And then later on, how you are obtaining. So this is also selection. It is autologous or it is allologous. So there are different kind of ethics and there are kind of problem associated with each step. That's why European Commission put very strict warning to use in some spatial cell therapies and to use iPSC cells. Here, just an example how iPSC cells, like in earlier case, when it will go for drug discovery. So similarly here, when you will look about an example of your diabetic patients, you obtain iPSC cells from the diabetic patients here, and then you obtain somatic cells by providing several reprogramming factors, transcription factors, external environment factors. Then you can go for diabetic patient specific iPSC cells. Further on, based on the gene correction, you have a corrected iPSC. So where you have expression of insulin secreting beta cells, and then you can transplant into the patient. And if you want to go for drug discovery, you differentiate, you will develop insulin secreting beta cells. Then you can for high throughput drug screening and plus also drug pathogenesis and drug toxicity assessment. Here, just an example here, when you will obtain the iPSC cell from embryonic stem cells and also maybe from fertilized egg, based on the different transcription factors, you are able to produce beta cells. So iPSC cells has a power to use in therapy, but as we said, like it's based on your environmental factor, which decide the fate that you are producing very good iPSC cells for the therapy, and also you can use for the drug screening. This is one of the beautiful example when I was looking, okay, the iPSC could be useful for treatment of skin disease or not. And just information to get, okay, iPSC cells, okay, dedicatedly used for the liver metabolism, but now are they able to transfer into different therapies? And then the beautiful example came into here, where the patient was treated for the dystrophic epidermolysis bullosa, where we use the requirement where you have a like invisible collagen layer between two sides of the skin. And then when you are able to provide the iPSC treatment, then it is able to provide like a collagen 7, and then you can see the beautiful formation of collagen layer here. And here in the patient, where you have a mosaic formation, but when you will provide iPSC treatment, the skin formation is there. Example, you take a fibroblast, you have a diseased iPSC, later on you have an affected keratinocyte, then you can go for drug screening in the same way, or you go for gene correction, and then you can go for cell replacement therapy. So this is another example where iPSC used for the skin therapy. I saw most applications also for iPSC in neurological disorders. In this example, the patient was treated for the stroke injured brain. So here you have a fibroblast and blastocyst, which are the origin for iPSC cells. And these iPSC cells are later formation of neurons and glial cells. And then injection of these two are able to form astrocyte network. And that was the beautiful example, because most of the times when we are considering like drug metabolism and drug pharmacokinetics, we are always getting problem about discovery for brains because of this blood-brain barrier. So this is creating major problem, but now this kind of iPSC treatment and then directly tissues which are the iPSC cells originated from here and to generation of these can be very good example for drug screening. Now, when we'll get a little bit insight about autologous and analogous, this is only the problem which could be here. For example, in autologous, where you have no need for immunosuppression and ethically non-controversial, but also, as we said, like when you have these kind of neuronal disease, most of the iPSC are coming from old age patients. So in that case, you have to just tackle these kind of problems. When you are looking for allogenic, you need to provide some kind of immunosuppression, and people are testing this immunosuppression externally as well as ethically non-controversial. But as we said, like external environment becoming major problem. So you have to match always the HLA haplotype, which can create a problem for the fate of the iPSC cells. In the same manner, when you will look about like genetic manipulation, so this is also one of the major problem when you are looking for brain treatment, because in case of drug, it's really irrelevant to test in vitro models. Here is another very good example how iPSC cells are used into for making spinal injury. So here you have like making spinal cord cell type, so which is the specific neural progenitor cells obtained from the iPSC. So here the people are used iPSC cells derived from the patient, and then they degenerated the forebrain, and then to understand whether the iPSC cells are able to make marker for those kind of forebrain, and later on, then some particular stages are iPSC cells able to obtain a spinal cord marker. So here, by providing good transcription factors, they are able to provide like forebrain formation within 6 days, and also a spinal cord cell type in 12 days. So you can see, based on this environmental factor, they are easily able to define, okay, where at which stage the iPSC is able to go for the forebrain, so which is also environmental dependent, and then further on how these iPSC cells are able to make a spinal cord, and that can be used for the therapy. Here example, the expression of different biomarker with the formation of forebrain, and then HOXB4, which is the biomarker for a spinal cord, HOXC4. So here is the example for immunocytometry, where providing information that how the biomarker are expressed during the spinal cord age and at the forebrain age. So this is a good example like how iPSC can overcome those kind of problems which is associated and with using in vitro model for those kind of drug analysis and the toxicity, and also for therapy, which is not possible by in vitro model. So iPSC has a big power, which we consider now in the field of drug metabolism and toxicity assessment, and also the same iPSC which could be derived for analyzing drug metabolism, drug screening for therapy. Every step have limitations, but this is the future where we can look about. So when you will look about advantage of iPSC, so iPSC can be generated from patient. Once you will generate it from patient, then you can easily propagate and also provide a renewable sources of the cells. It can be differentiated into cell types based on what environmental factor you are providing. And that's why you have to optimize this step, which is looking for either you want to look for therapy or you are looking for drug screening. And that's why these cells differentiated from iPSC can be used for disease modeling or drug screening. The disease causing mutation can be corrected in iPSC using different gene editing technology. Now this is quite successful. Still so much of work has to do, but the successful are still there. Then you can develop different cell types, and now at the end, generation of iPSC involves the use of adult cells, which is bypassing the ethical concern associated with the embryonic stem cells. But still, different territory have different ethical point of obtaining tissues from the donor, how to treat with the patient, how to take tissues from the patient. So there are different ethics are still associated, which we have to take care based on the territory. But there are still limitations associated with the iPSC. Like iPSC can acquire genetic mutation during reprogramming. So means that's why the environmental optimization is very important. Undifferentiated iPSC, if transferred to a patient, could form tumor. Yes, because if any reprogramming factor which is not able to provide fully differentiation of iPSC, that can cause a problem at the therapeutic stage. And as you know very well, when you are looking differentiation of those iPSC cells, then it's a very strict and then very, you can say, stringent protocol, like which kind of genes you want to mutate, which kind of technology is there. There is a specific selection or not, which kind of transcription factors you are using, which kind of other environmental factors you are using. So this is the trivial task for using iPSC cells, apart from which particular donor you are selecting. So thank you very much. So I give like how the iPSC can be used for drug metabolism and toxicity, and example how iPSC are able to use for therapy. And then I'm ready for the questions.
Thank you, Dr. Ashwin Sharma, for your inciting, stimulating, extensive talk on iPSC, induced pluripotent stem cell, what is started by Shime Yamanaka of Kyoto University, Japan. I think this field of research started with Yamanaka, and he first published in 2006. Yeah. Then he got Nobel Prize in 2012, and now 2020. Within span of, I think, 15 years, that much of applicability in this field of induced pluripotent stem cell is Very, very appreciative. For your talk, I think you have concentrated more on preclinical trial, mostly on toxicity studies for this iPSC cell line, iPSC cell line for extensively, and mostly it's applicable in the cell therapy, stem cell therapy mostly. So it is a very illustrative and extensive talk. Thank you very much for this talk. Now, Madam Majumala ma'am.
Yes, sir.
Collect the questions from the
Yes, sir.
audience.
Yes, sir. Thank you, sir. Thank you, Dr. Ashwin Sharma, sir, for a wonderful presentation on iPSC for therapy. And not only that, highlighting the importance of selection of in vitro models. Now, there are a few questions from the audience. The first question is from Dr. Vivek Bhatia. She has posted that since iPSC differentiate into their fetal counterparts, how will the data be correlated to hepatocytes in adult patients?
So what you can look about, because it's usually you can say like it's a fetal part, but when you will look about the adult counterpart, it's usually independent of your first primer selection. So that's not dependent on that, but you are dependent on, that's why people just only by iPSC, you cannot compare. You have to obtain some comparison with like a primary human hepatocyte. And everything is dependent on how the enzymatic expression are there. So this is the one most important step you have to look about. Like if you are looking for adult counterpart, so what is the enzymatic expression at that level? This is very much important. That, and then plus also when you are looking for drug metabolism and toxicity assessment, how the treatment of drug is influencing those enzymatic expression, because they are responsible at different stages. When you will look about the fetal part and when you are looking for the adult part, they don't have the same enzymatic expression. This is the one. Second, what I'm talking about, when you are looking about adult enzymatic expression, important which donor and which population you are looking for, because that's true, it cannot compensate all the population. Like if you are looking for this, for example, we work for Caucasian. If you are looking for Caucasian, so you should match this adult counterpart with your Caucasian. So it's usually independent on your fetal where it is obtained, but it is dependent on the stage of enzymatic expression, how it is correlating with your concerned population of adults.
So I hope, Dr. Vivek Bhati, you got the reply. And there is one more question from a student, Mr. Adish Majumdar. In fact, he started with, it was really a great lecture, sir. Thank you for sharing your insight and knowledge. I just want to ask that, suppose we are trying to understand the effect of a drug on kidney diseases, in which case the toxicity understanding pathway might be already affected due to the damage caused by the disease on the kidney. So in that case, how is the toxicity of the drug estimated?
Sorry, I didn't understand the middle part, what they were asking.
Yeah, if we are trying to understand the effect of a drug on kidney disease, in which case the toxicity understanding pathway might be already affected due to the damage caused to the kidney. So in that case, how is the toxicity of the drug estimated?
Okay. So when you will look about, so now there is the question, that's why I'm asking, it's mixed with the two part. One, either you are using the section of the kidney or you are using the in vitro model. Because kidney is the latest in vitro model which people are looking for. Before that, people are just concentrated on the drug toxicity. So means at the liver stage. Now people start concentrating on the elimination stage, means how the kidney is also playing the role with combination of liver. Now in vitro model, we call those kind of cells which are used, we call renal proximal tubular cells. We call this is known as a RPTC cells. So when they are talking about proximal, so means in kidney, you have different location, means different area of kidney, proximal area, caudal area. Still, people are having so much of difficulty to obtain those in vitro models. So that's why they have very good in vitro model for RPTC cells, so proximal area. So currently, when you will look about the toxicity and the drug which is already caused the toxicity, and if you will look about that section, means like a organ slices, that is very difficult, because usually you have to analyze based on the in vitro model, and in vitro model coming from the diseased organ which is already got toxicity by the drug, it's really difficult. So we have to select chemotherapy, how much kidney was damaged, which area of kidney was damaged, at which dose it was damaged. Then after that, you have to select, okay, can we obtain in vitro model for analyzing kidney toxicity or not? So if kidney, which is already, you can say, got toxicity from the drug itself, so getting in vitro model, it's really difficult because it's based on the area dependent, which area of in vitro model you are using. But if you have an in vitro model, proximal area is one of the good in vitro model you can use for kidney toxicity assessment. So organ selection, what is the quality of organ, which I said like a first part, donor selection and organ selection. So if your organ is already damaged, you cannot do in vitro model assessment. Then only you have to look imaging. There are then several techniques are available when you have a toxicity by the drug. You can do imaging of the kidney organ to understand what particular area was damaged by the drug. Then you cannot use in vitro model, then you have to use imaging model. We call this is like an artificial intelligence based imaging model of the organ.
Yes, sir.
Manjuvala ma'am, any more further?
Yes.
If not,
Hi, sorry, I'm Bhavna here. I had one question, if I can ask.
Yes,
ma'am, go ahead.
Hi, Ashwini. Very nice talk.
Yeah, hello, ma'am.
Hi. So I was just wondering, in your liver 2D cultures, I mean, do you make all cell types which are there in the liver? And do you then think that if 2D cultures are not, because sometimes you would need the 3D architecture as well to mimic the organ, particularly if you're not using animal model. So then is the plan that you also would like to, develop some organoid models, liver organoid models to do these drug toxicity, and perhaps they could be better models in testing for drugs and their toxicity?
So first of all, good morning, ma'am. I saw you after 20 years.
Yes, we've been after a long time.
Yeah, so
thanks to this conference.
Then before this IPSL was discovered.
Yeah, so I can tell you now, it's a very good question, which people was not thinking 5 years before. 5 years before, many things are depends on the 2D monolayer culture. Then it's transferred to the sandwich culture. So 2D and sandwich was occupying the whole drug assessment criteria, regulatory guidelines. Since 5 years, people start moving, okay, we should move towards the 3D culture. Now the question is coming, when they want to make 3D culture, then they have to understand which model they use. For example, when you will say, because why they want to use 3D culture, just I can give you the example as advantage of as compared to 2D, more stability of enzyme. This is one. Less number of cells required for assessment of the drug. This is second. Third, it's quite realistic if you will use 3D culture, like a close to the liver model as compared to 2D model. So that's why now you can see 3D bioprinting technology is coming. You can make a scaffold of 3D liver culture. So since 5 years, yes, definitely, the people are moving towards 2D to 3D culture. Only problem is coming, how much 2D culture is stable, like I said, based on the enzymatic expression. Because when you will look about the liver is like, you have cells, you have biles, and then after that you have a portal vein. So when you will make it 3D culture, When you will make 3D culture, if you don't optimize the number of cells, then your inside core become dead. So an outside could be live because you have cluster of so much of cells. People tried 50,000 of cells. People tried 20,000 of cells. So depends on the cell model. First of all, you should know which cell model you are using, like either generating from primary human hepatocyte or from. So 3D culture, yes, it's a very good example. Drug industries are also still they are not successful so much to use 3D culture for high throughput screening of drugs on 384 plate. They are trying, but still toxicity assessment is problem because they have to analyze each viability of cells, which is not so easy. But yes, this is one of the best example which can go for a good assessment of drug. HEPA-AGI is one of the cell model which are successfully used for the 3D culture, and they optimize the drugs, and then it also used by different industries like Sanofi. They are using for drug screening. And then iPSC cells, I also saw some research. People make human liver organoid from making transcription factors. So 3D culture, yes, it's a very good example to use now. But always compare with 2D. This is my recommendation because FDA guideline is still are more based on the 2D.
Thank you so much. Thanks.
Dr. Ashwini Sharma, can I ask a question?
Yeah, exactly.
Would you want to comment on the stromal component when you talk about iPSCs in terms of toxicity testing? Would the results be any different if you have paracrine factors playing a role? In the context of what Madam was asking, 3D and cultures, and you want to comment on that, and keeping in mind Dr. Deepa Bhattia's lecture this morning.
So, like when you will talk about, yes, things will be, I can say, yes, it will different 30 to 40%. When you will use the 2D culture, when you will use the 3D culture in case of either stromal, because we evaluated and plus also with the partners earlier, and plus also I saw some research also. When you will have a 2D culture, the stability of the enzyme or the tissues is become longer. First, number of days, means like in case of you will say 2D culture, based on which toxicity you are using, because you don't know if you are using new drugs, it's highly toxic, it's less toxic. So based on that, the drug can produce toxicity within 6 hours. Some drug can produce toxicity in, let's say, 12 hours. So you will look about systemic toxicity and chronic toxicity. And if your cell model is not able to bear based on the environmental factor and the toxicity, it will be quickly die, and then after that you cannot access. So that's why people start moving for 3D, because in that case you have a more stability of the tissues, you have a more stability of enzyme, and then after that you can evaluate longer the toxicity of the drug at different doses. So that's why a 3D model, which are even more problematic, for example, stroma, because in case of you don't have very good in vitro models, in that case, 3D model can play a major role because of stability of tissues, because of stability of enzymes, and then plus on, you can analyze different toxicity for longer duration.
But what about the extrapolation to the human situation in the context of inter-individual differences in metabolic capabilities? In fact, there are papers saying even in the mouse strains, inbred mouse strains exhibit remarkable differences in abilities to metabolize. So,
no, that's
please go ahead. Please.
No, that's, you are completely true in that respect. So that's why I was talking about like individual variability. That's why I said, like, when you are even looking for primary cell, and that's the controversial point. Like I discussed so I'm working now with OECD guideline people, which used to provide. Before that, for providing different cell models, so I worked with the FDA. We exchanged some meetings with them, but I attended also in India, so FDA meetings together. So what you said, it's exactly the question is coming. Why we want to replace animals? Because even the animal models have a problem for extrapolation of results to the human level. And now you said to use in vitro model. In vitro model, another drawback, as I said, you have donor specificity. Then after that, you have inter-individual variability of enzymatic expression. So based on that, I don't know whether people know about this technology or not. When you are using the models, then you cannot use only one primary cell obtained from one donor. What you will do now, then the another strategy they used, you have to pool several models together. So we call this process as a pooling, because in one model, you can have no good enzymatic expression. And if you will obtain the result from that just one cell type, and plus also you can say that coming from one donor, then the results are not extrapolated so easily, because what you said, the variability of enzymatic expression, plus also sometimes the enzyme are not good. And this, in that case, you need to perform pooling. So you will take several cells coming from one donor and you pool them. So pooling can go for 50, pooling can go for the 200 cells to make a uniform enzymatic expression. Then we consider, okay, now an FDA strictly said about which kind of, how many number of poolings you have to perform to have good uniform expression of enzyme. Then you extrapolate the result from the pooling cell models. This is the one way they try to solve the problem. Another one, yes, you cannot use the same drug coming from or tested on the Caucasian population donor on the Chinese. No. We analyze the enzymatic, for example, CYP3A5, it's not the same as Chinese population as a Caucasian population. So if you want to do the drug for Chinese, you have to perform same process for the Chinese, which we also recommend for Indian. So it's not the same population, and then but most of the times we follow the FDA guidelines, but it's also very strict sometimes to use a local population for performing drug metabolism and toxicity analysis. So currently extrapolation can be verified only by pooling step for uniform enzymatic expression.
Thank you, doctor. Thank you.
Thank you, sir.
My query, Dr. Ashwin, that any of this diabetes, skin grafting, and your spinal cord injury has got FDA approval for therapy?
No, this kind of, no, that's why I said like a therapeutic purpose is still long to go. Long to go, because so many ethics are there. That's why we were having discussion like how it is easy to India in stem cells therapy, how it is difficult to do in countries like Europe. In Europe, even when you will consider the, okay, I don't want to be biased, like France, it's very strict. What you can do in Germany, you cannot do in France. So it's based on the country's local ethical which come play. Some therapies are easy to implement, but many therapies are not possible. So it's a little long way to go.
So let me end with thank from the VIT University School of Biosciences and this International Conference of this stem cell technology. I thank you profusely for this illustrative, stimulating, and good reply for all the questions. Thank you very much.
Yeah,
Madam,
thank you very much.
Yes, sir. Thank you, Dr. Ashwini Sharma. We hope we will meet you in VIT campus very soon. Yeah.
Thank you. I got in touch with you through Dr. Sachin, and through Dr. Ashwini, we came to know about Dr. Kannan Krishnamurthy, and through Dr. Kannan Krishnamurthy, we came to know about Dr. Bhavna Muralidharan. So it's been a wonderful way to network, and hopefully we'll do something more collaborative in the future. And when the pandemic situation resolves, I hope you will come in person to VIT. Thank you, sir.
Thank you very much. Thank you. Thank you all.
Thank you, sir. Thank you, sir. So with this, we move to the next session. Next speaker of the day, Dr. Bhavna Muralidharan. I request Dr. Khora sir to chair the session and to introduce the speaker. Let me just share the screen.
Thank you, ma'am. Thank you.
Ma'am, Bhavna ma'am, 1 second. I'll just share the screen for the introduction.
Sorry about that.
Yeah, it's okay. Thank you.
Ma'am, can you enlarge a little bit?
Yes.
So it is my pleasure to introduce the next speaker, Dr. Bhavana Muralidharan, Assistant Investigator, Intermediate Fellow, DBT Wellcome Trust, India Alliance. And Dr. Muralidharan has started her career in PhD understanding the translational regulation of insulin in the body under the guidance of Dr. Vasudevan Seshadri at NCCS Pune. She did her postdoctoral work at Wellcome Trust, DBT, India Alliance Early Career Fellow at TRFRI, Bombay, in Professor Subha Tole's lab, where she focused on key regulator of developing neocortex. She did her postdoctoral work at UK Dementia Research Institute in Professor Andrea Isaacs' lab, focusing and understanding the disease mechanisms of amyotrophic lateral sclerosis, ALS, and frontotemporal dementia, FTD, using the iPSC-derived neuron as a human in vitro diseases model system. In her own lab now at Wellcome Trust, DBT India Alliance Intermediate Career Fellow, she combined the mouse genetics and human iPS as a model systems to tease out the molecular mechanism of cerebral cortex developing in health and diseases. So with this word, I welcome Dr. Bhavana to continue the session for her from beginning her talk.
Thank you, sir.
Thank you, Dr. Samantha, for the nice introduction. And thank you, Dr. Suresh and Dr. Manjubala, for extending this invitation to me for this conference. I would say it's a very important topic of discussion, stem cells and their usage for drug discovery and therapy. And after Ashwini's talk on how iPSCs are used for drug discovery and therapy, I will actually take you all back to a bit of more fundamental or basic biology, where I would like to tell you that how fundamentally we understand how these stem cells work. Because if we do not understand how stem cells work or what molecular mechanisms cause stem cells to make the different cell types and mice, the system I'm going to talk to you today is about brain, and particularly a region in the brain called the cerebral cortex. So if we don't understand how the cell types come about in a normal system fundamentally, or what are the molecular mechanisms, then perturbing iPSCs to make more of such neural cell types will not be possible. So thus, studies like this are very important to understand what happens in a normal scenario. So as I said, the system I work on is understanding brain development, and one of the regions which is the higher order, which does all the higher order functions in the brain, is the cerebral cortex. It's the crown which sits on the top of your head. It is involved in doing all your higher order functions, namely, language, thinking, memory, consciousness, movement, sensory movement control, and all of that. And today in my talk, I'll be telling you how molecularly transcription factors and chromatin modifiers interact with one another to bring about this structure. So this is the cerebral cortex, and as I said, it is the crowning glory in the brain, and in the humans, it has expanded so much that the entire cerebral cortex has also not been able to contain it inside the brain. It has folded into these gyri and sulci, the way we see the human brain, to sort of accommodate the entire expansion in the small region of the skull. The human brain has about 86 billion neurons and 3 times more, a trillion kind of glia. So the neurons are the basic cell type which are found in the brain. These are the firing units, and whereas the glia are the sort of support cells which support these neurons, and they have many functions in the brain. So the human brains has trillions and trillions of cells of different kinds. And what is intriguing is the fact that these diverse numbers are all made from scratch, from a common pool of stem cells. And this entire process is quite dynamic. And as I said, our understanding of this is we're still understanding molecularly how all these different kinds of neurons and glia are formed in the developing cerebral cortex. I hope everybody is able to see my screen and hear me properly.
Yes, screen
Yeah,
is visible. Yes.
Okay, great. I'll just continue then.
Yeah.
Right. So building the neural network consists of several steps, and the very first step which starts this process of building the neural network is that the entire brain comes from a common pool of cells called the stem cells, and we call them the progenitors of the brain. So these stem cells, they divide, they proliferate. They divide in such a way that they generate enough numbers of these stem cells so that they can sustain the entire development. So these stem cells divide, and you can see the stem cells here, they proliferate and they divide. Then these stem cells, divide by a mode of division called symmetric division. So one stem cell will divide into 2 cells, which also will be stem cells. So that's how you increase your own numbers so that you can sustain the entire process of development or make the entire brain. So the entire brain comes from these stem cells. So you should have enough number. So these divide by symmetric divisions and make enough numbers. The next step in this process in building the neural network is that these very same stem cells, they switch from a mode of division, which is symmetric, to asymmetric. So where a stem cell was dividing and making 2 stem cells, it will now divide asymmetrically. One of them will be a stem cell, and the other will go on to make a differentiated progeny, which will either be a neuron or a glia or different kinds of cell types which we see in the brain. So this process is called fate specification, whereas you either generate a neuron or a glia. So that's the second step in the formation of the neural network. Followed by this, because the neurons, I think the fundamental property of the neurons are, of course, firing and that connectivity. The entire brain is connected from one region to the another. And all this is possible because of the outgrowth of axons from these neurons. So the next step in the formation of the neural network is basically the outgrowth of these neurons and formation of connections. So what starts as a stem cell makes a neuron, which then outgrows all these dendrites and axons, which then connect with one another. And that's how you actually lead to the formation of a functional brain, as you can see here. And that leads to the formation of a functional network. Needless to say, any one of these steps going wrong leads to a range of neurodevelopmental disorders. For instance, if you have mistakes in stem cell division, you end up having a smaller brain, a disorder which is called as microcephaly. You have perturbations in formations of neuron or in axon outgrowth. You have a range of neurodevelopmental disorders which begin from autism spectrum disorders to schizophrenia to bipolar, and these are some of the disorders which happen because there are mistakes fundamentally in one of these steps which go wrong during development. So thus it is important to study these steps to understand what happens in a normal brain so that we can then understand what goes wrong during neural developmental disorders. So as I was saying, that a hallmark feature of this entire process is these stem cells, which we call them as the cortical stem cells, or we call them as the progenitors. they give rise to all the different kind of neurons and glia, and this happens over a time course of development. In case of humans, it is the 9 months of gestation, and the development continues even after gestation, that postnatally when the child is born, the brain keeps developing. So when we look at what sort of regulates this process, I mean, it's the same stem cell, then what is happening inside the stem cell that is telling the stem cell that, you know, time X in development, you need to make a neuron. Time Y in development, you need to start switching and making a different kind of neuron. I mean, all these colors are different kinds of neurons which are being made in the
I think we're not able to hear Dr. Kora, Dr. Manjubala.
Disconnected, I think.
Dr. Manjubala. Dr. Manjubala.
Dr. Bhavana? I think
it was a problem of internet.
Yeah.
Suddenly audio gone.
I think she will log back on. I think she's logging out and logging back in, I think.
Manjubala ma'am?
Sorry, I think
something disconnected. Some interruption was it. Bhavana ma'am, we continue now. Now we continue.
Can I continue? I'm so sorry about this.
Okay, please continue.
I was just speaking and I just lost my connection. I couldn't figure out what happened.
No problem, madam. Please continue.
No
yeah.
Please.
So I was saying that what happens during development is that so the same stem cells give rise to the different kinds of neurons, and then they start producing different kinds of glia. What is that fundamental feature in these progenitors or these cortical stem cells which regulate this process? And that is chromatin regulation. And what I mean by chromatin regulation is that the interaction between transcription factors and epigenetic or chromatin factors, which then leads to, which come together at very specific sites on the genome, which are called promoters or enhancers of a particular gene. They then regulate the expression of the gene, and they do so by actually making a lot of changes in the marks, histone marks. Histone marks are basically, active or repressive marks which are present on the histone to enable the chromatin to open or close, so that the gene expression can be either activated or repressed. So these interactions change the epigenetic modifications around this loci, which then regulate the gene expression, which then leads to formation of a protein. And the formation of this protein determines what sort of a neuron will be made. And that then determines how you go on to make a functional brain. So today in my talk, I'm going to tell you this connection, which starts very molecularly at the level of transcription factor and chromatin factors, how it regulates gene expression, and that how this gene expression determines what kind of a neuron is being made, and that determines what sort of a brain will be made. So this sort of hypothesis has been been there, and one of the first proponents of this epigenetic landscape was Waddington, and he proposed this as early as in 1957, where he said that this progenitor or the stem cell here has, there is sort of an epigenetic landscape, or there is this crests and troughs which this progenitor or the stem cell sees, which could be changes in methylation, which could be changes in acetylation. So some of these methylation, acetylation are those marks which I was talking about, which these epigenetic modifiers put on those, different genomic loci. So depending on what sort of methylation or acetylation pattern or phosphorylation this stem cell sees, as it rolls down this landscape, it will either form a cell of state A or it will form a cell of state B depending on the time. So this axis here shows the cell differentiation. So the cell differentiation will keep changing across time in development. It will form A, B, C, D, depending on the sort of epigenetic landscape it sees. Similarly, as time progresses in development, the chromatin also keeps compacting to enable all of these processes to happen. So as I was saying that our key to understanding what happens in neurodevelopmental disorders comes from understanding this chromatin regulation. The reason being that a whole range of mutations or risk variants have already been identified in some of these chromatin modifiers, which are here, for instance, ARID1B, CHAD, or MBD1. So these are all chromatin modifiers which take part heavily in doing the chromatin regulation, and mutations in these lead to chromatin misregulation, and which is thought to be at the root cause of several of these neurodevelopmental disorders, which then go on to affect synaptic functions in the brain. So it's important to study this sort of chromatin regulation and how it happens during normal cerebral cortical development. So as I said, the stem cell, depending on the time at which it is in development, it is forming one particular sort of a neuron. So two things which are very, very important during development of the brain, and in particular the cerebral cortex, is that that of timing and that of location. So the stem cell here will form a differentiated progeny depending on what time in development it is, and depending on the location. So these are the two important factors which determine what sort of a fate the stem cell will take. And today in my talk, I'll be talking to you majorly about this regulation of this timing, or the temporal regulation of this process. And I'll probably give you a very brief hint. Is there a spatial angle to this, or location is also important in somewhat deciding what fate the stem cell takes? So today in my talk, I'll be telling you how the cortical development happens in mouse as a model system. And the mouse has been a very valuable model system because cerebral cortex is a higher order, it's a function of a higher order system, a vertebrate system. And mouse is one of the simplest organisms which one can study, where there are many of these features which are conserved between the mouse and the human. And thus we use mouse as a model system to study how the cerebral cortex develops. So shown here is a schematic, and this is embryonic brain, mouse brain, and this is at embryonic day 12.5. So when I say embryonic day 12.5, what I mean is when the sperm fuses with the egg, we call that as day 0. That's called embryonic day 0. So this brain is at embryonic day 12.5. This is so 12 days post-fertilization. And this is the time during which the neurogenesis has begun in the developing cerebral cortex. So this shown here is a section. So if this is the brain, as you see it, if you cut it face-wise, this is called as a coronal section. So what I'm showing you here is a schematic of the coronal section. And if you see that here in this brain, this region is the cerebral cortex. And you will see that there is sort of a, ventricle or a fluid system which lines this developing cerebral cortex. It sort of nourishes the developing cerebral cortex. It provides with all the nourishment which is required for the developing cortical primordium. So when you cut the section, you would see that it consists of two zones. At this stage, the brain mostly consists of stem cells. So this yellow region, which we call as the ventricular zone, consists of proliferating cells or the cortical stem cells. And at this stage, there are only very few neurons, as seen here in red. This soon will change within, say, 8 days in the mouse, because by E18.5, the entire brain is made in the mouse, and the pup is being delivered. So within 8 days, this structure has to go from just looking like a simple sheet of neuroepithelium or a simple sheet consisting of stem cells. It has to look like this. It has to look like a proper neocortex, which consists of 6 different layers of neurons. So each color here shows different layers of neurons. And each layer has a very important function. And I'll come to that in a bit, that, what does each layer of neurons do? Because each layer is devoted to doing something different. Each layer has a different different architecture, different molecular signature, and has a different function. So how does this then simple looking epithelium sheet then become this complex 6-layer neocortex is a question which is being asked quite often in developmental neurobiology, that it is the same stem cells, but some sort of a temporal regulation decides that what sort of neurons are being formed. So just to tell you that how these 6 different layers are being formed in the neocortex is that, as I was saying, that to begin with, it is all just the stem cells. This stem cell at, say, embryonic day 11.5, which is 11 days after fertilization, will divide and undergo asymmetric division. So before this, all the symmetric divisions have happened, and the sort of cortical stem cell pools have been generated. They will undergo a asymmetric division, whereby this stem cell will divide and give rise to one stem cell, which will sit again back in the zone, proliferating zone called the ventricular zone. It will give rise to a differentiated progeny, which is called as layer 6 neurons. And these neurons go and sit on top of this proliferative zone. So this happens at embryonic day 11.5. Look at in an adult brain, they look like this. And I'm showing you this here because in my talk later, I'll be showing you more of such images. So it is important to sort of know how a layer 6 looks like in a postnatal brain. So this stem cell divides, give rise to layer 6 neurons. This happens at embryonic day 11.5. And this here is marked by the marker TBR1. The very next day in development, these same stem cells, they switch. So they were producing layer 6 neurons at 11.5. At 12.5, they switch. And instead of making a layer 6 neuron, they start producing a layer 5 neuron. So the stem cell will divide, make one stem cell and one daughter neuron. The daughter neuron will now migrate past this layer 6 here and go and sit on top of this. And that forms the layer 5 neurons. And this here is marked by this marker called FESF2. As you can see, this is layer 6, this is layer 5. So layer 6 is here, layer 5 is on top. The layer 6 is produced from, the layer 5 is produced from beneath. It migrates past the layer 6 and goes and sits on top. And this keeps happening till all the 6 layers are generated. So this entire process happens during development, where all the 6 layers are generated. So it's here marked is layer 4, which happens at 13.5. And here is upper layers 2 and 3, which develop during embryonic day 14.5, 15.5. And so in this manner, the entire 6 layers of the neocortex is generated. So a very important question which we wanted to understand is what prevents or what sort of regulates this timing. So what tells the stem cell that you to now produce layer 6 or you should not now produce layer 5? So this is a sort of question which we wanted to ask and understand, that what regulates this temporal timing. So to understand this, we looked at the expression of a transcription factor, LHX2. So this is a homeodomain transcription factor. Basically, it binds to 80 rich regions in the genome. And we looked at the expression of this transcription factor in the cortex, and we looked at the expression of this transcription factor over a period of developmental time points. So we started, say, with embryonic day 12.5. This is the age, if you all will recollect from my previous schematic, that this is the age when your layer 5 neurons are being born, layer 5 and 6 neurons are being born in the cortex. So at this stage, this transcription factor is expressed in all the stem cells. So this entire zone is the stem cells in the embryonic cortex now. So this transcription factor, this is an in situ. So all this which you see in blue is the expression of LHX2 in the developing primordium. So this transcription factor is expressed in the entire cortex as seen here in the white asterisk. Now, if you look at the same expression 3 days after, say after embryonic day 12, look at embryonic day 15, This is now again the proliferative zone, or the zone where all your cortical stem cells exist. And this is now the differentiated zone. So as I said, the stem cells will produce differentiated progeny, which will go and sit on top of the stem cells. So this is now the differentiated progeny. In this case, this is layer 6 and layer 5. So when we looked at this, we saw that the expression of LHX2 is still only in the progenitor or the proliferator or the stem cell zone. It is being excluded .
I can't hear. Is it?
Yeah.
Madam?
Hello. Yes.
Now continue, ma'am. It was not visible. Dr. Rana, continue now. It was not audible. Now it is okay.
Oh, okay. Is it okay now?
Yes.
Okay. Yeah. Thanks. Yeah, please feel free to stop me and give me this feedback, because from time and again the internet connection does.
Okay. As and when we are going to remind you.
Yes, please. Thank you so much. I don't mind the interruption at all.
Thank you.
So, okay. So postnatally, so this expression of LHX2 then postnatally comes up in these upper layers, or layers 4, 2, and 3, but it's still excluded out from the deep layers, layers 6 and 5. So this led us to thinking that perhaps LHX2 may have some role in regulation of the formation of the, or some sort of a regulation it may be doing between formation of layer 6, 5, and that's why perhaps its expression is excluded out from these layers. Yeah. So, right. So then to understand that what may LHX2's role be, we took help, or we looked at the genetic mutant of LHX2. Now, in this case, the mutation or the loss of LHX2, I think there's some background noise with somebody not being muted or something. Yeah, it's fine now. Thank you so much. So we, so we took this genetic mutant of LHX2, and it is a conditional mutant, which means it LHX2, it's a loss of function of LHX2. LHX2 is removed from these mutants, and it is removed in such a way that it's only removed from the cortex. So it is not a mutant which will remove LHX2 from the entire body, but it only removed from the cortex. So it's a Cre-driven line where the Cre is very specifically removing it only from the stem cells of the cortex. And it is also timed. So it's both spatial and a timed removal of LHX2. The timing is such that it gets removed only at embryonic day 11.5. So function of LHX2 before that is not affected at all. So from 11.5, when the layer 6 and layer 5 neurogenesis is happening in the cortex, that's when the LHX2 is being removed. So, so this here I'm showing you is a control brain. And I've told you before as well that TBR1 marks layer 6 and FESF2 marks layer 5. And I'm showing you two other markers for layer 5, ER81 and ID2, as you can see here. In the absence of LHX2, we saw two stark things. The first stark thing we saw was that the expansion of layer 5, as you can see here, that between the control and the mutant, you can see that the layer 5 is expanded. That is, the layer 5 is more in the mutant of LHX2. As you can see here, visible better with the marker ER81 and also ID2. Simultaneously, we saw that not just was layer 5 expanded, the expansion of layer 5 happened at the expense of layer 6. As you can see here, the TBR1 is reduced. And this is just not a marker change, but also we quantitated these changes. And here shown is that marker for layer 5 is CTIP2. As you can see that there is definitely an increase in layer 5, and that increase is concomitant with the decrease in TBR1. So which means the moment you remove LHX2, there is a switch. The switch happens that the layer 5 gets expanded simultaneously, and this happens at the expense of the production of layer 6, which means that LHX2 is somewhat involved in regulating this switch to maintain the balance between the right numbers of layer 6 production versus layer 5 production. So this then led us to think that, this is a transcription factor So before going to that, as I was saying, that what does it mean when you have these different neural subtypes in the neocortical layers? Is it only that a marker expression, or is it just a molecular signature or architecture? What is it that does it affect the function of the brain in any way? The answer is yes. So what are the different kinds of neural subtypes in the neocortical layers? Is the layer 5, which is marked by TBR1, they make a set of neurons which are called corticothalamic neurons, and they are shown here. So this is a cortex, right? So the cortex is here and the spinal cord is here. So you want the cortex speaking to the spinal cord, and then the spinal cord gives out neurons in the entire part of the body. You also have neurons, you have the sense of touch, you have vision, you have hearing. So these are all the sensory modalities. So you want information from the outer world to come to you, and that comes to a region in the brain called the thalamus. So this is the thalamus here. So this thalamus integrates information from the outside world and then gives it to the higher center in the brain, which is the cortex, where the cortex then decides, to give you an example, suppose a ball is coming to you from a far end, the eye will see it, the eye will give the information to the thalamus will pass it on to the cortex. Now the cortex will then calibrate and see, based on previous information coming from memory or anything, decide and say, oh, this ball is coming to me at this fast speed, I need to move my hand or I need to duck to do something. So that the cortex will decide. So then the cortex will give information back to the thalamus, and the thalamus will then relate to your hand saying, put your hand or catch the ball or something. So that's how information processing happens in the brain. And so this thalamus is that relay center. Now, what does layer 6 neurons or the TBR1 neuron do is that they take the information from the cortex. So here is layer 6 in green here in this schematic. So they take the information from this layer 6 and they take it to the thalamus. So once a decision is made in the cortex, that comes through this corticothalamic neurons to the thalamus, and no other neurons in the brain do that. Now, what is layer 5 neurons? The layer 5 neurons are a complete different set of neurons. You want control of the movement, and that control is done by these subcerebral neurons, which are, so layer 5 consists of 2 different kinds of neurons, the FESF2, CTIP2 positive, or the ones which have those kind of markers. Those are subcerebral neurons, and one class of subcerebral neurons are the ones which come from the cortex and project to the spinal cord, seen here in red. So can you see this is layer 6, this is layer 5, the layer 5 projects to the spinal cord. So that affects all your movement is all done by that. The third the other kind of neurons, which are there in the layer 5, are the SADB2 or the callosals. So now you have 2 hemispheres, right? One hemisphere has to talk to the other hemisphere. So these are the sort of neurons which are called callosal. One class of these neurons are the callosal neurons, which help talking one part of the brain to the other. So that is the other neurons which are present in layer 5. So they essentially 2, one are CTIP2 FESF2 positive, the other SADB2 positive, and these are callosals which project from one hemisphere to the other. And these are the different neurons. So which means not just the molecular signature, but the function of the neuron itself is different in these kind of, when you have different neural subtypes, right? Now, what do we know? So when I was talking about, so when I said, when we removed LHX2, we saw a switch. We saw the switch that the balance between layer 6 and layer 5 was somehow gone. So you had more layer 5 and you had less layer 6. Now, we wanted to understand that what is happening molecularly to affect this kind of a switch. So some of it is already known, and what is known is there are about 2 transcription factors, TBR1 and SOX5, which are very important for the formation of layer 6 neurons. So when the layer 6 neurons are being born in the cortex, they repress the expression of FESF2. Now FESF2 is a transcription factor, which is very important for the formation of layer 5 neurons. So literally TBR1 and SOX5 repress FESF2 because you want to make sure that the brain is making layer 6 neurons and not layer 5 neurons. So you repress FESF2. But when the switch has to happen, that now it has to start producing layer 5 neurons, what literally happens is SOX4 and SOX11 literally knock off SOX5 from the FESF2 locus and activate FESF2. And once FESF2 is activated, it in turn goes back and represses TBR1. And that's how you decrease the production of layer 6 neurons and you start making layer 5 neurons. FESF2 will also activate CTIP2, which is another important factor for making layer 5 neurons. Now, separately, I don't want to go into the details, but separate set of neurons will start getting the expression of SADP2, and they will form callosal neurons. These will form the subcerebral neurons, and these will form the corticothalamic neurons. So since this was highly molecular and this was a transcription factor, one of the techniques which we thought will tell how this entire process happens is we thought we should perform ChIP-seq. So this ChIP-seq is a technique where you, the transcription factor binds to the genomic locus. You do a pull down. So not only you get the transcription factor, but you also get the associated genome with it. And where the genome is associated will tell you which genes it activates or represses. So from this, we found out that LHX2 was bound to two of the genes which are important from the formation of subcerebral projection neurons. These are FEZF2 and SOX11. And if you'll recollect from my previous slides that these are very important for making the layer 5 neurons. And bang on, LHX2 was bound. So this is a profile. So anything which is, so this is all just background here. So LHX2 is bound at these two positions on the FEZF2 and the SOX11 locus. And this is here a ChIP-qPCR validation. So we looked at the region where LHX2 is bound. There was a TATTA binding motif, and LHX2 is bound there, both at FEZF2, SOX11. And any other region which does not have this motif does not show a binding. So this says that this binding is specific on this particular locus. Now, so I told you what happens when you remove LHX2. You see that layer 5 neurons are expanded and layer 6 is gone. So we asked the question that if we remove LHX2 within a day, what happens to the expression of these two target genes, that is FEZF2 and SOX11? And as you can see here, this is the control brain and this is the mutant brain. The moment you remove LHX2 within a day, the expression of both these transcription factors, they go up in the brain. As you can see here, look at the expression of FEZF2 and see here, this is SOX11 in control, this is SOX11 in the mutant. This continues even at embryonic day 13.5, as you can see here in the mutant. FEZF2 is upregulated and SOX11 is much, much upregulated in the cortex. So clearly what happens is LHX2 is repressing FEZF2, SOX11. And the moment you remove LHX2, FEZF2, SOX11 expressions go up. Now the stem cell which was actually supposed to produce layer 6 neurons starts to produce layer 5 neurons because you have removed that important regulation or that repression. And because you started producing layer 5 neurons, postnatally, when you look at the brain, you see that the layer 5 is expanded at the expense of layer 6. So this is what is happening molecularly. So we were still not satisfied because we knew that this is a transcription factor. It has to interact with chromatin modifiers. So we did LHX2 interacting partners, and we found that LHX2 is interacting with chromatin modifiers called LSD1, HDAC2, and RBBP4. These are a part of a repressor complex, a nucleosome repressor complex. So as I was saying, these are chromatin modifiers. They keep the genes repressed by removing active marks or removing active marks from the locus. So they keep the locus of the genes repressed. And we found that indeed LHX2 was interacting with these proteins. This is IPs. So when you do LHX2 pull down, you get HDAC2, LSD1, RBBP4, and reverse IPs show that when you do IPs with HDAC2, LSD1, RBBP4, you pull down LHX2. So indeed, so LHX2 is now interacting with these chromatin modifiers. And perhaps next question we ask that we have two things here now. We have LHX2, we have FEZF2 and SOX11. We know that LHX2 regulates FEZF2 and SOX11. Now we wanted to know whether these chromatin modifiers also bind to the FEZF2 SOX11 locus. So what we did was we did ChIPs again. Now instead for LHX2, we did ChIPs for these LSD1, RBB4, HDAC2, both at the transcription start site of FEZF2 SOX11 and also at the site where LHX2 binds, because LHX2, from what we saw, was it was binding to putative enhancers, which are far removed from the transcription start site. And I'll come to that in a bit, that the chromatin actually loops for the enhancer to come in close proximity with the transcription start site. So we indeed saw that, LSD1 clearly was bound both at the FEZF2 locus and the SOX11 locus, wherever LHX2 was bound, and also at the transcription start site. RBBP4 was only bound at SOX11, whereas HDAC2 was bound at the LHX2 binding regions. Wherever LHX2 was bound, these modifiers were also bound. So clearly what's happening is perhaps that binding of LHX2 is facilitating the binding of these chromatin modifiers, which are then able to access the histones. And then the question was, are they now able to change the epigenetic marks? So that's what we next asked, that when we remove LHX2, are the marks changed? And we asked that what happens to the active marks, because in this case, the moment we removed LHX2, which was repressing, the expression of FEZ2 and SOX11 went up. So we indeed saw that active marks like H3K4 trimethyl mark, which are present on histones, they got increased in the absence of LHX2, as you can see here. And so did the H3K9 acetylation mark. So at the binding site and the transcription start site of FEZ2 and SOX11, both H3K4 trimethyl and H3K9 acetylation marks had gone up. Indeed, so sort of model which we have, I'll come to that, the model we have is that LHX2 recruits, perhaps recruits some of these modifiers to the enhancer and the transcription start site, which then sort of keeps the locus repressed at the time when layer 6 neurogenesis is happening in the developing cortex. So till now, I have been telling you that LHX2 is a necessary process, necessary. For means if you remove LHX2, you get an expansion or an increase in layer 5 neurons. Now, if you overexpress LHX2, what happens? Are you now able to repress layer 5 fate? And that's the question we asked. So to understand this, we performed a technique called in utero electroporation, whereby we go do a surgery on the pregnant mother embryonically, say at 12.5 when layer 5 neurogenesis is happening in the brain. We perturb it and then we look at postnatally what happens. And so in this case, we overexpress LHX2. And these are the sort of images, but I'll directly take you to the data here in the form of a bar graph. So at embryonic day 12.5, when we just do a control electroporation, close to 55% or 57% of the cells are making CTIP2, which is right, because it's layer 5 neurogenesis which is happening in the brain. But the moment you overexpress LHX2, this goes down to about 27%, less than 30%. So which means moment you overexpress LHX2, it is sufficient to repress the CTIP2 fate in the developing brain. Now, these neurons, which are now not CTIP2 positive, we ask the question, what is the fate of these neurons? And interestingly, the fate of these neurons had switched from being CTIP2 to that of SADB2 positive neurons. So all these LHX2 positive neurons, a large percentage, more than 60% of them were SADB2. So if you recall, that in the layer 5, there are two different kinds of neurons, right? One FEZ2 CTIP2 positive, the other SADB2. So which means just simply by overexpressing LHX2, we've switched the fate, a fate of a neuron which would have gone on to make a subcerebral projection neuron. We've now switched and for it to make into a callosal neuron. So that's the sort of switch we were able to do simply by the removal, by overexpressing LHX2. So this, I've nearly come to the end of my talk, and this is the model which we have for how LHX2 may do this role in a cortical stem cell, where LHX2 recruits, this is the putative enhancer, which has an LHX2 binding site. So the presence of LHX2 allows for these chromatin modifiers to come close to the enhancer and the transcription start site. And because these modifiers are remove active marks, both the locus of FEZ2 and SOX11 has less active marks, and that's why it's kept repressed. The moment you don't have LHX2, they're not able to now access. The active marks go up and FEZ2 SOX11 expression goes up. So now the stem cell starts producing layer 5 neurons at the time of layer 6 neurons. And that then leads to an expansion of layer 5 at the expense of layer 6. What we have now done here in this switch is we have placed LHX2 regulating or repressing two of these important transcription factors, FEZ2 and SOX11, at the time of the switch from layer 6 to layer 5 production. Now, this is absolutely my last slide here. So as I told you, till now, I've been telling you only about the timing part of it. Is there a spatial angle to it? I think there is, and I'll very briefly touch upon it on the spatial angle. Like, does the location matter of the stem cell matter, that what kind of neuron it forms? So from mouse to humans, as I was saying, there are many features which are, some features which are conserved between mouse and human. And one of the features which is conserved is when you have a brain like this. So always the front part is the part which is devoted for motor, followed by somatosensory, auditory and the visual, which means any input you get from the outside world, anything which has got to do with movement will all be processed here. Anything vision will all be processed here. Auditory or hearing, ears will all be processed in the auditory cortex, and somatosensory is all your touch. So this organization is sort of conserved between mouse and humans. So so you always have the motor cortex at the front. When we looked at the expression of LHX2, so the motor cortex is the area where you have a large expansion of your subcerebral projection neurons or the corticospinal motor neurons, which are layer 5 neurons, because those are devoted for movement or for doing all your movement activities. So when we looked at the expression of LHX2, we saw that there was a gradient of LHX2, and the gradient was in such a way that it was high caudally or at the back of the brain, as you can see here, that it's high here, and it was low rostrally or the front of the brain, which will go on to form the motor cortex. So perhaps what we think is LHX2 is part of a mechanism which regulates the number of neurons which are being produced. It's not the sole driver, but it's part of a bigger mechanism or a bigger transcription network or a bigger gene regulatory network, which decides what number of neurons are produced in which region of the brain. So it perhaps controls the number of neurons which are being, so by doing so, it perhaps enables that the motor cortex should develop at the very rostral end and not, at miss sites in the brain. So perhaps this LHX2 is part of such a process to regulate, you have what kind of neurons being formed where in the brain. So this is a sort of location angle or the spatial angle to this entire process. So I'll just conclude my talk saying that today in my talk, I told you about a very fundamental mechanism of how these stem cells in the developing brain function or how they make the different kind of neurons. And this sort of creates a framework, because once you have these iPSCs, there are many techniques which use some of these combination of transcription factors to drive these cortical stem cells into very specific lineages. As I said, to lineages which will make the motor neurons, which will make the corticospinal motor neurons, lineages which will make into the upper layers. So understanding of what drives this process is very important, so that tomorrow, if we want to make some of these neurons for therapy, understanding from such studies will be very, very useful in driving the iPSCs into very specific neuronal classes. So what I told you today is how this progenitor makes a neuron X in time, whereas switches and makes neuron Y in time Y, and that this process is controlled by chromatin regulation. And with this, I would like to thank you all for your patient listening. And this work which I presented today, I did in my postdoc in Professor Shubha Tole's lab, Sanjeev, and I would also like to thank Shubha, and Sanjeev has been a collaborator and a mentor in this project. And these are my lab members, current lab members, and I would also like to thank all the funding bodies which have sort of have funded the work which I presented today, and also been funding my lab, are funding my lab in the future. So thank you all for your patient listening, and I'm now happy to take questions.
So thank you, Dr. Bhavana Muralidharan, for your insightful talk on this chromatin regulation control on the neurogenesis, especially the 6-layer cortex of our brain. All right. So I think we have been knowing that embryogenesis, but neurogenesis has different type of mechanism that are regulated by the more of the molecular aspects of regulation. Thank you very much for this talk. And now I hand over to Professor Munjwala for a question answer session.
Thank you, sir. Thank you, Dr. Bhavana, for delivering lecture on a very good topic on neurogenesis. As such, I have a very basic question to you. With the understanding of this neurogenesis, is it possible to address the neurological disorders
Yes.
such as epilepsy or any other neurological disorders?
Yes, definitely. So as I was saying, that neurodevelopmental disorders, particularly autism and autism spectrum disorder or schizophrenia, they have a whole, I mean, time and again, a lot of these variants, risk variants or mutations are found in some of these chromatin remodelers. A classic example is the mutation in CHAD8, which leads to, autism. It is seen that mutation in CHAD8, which is a chromatin remodeler, involved in precisely doing this. It regulates neuronal fate specification and all of that in the developing brain. So there are two aspects to it, right? One is if we don't know what do these chromatin remodelers do in the normal brain development, it will be very difficult for us to then understand that what steps in development do these neurodevelopmental disorders affect, because we don't, studies started from understanding what CHAD8 does in normal development, in cortical development, which part of it. So as I was mentioning, there are several steps. Does it affect the proliferation of the progenitors or the cortical stem cells? Does it affect the fate specification? Does it affect neuron outgrowth? Or does it affect the final synapse formation? So it was important to understand all of that. So studies like this, so my own lab is looking into understanding what some of these remodelers, which I mentioned to you, the LSD1, RBBP4 and HDAC2, they work in normal development because there are several mutations in these remodelers which lead to intellectual disability. So I think both have to go hand in hand, that you have to understand what they do in development so that you can then understand that what mutations could affect cellular phenotypes which lead to a dysfunctional brain.
Yeah, in this case, when you talk about development, you are looking into embryonal to a certain stage. But in general, if you look into neurological disorders like dementia and others, it is more related to aging factors.
Yeah. So there are, so there are different kinds of neurological disorders. The ones which you're talking like dementia, ALS, they are aging disorders. Those are neurodegenerative disorders. So they are disorders which come with age, right? So there are neurodevelopmental disorders like intellectual disability or autism. When the child is born, you will see developmental delays. They are not able to learn. Their speech is affected. So those disorders, they happen during development. And having said that, I must also mention that what is also happening in neurodegenerative, particularly for Alzheimer's, is people are now started to think about hypothesis that even perhaps Alzheimer's is a disorder which is set in during neurodevelopment, though it is being manifested as an aging disorder. So you really don't know that, given that it's a neurological disorder, you don't know when the disease kicks in. The manifestation can happen, say, 3 years within birth. It can also happen 50 years after birth. So that's the sort of hypothesis which is being sort of considered in the field now, that, it's important to know when the disease manifested.
Yeah, that's true.
Dr. Bhavna, what about the role of the diet in the developing brain? We want to comment on that, the role of the diet.
The role of the diet. So in my talk today, I told you mostly about cell autonomous functions of these transcription factors, right? So for instance, what drives cell intrinsically these stem cells to become neurons and everything. But you're very right. There is a whole angle of environment to this. So these developing cortical stem cells, they integrate inputs from the environment also. And then that also decides the fate that sort of kicks in particularly during axon formation and synapse formation, sort of little later in development, later as in not just during specification, but when axon outgrowth and everything happens. So diet plays a very important role in that aspect. So today I didn't tell you about how environment or diet or what the mother's dietary patterns help in sort of, that's why it's important for the mother to have a whole lot of these supplements so that, at times when environment plays a very important role in deciding synapse formation, you want it to happen correctly, right? So it is both a combination of cell intrinsic and extrinsic factors, and their environment and diet plays a very important role.
Dr. Bhavna, there is one question from Dr. Kavita Tirumurugan. She has commented that you have given a very nice talk.
Thank you so much.
The question is on brain injury or lesion, how it affects the expression of LHX2.
Okay, so we have not looked at LHX2's role beyond embryonic stages. So that's, I may say that's a very good question, and that is something which is there in our mind, that what does LHX2, does LHX2 have any role to play in the adult brain, because it's expressed in the upper layers, layers 2, 3, and 4. So maybe it does have some role to play in maintaining the neuronal physiology. And then perhaps, since it's important to may have a role in neural physiology, then maybe it'll have some role in neuronal lesions or something. So that's something which we've completely not looked at. Something is there at the back of our mind, that is something which we should definitely see.
Okay. So thank you, Dr. Bhavna. As far as I see, there are no more questions. But I would like to
before I conclude, Dr. Bhavna, I want to know that as you have mentioned, since the fertilization, the neurogenesis gets started and develops, and it is common saying that before birth, even the mother's womb, whatever development is there after postnatal of 5 years, whatever the neural development or intelligent development, everything happen in the brain will continue for long. So how this early stage of brain development can continue, or whether we can acquire knowledge by our exercise in the brain, IQ and other things, how you can correlate with molecular aspects of this?
Yeah, that's a very relevant question, I would say. So postnatally, whatever the child experiences is very important. Particularly, there are studies which show that stress to a very young child affects its development, brain development, particularly in kids who already have a genetic disposition to, say, schizophrenia. Any stress or traumatic emotional stress, some of the paradigms could be that very abusive childhood or something like that. Then in those cases, they've seen that schizophrenia, which perhaps comes a little later, say during teens, there is a predisposition, or it presets. It comes up early in such teens who had a very traumatic childhood. So which means that these early life experiences are very, very important in determining how the brain synapse formation is affected. So in that context, so you're asking how molecularly it happens. It's been studied that some of these signaling factors like BDNF, which regulate, early genes which are involved in synaptic plasticity, or what happens, the crosstalk between synapses, those genes which are involved in all of that are involved in such kind of experiences. And similarly, it's known that exercises boost such strengthening of the synapses. So these are some of the ways by which, how you can, how early experiences or perhaps even exercises help to boost the brain.
So with this, I profusely thank you on behalf of the VIT, SBST, and specifically the organizers of this international conference, and hope we will be needing your association in our SBST in the future. Thank you very much for this kind talk.
Thank you so much, Dr. Sawan.
So thank you, the organizers, for giving me this opportunity to chair these 2 sessions. And thank you very much.
Thank
Professor
you.
Suraj, Manjuvarna.
Yeah. Thank you, Dr. Thora, for sharing and inaugurating with the sessions and then interacting with the speakers as well. Thank you so much for your continuous support.
Thank you, madam.
Now we move on to the next speaker. So we have next speaker, Dr. Krishnamurthy Kannan. He will be delivering a talk, and for this session, we have Dr. Asha Devi as the chairperson. Dr. Asha Devi is the head of the department in SBST, and I also request Dr. P.K. Suresh to moderate the next two sessions for the evening program. And now I hand over to Dr. Asha Devi to introduce the speaker. Let me just share the slide.
Thank you, ma'am. So good evening, everyone. It is my honor to introduce Professor Krishnamurthy Kannan, the speaker of this session. Professor Krishnamurthy Kannan is an academician and scientist of global repute with 40 years of experience in several areas of biotechnology, including stem cells. He is actively involved in academics, industry, research, and cultural activities. He has served in several leading organizations, including Guru Gobind Singh Indraprastha University, New Delhi. He worked on the ubiquitin molecule, a biomarker for hematopoietic stem cells. He also served as the Vice Chancellor in Nagaland University. He has worked in SPAN Diagnostics, Surat, and Institute of Medical Sciences, Surat. He co-founded Upstablished Food Technology Private Limited, New Delhi, a startup involved in plant protein-based products, and as director, Nivi Agrofoods, Bangalore. He has worked in the biochemistry laboratory, Imperial College, London. He has contributed several articles in leading newspapers and has been acknowledged as one of the, it's hiding some. Sorry for the interruption. There's a, okay, ma'am, it's fine. Sorry, he has contributed several articles in leading newspapers and has been acknowledged as one of the technology leader in India. He has written a textbook in biotechnology for school students. Based on information in the public domain, it has been reported that he narrowly missed the Nobel Prize in chemistry. Actually, this information was published in the leading newspapers such as the Hindu, the Hindustan Times, etc. Today, he will be talking on discovery of the role of ubiquitin and hematopoietic stem cells in neuron systems. So with this short introduction, I welcome you, sir. Over to you. You can start your presentation.
How do I get into this? Just 1 minute. How do I open my screen? Just 1 minute.
You just speak, it will come.
No, it is not coming. Can I request you to shut your this thing?
Sir, I have stopped sharing the screen, so you can, I think it's coming, sir. Yeah. You can see your Internet Explorer. You can open your PowerPoint, sir.
Yeah, that's what I'm trying to look at. Where is it? That's 1 minute. Suddenly.
View share.
Huh?
View share.
And just 1 minute.
Sure, sir.
Sure,
No
sir.
problem.
Sure.
Somebody else's screen is coming.
So you can close this Internet Explorer and maybe.
Okay.
Where is the Internet Explorer?
Run either reduce number open manda.
Yes. Sir. Yeah.
Okay.
Now your desktop is visible.
Yeah.
Yes. Yes, sir.
Then 1 minute. இங்க mute பண்ணு. இதை க்ளோஸ் பண்ணுறேன் அதை அங்கே அந்த how to open the sorry. Yeah, is that okay? Can you hear me?
Yes, sir. Yes, sir. We can hear you.
Okay.
Yes, sir, it's perfect.
Yes, sir. Yes. Sir.
Okay. Today I thought, we are going to go back into how stem cells were discovered and, how we do experiments. So what you heard today, or most of the speakers, were all current ways of doing it. But today I will take you through the journey of how original experiments were done, even to prove the existence of stem cells. You might ask, what has ubiquitin got to do with stem cells? It was more an accidental one because, I had a lot of nice mentors in my life, and one of the person was Sir Aaron Klug, who got the Nobel Prize in 1983. So we were together in Greece in a conference, in a workshop, and we were sitting and talking, and I casually asked him, sir, if you were young, what will you work on? Mind you, Sir Aaron Klug was the man who worked with Rosalind Franklin early years and a very famous crystallographer, So casually asked him, sir, I want to go back to India. What should I work on? Then he gave me a 4-hour lecture on why ubiquitin is interesting. So that's how I got introduced into this, because he used to work on histones and he gave the histone structure, he gave the tRNA structure and all that. So he said they were interested in a protein called A24, and they found that one of the histone is getting ubiquitinated, and that is how ubiquitin was first recognized. So A24. And in 1978, suddenly a report appeared. People have been wondering how proteins are degraded in the body, because people have been looking for various mechanisms, and 30, 40 years, everybody thought, enzymes are there to degrade. So what is the half-life of a protein? Because if you have a secreting insulin, it should act and then suddenly should disappear. So as I always say, there is what is called Brahma, Vishnu, Maheshwara, as we say in our mythology, that Brahma is the creator, Vishnu is the propagator, and Shiva is the destroyer. So everything has a life. So there is birth, there is proliferation, then there is death. So the question was, how is these proteins which are there in our body are turned? So that's how the whole interest happened. And in 1978, a very interesting paper appeared that protein degradation needs ATP. And I won't go into the discovery of the whole process. It's again a very fascinating, but for the first time they saw that the ubiquitin is linked to a protein via isopeptide. So I just want to emphasize this, because normally amino acids are linked via alpha amino, alpha carboxyl group. But in this case, ubiquitin is linked via the epsilon amino group of lysine with the carboxyl group of the ubiquitin from the C terminus, okay? And it's a very small peptide, so we can't even call it a protein in the strict sense of the word. And suddenly this group from Israel claimed that the moment ubiquitin is put onto a protein, it will be degraded. So that created a lot of excitement. And so I was more interested at that point of time, how does the ubiquitin recognize which protein to be degraded? And I spent a lot of time coming back to India when I was in CCMB Hyderabad. We spent a lot of time trying to understand this phenomenon. And to reproduce the assay was also quite difficult those days. We couldn't reproduce it for a long time. So when I got frustrated trying to look at the intracellular degradation mechanism, because then I suddenly realized it's a very difficult question to ask. It is like saying, there are so many boys around, how do you know who is going to get married? The moment you see a ring in the hand, you see that the person is married. Before that, it's very difficult to predict who is going to get married. So ubiquitin is something like a ring. If it just goes and attaches to a protein, it will get degraded, okay? And same thing, so this was an excitement. And my only question was, at that time, being very young and very ambitious, wanted to make a big discovery. So I asked a very simple question. What will be the effect of ubiquitin in chick embryos? Okay, so that's how we did. The data was too complex to even interpret those days, but something very interesting was happening. Now, to cut a long story, so I went to give a lecture at Cancer Research Institute in Bombay, 1986. And the title of my talk was, how does the protein know when to be degraded? So that was a very unusual title. So there are a lot of people around. And I said, look, this is what I'm trying to look at. Then I said, look, so there was somebody in the audience who was studying hematopoiesis. So he was the only person who was working on how blood cells are formed. So we were chatting. I said, can we look at what will be the effect of ubiquitin on hematopoiesis? That's how the whole story started. And I'll take you back to the story in short while. But I wanted to know that so everybody was looking at the intracellular role. So one of my mentors, Fred Sanger, always told me, if you want to succeed in life, look exactly the opposite way than what people look at. So everybody was looking at the intracellular role. So I remembered his piece of advice. And I said, okay, let me look at the extracellular role. So that's exactly how I got interested into studying the extracellular role and what is the role of extracellular ubiquitin on hematopoiesis, okay? So this gives you that there are certain level of ubiquitin in our body, plasma concentration is how much, etc., etc. So we won't worry about it. And in certain diseases, the levels goes up, especially in autoimmune disease, neurodegenerative. These are all things reported. But so I thought, let me define what it is. Okay. So a lot of ubiquitin storage, secretion, a whole lot of data is there. To cut a long story, let me just come to the too many reports have been there. So I'm just going to discuss how we demonstrated, there is a ubiquitin binding site on murine. Murine means mouse, okay? Hematopoietic progenitors. Now today I heard a lot of progenitor cells, and these are one of the early experiments of demonstrating a progenitor cells. And you heard today Professor George speaking, and he said, bone marrow transplantation, nothing but you get the cells and simply inject it. It will go and sit in the bone marrow and proliferate. So that's why we call it homing and adhesion, okay? So our work led us to understand the mechanism of bone marrow transplantation in mouse. And you also heard that to publish in this journal, and mind you, I was not a hematologist, I have no background in this area. I accidentally fell into it and managed to publish it in such a journal, okay? And I'll just take you through the journey of it, okay? So basically what we do, I'll just try to tell you. When we started the work, First thing my friend Dr. Anand Rao and CRS said, can you get me antibody against ubiquitin? So this was 86. And, I said, okay, I'll try. And I thought, just like everybody thinks you can make a vaccine, I also went about trying. My student Porch have tried. Then suddenly we realized we could never make the antibody. Then when we realized that, look, ubiquitin is present in the body, so there's a mechanism that the body has a mechanism to distinguish the self from the non-self, which is a very famous piece of work, which in immunology everybody teaches. So we realized that how to raise this antibody. So if I don't raise the antibody, and at that time there was no commercial antibodies, nothing available. So we said, okay, luckily I was working in a diagnostic company. We had goats and we had rabbits and we had mouse. It was a very interesting lab run by a pathologist who used to make various diagnostic products. So we used to make antibodies, etc. So I said, okay, let us start attempting. Then we realized that if you want to raise against any protein with antibody, you have to link it to some toxic protein. Okay, so to cut a long story short, we conjugated it and then produced the antibody. So after 2 years, I called up my friend. Do you remember? You said we can do the experiment the day I bring the antibody. I said, now the antibody is ready, so let's get going. So I sent my student to Bombay. And, those days we didn't have all this animal ethics committee and things like that. And this was a very standard assay. So to cut a long story short, we did an experiment, which was the one which first proved the presence of a stem cells, because people could never detect stem cells. They have looked under a microscope. Even today, you would have heard in all the lectures, nobody has seen a stem cell. They say, no, it should have CD34, this marker, that marker. You will hear hundreds of things. But basically, nobody had seen. And imagine when we were doing those experiments, it was a mess. Only way is what is called a functional assay or indirect assay. So the most famous experiment, which was developed by Till and Matlock, and as you heard Dr. George said, the interest in hematopoiesis essentially came out of the nuclear leakage, the Hiroshima, Nagasaki bombing and all that. So people wanted to know how much radiation can the body withstand? Can we, repair because these stem cells, if they are destroyed during radiation, can we do it? So that's how the whole work happened. And Till and Matlock were the first who demonstrated this experiment. Now, I must tell you a little bit about these experiments, because one other thing is that it is like you are all listening to this talk. So many people have spoken. And if you were to conduct an examination on what you have understood, we are going to get, somebody will get 0, somebody will get 5, somebody will get 10. And we do not know whether our lecture was good or bad or ugly or whatever it was. So it is very difficult to interpret when you don't have. So a guy called Snell in the early years, he discovered that if you made the brother and sister of mouse, you get a population which are genetically identical. So all these white mouse you see, they will belong to bulb C mice. So there are a whole lot of varieties of mice. So we took one set of mice which are genetically identical, so that the results can be interpreted. So this is a classic experiment we did. We had a population of mice, controls were there, experiment were there. And the beauty of a mouse system is that, whenever you inject cells, especially, I'll tell you how we do it, we find that if you take the bone marrow, and today George was telling that you take the iliac crest and make a hole in that and collect the cell. So in case of mouse, what we do is, because there are so many mice available, we sacrifice the mice, take the femur bone, flush it out. And people had established that the population of cells on centrifugation at 1.077 density gradient are likely to have stem cells. So this has been well established that 1.077 density gradient cells are rich in stem cells. Then the next question comes, how many cells to inject? So people have attempted with 10 to the power 1 cells, 2 or 10 to the power 2, there are 100 cells, 1000 cells, etc. Then they discovered that 10 to the power 5 cells is the best one. 10 to the power 5 or more will give me more. So the beauty of the mouse system is that, unlike our system, the spleen also is a hematopoietic organ. So when you irradiate a mice and then injects this mouse cells, 1.077 density gradient cell, and if you inject it, and after 12 days you kill the mice and look at the spleen, you will see colonies on the spleen. That is why it is called colony forming units on the spleen. Just like you see bacterial colonies during a bacterial culture, you will see this. Now, if you take any of this colony, irradiate another mouse and inject these colonies, again the mice lives. If you don't inject, the mice will die because there's no production of this thing. So this is how Still and Maglock for the first time showed that in the bone marrow, there are cells which can form colony forming units on the spleen, and this can be this. Okay. So people have started to see, okay, it's very interesting that from the spleen we are able to understand. So the next question happened, what is the basis of these cells? Because we can't see them, but we can see the colony forming unit. So if there are 25 colonies formed, means the calculation for spleen was there are 25 stem cells. Each colony is called one stem cell. So this is a standard assay, and to cut the long story short, before that people have tried with sugars, trying to compete with sugars, and maximum inhibition of colony forming units they got was, in the world was only, even with the RGD peptide, only 40%. But when we did the experiment, for us, for the first time, we got 78% inhibition. That means we knew that how did I prove it? So what you do is you grow the bone marrow in the lab, and then you take this femur bone, that 1.077 density gradient cells, and then you inject, say, so many cells, 10 to the power 5 cells into any irradiated mice, calculate how many colonies are formed, and as you can see, you have 28 plus minus 1.40 colonies formed. Then what we do is, next one is that we treat the the marrow with antibody against ubiquitin, wash off the excess antibody, and then take this 1.077 density gradient cells and then see how many are not binding. The other way around is you simply put your 1.077 density gradient cells onto the stroma, so the stem cells will bind. So you calculate how much is, and if you put antibody, how much is in the supernatant. So every time we take the supernatant, inject it, and see how many colonies are formed. So as you can see, there are 28 colony forming cells. If you put it into the stroma, which has not been treated with antibody, and then look at the supernatant, we get so much. So 28, it has become 18. But if you put antibody against this one, we get 25 colonies formed. So we calculated that we could inhibit the binding of hematopoietic so-called progenitor cells, because at that time, nobody talked of stem cells. Let me be very clear. So we could do the inhibition. Okay. So the next question is, we also did, another set of assays. So as you can see, this assay we again repeated. So people said you must see the effect of treatment of light density non-adherent cells with ubiquitin on homing of CFUS to spleen. And finally, and now this is interesting in the sense that if ubiquitin is involved in binding the hematopoietic progenitor stem cells, suppose I treat this 1.077 density gradient cell directly with ubiquitin, wash it off, the unbound one, and then inject this ubiquitin treated cells into the mouse, and see how many colony forming units are formed. And we found we could inhibit the binding directly by treatment, but to the extent of about 58%. So we showed that that ubiquitin is some way involved in the homing and adhesion, because the earlier experiment showed that it goes and sits on it. I'm able to inhibit it to the antibody, so it doesn't home. Okay, and then and I could also prevent the homing by treating with ubiquitin and show that both homing and adhesion both are affected by ubiquitin, which is a totally new finding for the first time. Okay, so the next question we asked is, oh, is really ubiquitin there in the stroma? Okay, that is the bone marrow which we have grown. So we isolated that with an antibody with this thing, then we ran the gels, and finally we showed that there is a 54 kilodalton protein which is ubiquitinated in the stroma to which it binds. Okay. And to cut a long story short, so we knew. Now the next question which comes to us is that, if this is true, what we are saying, then I should be able to stain the cell with ubiquitin. So we attached it to biotin, streptavidin, alkaline phosphatase, and as you can see in the middle there is a cell which is red in color. That's about because alkaline phosphatase gives this color. Now this morning Deepa Bharti was talking about VSEL cells. In those days, we also found that these cells to which ubiquitin is binding is 95% nuclear material and 5% cytoplasm. Okay, so this was, earlier somebody had predicted looking under the microscope that these are called transitional cells. Okay, so the early word was transitional. Today the terminology has gone. So we were the first to show that we could pick up this cell. So it is called progenitor cell, which is practically loaded with nuclear material, but with very little cytoplasm. Okay. And we found that if you take photographs slowly, it is going inside. Okay. And and we in fact, we found that, we had a group which was working on monoclonal antibodies. I'm talking about 1988, 89. We were the first companies to start making monoclonal antibodies. And we found that if you, when they were killed, and then we took the marrow and studied, we could see dividing cells, asymmetric division happening, which also we have seen. I haven't shown it here, but just to show you, because I didn't expect Dr. Deepa to discuss in great detail, but these similar things were seen with ubiquitin. Okay. Now, when we did this staining, mind you, everybody is talking about antibodies, monoclonal antibody, polyclonal antibody, and here we just take a protein and we find it is able to specifically bind, and it had all the beautiful that it was very specific. It identifies a discrete class of cells. It was very selective. Okay. And it is because it is rich in 95% nuclear metal and 5% cytoplasm. So there was no confusion in the whole thing. So it was specific, selective, and objective. So because I was in a diagnostic company, we talk of false positive, false negative, which you hear for COVID these days, but those days we are quite as. So I proposed in the paper when we wrote, probe may possibly bind to regions such as binding cleft of receptors, which, because we only talked it as binding pocket. To show as a receptor is a much more complex those days, okay, which are normally inaccessible to conventional antibody probes, and in conjunction with such conventional probes may prove useful in identifying active versus quiescent receptors. So this was something which we predicted in our paper, okay. So as you can see, we were looking at the niche, the hematopoietic niche, which I talked to you, because I told you in the morning that stem cells per se, if they get a signal, they will differentiate. Otherwise, they'll just sit there quietly. So hematopoietic niche within the stromal microenvironment today, a lot of, we are talking about 25, 30 years back, so we saw a 55 kilodalton ubiquitinated protein, okay? Now, because it was a post-translational modification, so if the molecular weight is say about 8,000, so 55 minus 8,000 is the protein. So the question was, is ubiquitin binding or it is antibody which is binding to that region and probably covering some other protein, and ubiquitin blocks that particular site, the antibody from binding the stem cell. That's why we did that experiment of treating the 1.077 density gradient cells directly with ubiquitin to prove that ubiquitin is involved and not some other protein. So that was another new finding which we showed, okay? And we found that this progenitor cells, the population is about 0.347, 0.35%, okay? So that is quite a large number. This morning, VLSAL, what madam was saying, also the number is large, progenitor cells. So, I mean, when we did these kind of experiments, many of the words what people today use was already, many of us used to talk about it, but then there are not too many workers in the field to even talk about it, okay? So, but what I want you to understand is that we were able to explain, to cut a long story short, that the ubiquitin mediated mechanism for homing, thus appears to be more generalized and therefore possibly more primitive mechanism, because ubiquitin is conserved from plant to animals. Among animals, there is no change in sequences. The difference between a plant and animal ubiquitin will be 1 or 2 amino acid at the most, okay? And so as you can see, all our observation clearly showed that ubiquitin has a major role in this thing, okay? Now, this is a beautiful piece of work the earlier speaker, Dr. Bhavana, did as a MTech student for her MTech thesis. This was presented at the Second International Stem Cell Conference in San Francisco, 2005, and where we had some very interesting data which we presented. And I just want to show you that she tried some very beautiful experiments, and she could differentiate both HL60 cell line and K562 cell line to become megakaryocytes, and we could mature them. So in leukemia and all that, the cells don't mature. So moment we put ubiquitin, and it's a beautiful series of experiments which Dr. Bhavana did those days when he was a student, and we could see megakaryotes, and these are all beautiful pictures she has taken, okay? And so varying concentrations we have tried, and these are all the various pictures. So this is a very interesting fact of it. Now, ubiquitin also is involved in epigenetics, okay? So there is also a lot of points which comes, but I just wanted to show you this, that at that time when we were talking, this was the first experimental evidence for an extracellular role for ubiquitin. When the whole world was looking at intracellular role, involvement of E1 enzyme, E2 enzyme, E3 enzyme, ATP, etc. The whole world talking, we were sort of trying to focus on this, and we had a lot of interesting data, okay? Now, so I just want to cut this thing. Today, like in all literature happen, we have been acknowledged in textbooks and all that, Williams hematology, specifically the CD34 positive binding and all is happening due to ubiquitin. So it's very well recorded in the Williams hematology textbook, which is the Bible for the hematologists. But today people have shown that we didn't talk of receptor, we talk of a binding pocket, but now they think it is a CXCR4 receptor and CD184 agonist. So extracellular ubiquitin, and it has been shown that this extracellular ubiquitin promotes intracellular calcium fluxes, reduces cyclic AMP. So the whole field has opened up to such an extent today that everywhere ubiquitin is getting implicated, And just like we talk of phosphorylation, we are also going to see if techniques are available to see the ubiquitinated proteins or peptide, a whole new world will open up. Of course, when we were working on stem cells, there was a total ban on working on it. So we literally had to, just look at some of these things. But today, the world has changed a lot. But I want you to know that, people talk about extracellular, ubiquitin during inflammation. People have proposed mechanisms. Matthias says this thing. Of course, in the review he published, he acknowledged that we were the first to see such an observation. A lot of people have taken this further. But today, when we are talking about COVID and inflammation and things like that, ubiquitin probably has a role which we need to be investigating. But now I just want to just spend a bit of time in this, that this ubiquitin has about 7 lysine residues. So this where the ubiquitin, binds, which lysine binds to which region, is very, very important. For example, say there's a polyubiquitin chain, things like that. So they have shown that, if particular K48 lysine is involved in ubiquitin, it becomes target for proteasome or degradation. If lysine 63 or things like that are involved, some other phenomena. So today, for each of those regions, people have started giving an interpretation what can happen. So I think today, when I look at the ubiquitin field, it has become another field totally. I mean, the E3 enzymes people have reported is so much that the whole degradation seems to be much more complex than we have ever understood. But I just wanted you to take that these kind of experiments are the ones which first established the presence of stem cells. Okay, and all these monoclonal antibodies, all these, and they're okay, they are structural assays, they are not functional assays. Until you do a functional assay and show that that particular cell is doing it, and you will see all of them are injecting 10 to the power 4, 10 to the power 5 cells. Even you heard Dr. George saying today, we inject 1 million cells per kilogram body weight, that is 10 to the power 6 cells. So we do not know among that, it's only a probability guess how many stem cells are there. So stem cells per se are very few in number. Just to give you an idea, for example, those days in mouse and all, we used to say probably there are hardly about 1,000 stem cells per hematopoiesis, okay, which is the mother stem cell, which produce this thing. Our body, for example, probably may have hardly about 40,000-50,000 hematopoietic stem cells, which every day produces 1 billion RBC, 1 billion platelets, 1 million T cells, 1 million B cells, neutrophils, basophils. It is churning out every day. And and everybody is looking at, everybody talks about quiescence and all that. It's not that easy to demonstrate, but ultimately the rate of production is what matters the most. And lastly, I must say, for biotechnology people, studying the hematopoiesis led to a lot of important blockbuster molecules. Most famous is erythropoietin, which was the first success in biotech, which made $40 billion for the first company which started that. So I'm just saying that the biotech boom has a lot to do with trying to understand how stem cells differentiate, proliferate, self-renew and today you just saw Dr. Bhavana's presentation on the cortex and what kind of diseases can it cause. So stem cell has opened up a whole new field and a lot of exciting things, but we need to be very careful when it comes to human application. And I hope people who work on it are conscious of it and not be in a hurry to promote, because in the hurry to promote, if something goes wrong, there'll be a total ban. That's what happened to gene therapy. A lot of dramatic experiments are done, the patients died, and there is a total ban. So I think we have to walk carefully. A lot of claims can be made, but to prove the thing is tough. And I hope we have mechanism to do it. Thank you.
Thank you, Professor. Thank you for your insightful talk on ubiquitin. Now I request Professor Suresh to take care of question and answer session.
Thank you, Dr. Asha Devi. Thank you, Professor Krishnamurthy Kannan, for delivering a second lecture after the inaugural. It's quite informative, and the work that you did at that time was really groundbreaking. Due to paucity of time, I think we have time for one question. The question is being raised by a participant by the name of Munaza Rashid. The question is, what are the obstacles that must be overcome before the potential uses of stem cells in cell therapy will be realized?
It's a very, very important point. First thing, you should know what is the stem cell you are using. How do you do the quality assurance of it? Number two, that HLA typing becomes very important for many of these cells because there is graft versus host disease. Only bone marrow transplantation, that also happened because of Donald Thomas doing a lot of hard work on dog and establishing it. And from 1967 only bone marrow transplantation became a reality. So I think it requires it is like trying to send a rocket into the space. You need a whole team of people with various specialties. And in India, unfortunately, our MBBS community, we don't, they are not that, they are more busy with clinical thing. And we have scientists who are interested in basic, the marriage of the two to have a common ground of working together has been missing. So in India, at least I can say, abroad, the same person has done his MD also, he has done his PhD. So they become the principal because they appreciate both the sides of it, take it together. But in India, we are yet to see, and the togetherness of the scientific community and the medical community, lot needs to be desired for success of any of these kind of programs. Otherwise, we'll only see foreigners bringing their products and we'll be simply using it. But I think there's a lot of scope because what I see is that our population variation is different. So the methods which works there may or may not work here. And everybody is talking about personalized medicine today. So I think we need to be very careful. And our government, our regulatory agents should have proper procedures. And I can tell you ICMR now only has come out with the new guidelines as of 2020 to do it. And number two, it is different from a drug, even though it is called as a drug, because every time you prepare the cell, it is a unique cell. So there's nothing called mass production of the cell. That's simply not possible. So every time you are creating a new cell for treatment. So it is a quite different from pharmaceutical products. I hope I could clarify what is the bottleneck.
Thank you. Thank you. Dr. Manjugola, madam. Please unmute, Dr.
Yes.
So much, sir.
Yes, sir. Yes, sir. Thank you, sir. Thank you for very good insight into basics. I have a very simple question. Maybe it can be a silly question from your point of view. You mentioned that we have to be cautious with all our experiments of cell culture. My question is that all these stem cells are influenced by the external environment for their differentiation process. Now, taking into account the surface roughness of the Petri dish in which we do the cultures, will that not influence the differentiation process of the stem cells?
It will definitely. I mean, in the sense that people have tried with glass, they have tried with plastic, they have coated things. Whole lot of developments are going on. But the point is, normally in stem cells, when we do not try to grow them in the lab like one does. Like, for example, as Dr. Biju George was saying, you inject GCSF into the patient, get the peripheral blood, collect the apheresis you concentrate it, CD34, and directly inject it into the patient. So there's nowhere your Petri plate or anything is coming that way. Okay.
No, then the results are only one lab oriented, na? So it cannot be like extrapolated to another lab, which what they are doing.
I agree. That's why I told you that the problem with this whole field is that people have different body constitution.
Yeah.
And for example, say if you want to cord blood, everybody was making noise about cord bloods. Now, what is the probability that that will match for a particular recipient? That's why everybody says it is a waste of time trying to have that. Unlike blood banking, which also happened only in 50s, when people realized that we have, otherwise, there was no blood bank before 50s.
Yeah.
Okay, so it takes time, and people are now talking about haplobanking. That's a new concept which is coming. We hopefully that will do for your thalassemia and sickle cell anemia, make them free of the disease. So a lot of work is going on that. Japanese have done a lot of work on this direction, but we need new tools, new approaches, and handling cell is a different ball game.
Yeah.
It is not like other things. That's what I would like to say.
Doctor, one last question, very interesting one from the same person, Munasa Rashid. Where do stem cells come from?
Okay. To begin, originally, when the egg gets fertilized, okay, it keeps on dividing, so they don't know what they're going to become. And if you remember, gastrulation is the phase when the fate of the cell is decided. So gastrulation is the most important in any developmental process. So initially, as today even Dr. Bhavana was saying, initially it multiplies in large number of stem cells. Then as there are enough cells, then it stops dividing at that rate. So basically they are all produced from after fertilization. Okay, that's why ES cells are pluripotent, we say. So when it is totipotent, when it is pluripotent, when it is multipotent, there are different stages. So it's a much more involved. It can't be answered in 1 minute, but these are all generated, and like a stock market, you keep the stock safe and only play with the game, but make sure you don't lose your money. That's what happens in the body.
Thank you, Professor Krishnamurthy Kannan. Now over to Dr. Asha Devi.
On behalf of the convener and organizing secretary, I thank you once again, sir, for your talk on discovery of the role of ubiquitin and hematopoietic stem cells in immune system. Thank you, sir.
Thank you.
Thank you, sir, for your wonderful talk. We will proceed on to the next speaker of today's session, Dr. Sajan George. Dr. Sajan George is a faculty in VIT. I request Dr. Asha Devi ma'am to chair this session, as well as Dr. P.K. Suresh sir to moderate the session. Over to you, Dr. Asha Devi ma'am, to introduce the speaker.
Yeah, thank you, ma'am. So it's my pleasure to introduce Dr. Sajan George, the next speaker of the session. Dr. Sajan George had his PhD in biomedical technology from University of Johannesburg and master in microbiology from University of South Dakota, USA. He is a veterinarian surgeon as well and functions as a veterinarian and poultry specialist. He has master's degree in veterinary clinical preventive medicine. Dr. Sajan George is an enthusiastic researcher in the areas of cancer biology, infectious disease, and stem cell differentiation. His research using low-intensity laser has differentiated human adipose-derived stem cells to neurons for replacement therapies. Previously, he has identified the biological role of feedlot antibiotics as growth promoters in weanling piglets. He has also performed preclinical trials of antifungal drugs in companion animals and transcriptomic analysis of animal pathogens. Today, he is going to deliver a lecture on engineering 3D model systems for tissue engineering. With this short introduction, I welcome you, sir. You can proceed with your presentation.
Thank you, madam. Thank you for the warm welcome. It's getting late evening. I'm sure that people, our audience, are getting restless. So I will make a presentation as quick as possible and as short as possible. I'm going to share my screen, and I hope you can see my slides now. Is that visible? Am I audible?
Yes, yes, Dr. Sarjan. Please go ahead.
Yes, perfect. So the slides are working. So once again, thanks for the organizers for putting me into this slot. It's late evening, and some of these presentations I have tried to put a lot of animations to make it enjoyable. I understand that a lot of audience are students, so I believe this will be helpful for them. So the title, again, it is Engineering 3D Model Systems for Tissue Engineering. So a lot of things you might have heard already throughout the day, talking about the stem cells and how the role of stem cells in differentiation, and then also how you are going to learn a lot about tissue engineering in the next session, in the next tomorrow. So right now, I'm just giving kind of a brief introduction about the good points. So the stem cells are some good points, which is put on one side of the slide here, and on the other side there is like some bad effects, which is like manifested as cancer stem cells. And there is a sword I have put in between. Sorry to put that, it's a weapon, but there is a reason for putting that, and the reason for putting the double-edged sword in between, you will come to know by the end of the talk. So there's one side is the good side of the stem cells, and the other side, it's a bad side of the stem cell once it becomes the cancer stem cells. And I'm going to talk only about the good side today, and that is what this talk is all about, because it's a stem cell conference, and basically we are dealing with stem cell differentiation as well as replacement therapies and tissue engineering. So this is a topic which is relevant for now, and I'll get you into this in the next 30 minutes time. So the first slides include these organoids, spheroids, and comparison, and some of the aspects of light-induced stem cell differentiation, and some of the aspects of the sonosensitization, which is the sound. The sound, how does the sound induce differentiation of the stem cell? So this is a very broad classification. If you kind of look at how we can engineer 3D model systems, how we can devise the 3D models, starting from the pluripotent stem cells or the mesenchymal stem cells or the VLES, as we heard today. You can divide. I mean, you can kind of work on the stem cells, whichever you have, and you can put them into either way, like the differentiation of the cell and subcellular level, or the tissue and or the organ level. So the differentiation of the stem cells, basically that's what we talk about in the cell and subcellular level. So in this, actually, we most of the time we deal with the cells which are like in the 2D systems, and I'm not going to deal with each of these engineering technologies because that's a very broad topic. But I'll just give a kind of a brief overview of some of these during the talk. And then this actually will lead to generation of the stem cell organoids, which are like the 3D models. And for people who want to know more about each of this technique, I would suggest go through these reviews, which I found, especially the review which is like 2019, the design principles of the pluripotent stem cell derived organoid engineering, which kind of basically narrates a lot in detail and specific about each of these procedures. So let's move on to the slides and then get it done as quickly as possible. And as I told, there is a lot of animations, but it will be enjoyable to you. So please stay tuned. So under the skin, you know the mesoderm and the mesodermal stem cells. You will find these adult stem cells, of course, and adult stem cells is from where which you can kind of differentiate into spheroids, and the spheroids are 3D structures. They remain in suspension, and they can be generated in the low attachment flask, the flask which does not allow the stem cells to attach on the surface. So if it does not allow, then basically they can form into like 3D structures like the spheroids. The spheroids is like spherical, rounded, or it has got a central core which has got cells which sometimes get in the state of hypoxia, and the cells which are in the periphery, which get a lot of aeration, which is in the aerobic medium, and then the cells in between the center, the necrotic core or the central core and the outer periphery. And those cells which are like in between, those cells will be the differentiating stem cells. And the cells on the periphery, those will be proliferating cells. So if you look at the spheroids, the 3D models or 3D structures or the stem cells coming from beneath your skin or beneath any organ, which has got other structures like the fat, the capillaries, and the stroma, which contains this fibroblasts and all that stuff. And if you get the stem cells by the procedure, by mesenchymal stem cells, what they are called actually, you can differentiate them easily into spheroids. But that is not the case once you kind of try to differentiate them into organoids, because organoids is more polarized, and the cells are like very organized. They get assembled. They form like different layers, and each of these layers has got specific functions, and cells undergo some migration so that it develop into kind of miniature organs. And what science is looking at the moment is to develop these 3D models, the cells, the mesenchymal stem cells, into the organoids, or the mesenchymal stem cells, or even the pluripotent stem cells, that the stem cells that we obtain from the umbilical cord or the cord blood. In fact, those are the sources for generating these organoids. And a lot of work and investment is done in this direction, because this is kind of closer to the organ, actually, our own organs. So it's miniature organs, but truly not the organs. There are a lot of caveats. It's early stage. So early stage organs is sometimes good for disease modeling, drug screening, and also it's a better option than doing spheroids. And above all, there is something called this organ on a chip. And this is quite interesting. A lot of interest is being taken in this line also, where we can grow cells in specific channels on a chip. And then each of these chips can represent an organ of our body, especially this chip, which we can kind of represent as a lung. And the lung, there is like cells, and the cells which are like, there is an airway at one chamber, and there is blood vessel on another chamber, and the cells which are like laid in between. So there is kind of an interaction between this airway, the outer air, the air which is coming, that is basically what is happening in the lungs, organs like in lungs. Similarly, this kind of chips can be developed for other organs like liver and kidneys, and where we can assess a lot of disease conditions, and we can try different drugs. And in these days, people talk about human on a chip. So different organs working as a cluster in different parts of this chip. So there's like two substitutes here at the moment, and I'm not the expertise in that area. So I'm not going to deal with this one, but I'll be dealing with the spheroids and also some techniques which could lead to development of organoids. So moving on, again, this generation of organoids in vitro, as I told, the stem cells, of course, obtaining the mesenchymal stem cells or pluripotent stem cells, and then getting into spheroid structures or rounded structures in the low attachment flask, and then they try to self-assemble. And once they try to self-assemble, and if they want to develop into an organoid, they have to undergo some patterning. So that patterning is quite interesting, and because that is always dependent upon the external cues, external signals, and that's something very, very interesting. So how we manage this external signaling, external or environmental cues, and how the stem cells can undergo self-patterning is a very, very interesting topic, and it's a lot of work is going in that direction. And again, once that is done, it can get into a stage of self-morphogenesis. So in self-morphogenesis, actually, the cells get organized, they migrate, they take individual functions or unique functions, and again, they try to develop into specific or miniature organs. So this is actually this process happening in the organoids, the development of organoids. So again, if you try to, the one in the top panel, it is actually talking about theory. So now once we try to put this into laboratory practice, something which is in a practical sense, always try to translate the theory into practice. So then again, as I told in the previous slide, we need some low attachment flask where we can kind of we can grow the stem cells, but the stem cells does not attach; they remain in suspension, and over a period of time, they actually get clusters, and those clusters actually they start beginning an early stage of those clusters. Once we obtain those clusters, once we spin that down, we can get the cell aggregates or the stem cell aggregates. These stem cell aggregates actually can be embedded with the extracellular matrix, and that extracellular matrix, this is like the clusters embedding into extracellular matrix, and then once the extracellular matrix is submerged, this technique is called a submersive technique. submersive technique of growing the extracellular, growing the organoids in the extracellular matrix. And then over a period of time of 10 days, you will find these clusters started getting undergoing self-morphogenesis, and they become like they assemble, they start patterning, and then cells start migrating, they attain polarity and different other things. They develop into specific organoids. So this is one technique which is called as the submersive or the submerged technique, where the stem cells, the stem cell clusters or stem cell aggregates, which is grown as attachment flask, where you can embed into this extracellular matrix and then grow in the growth factors. And then over a period of time of 10 days or within two weeks time, they can self-organize and assemble and undergo morphological changes to become organoids. And our task, something which we planned, which is very unique, so far nobody has done. So we actually have got some plans which we have not executed in the laboratory yet due to some reasons. And so in this case, we want to generate organoids of tendons. So tendons and ligaments are something which you might have heard already. Ligaments are something which is connecting between the bones, bone to bone. So you can see one bone is connected to another bone. But in the case of tendon, it's something like a ligament is, it's like a fibrous structure which connects between the bone and the muscle. So this tendon is slightly more sophisticated compared to the ligaments, because the ligament is between bone and bone, so it's simple structure, whereas this tendon is slightly more sophisticated. It has got tendon-specific progenitor cells, the tenocytes, which always lay around, and then the fibroblast, which has got a lot of collagen, and the collagen fiber, and some outer coverings. So once you try to develop organoids of tendons, the entire reason for doing this is to address the replacement or to provide replacement therapy for athletes or for race horses, which undergo some tendon injury or tendon crushing because of the violent movement. Or because of some old age, some replacement therapy. So if you want to generate organoids for this tendon, again, we try to do in a different fashion, which is called this hanging drop, or similar to hanging drop, which is the transfer plate. It is not something very new, but it's been already practiced for more than 1 or 2 decades, where we can grow the stem cells, mesenchymal stem cells, especially on these transfer plates, and then embed them in the extracellular matrix, and then try to see if they are differentiating. In this case, these stem cells are grown in the transfer plates, and it is submerged, but at the same time it is suspended. So it's like a suspension method where the stem cells get more more external cues from the environment, and it allows them to differentiate into organoids or much more sophisticated than compared to the spheroids. So the structures which are like very similar to what we can kind of use and foresee to develop as organoids for the tendon. So that is something which we planned but not done so far because of some technical reasons. And so when I looked at this literature, so right now I don't have the data for that, but I'm just looking at some literature and then see how does this work actually. So then we will see, as I told, the external cues, the environmental cues, the growth factors, the growth medium, everything plays a greater role here. And most of the time, this tendon differentiation from the stem cells is achieved through the TGF-beta and BMP signaling pathway. So how does the TGF-beta, BMP signaling or GDF signaling pathway works? That's the sort of literature which I found so far. Most of them, almost 90% of the papers in differentiating these stem cells into tendon-specific progenitor cells or the tendon or the tenocytes. They talk about the TGF-beta and BMP signaling. So the signaling works in a very simple fashion where the ligand comes and attaches and then the signal gets transduced all the way. And then that actually, because the SMARTs lose inhibition as soon as this ligand comes on the receptor, the inhibition is lost, and basically the SMARTs get phosphorylated by the phosphorylated SMART, and the SMART and the phosphorylated SMART both combines and enter into the nucleus, and then the gene transcription starts. So that signaling, once that's a part where the growth factors are environmental cues. can do on the differentiation of the stem cells for tendons or tendon organoids. So again, that's not the only case. That's not the only candidate. We need to look at the extracellular matrix, because extracellular matrix is quite important, especially once we want to generate organoids and not the spheroids or any other differentiated cells. So the extracellular matrix most of the time is hydrogel. It's based on tendon tissue engineering. And a lot of times, I'm not going to read all these lines, but the take-home message from this slide is that from the different references which I have done so far, most of the time the hydrogel, along with some of these growth factors, would complement for the development of the tendon or differentiation of the stem cells into tenocytes or the tendon-specific progenitor cells, or TSPS. Okay, so TSPCs. So that's a take-home or summary of the slide. And moving on, again, tendon tissue engineering, we have already dealt with a couple of things about signaling, that is the TGF-beta BMP signaling already. And then again, the extracellular matrix, which is this combined, it can be either hydrogels or peptides or like scaffolds, anything of that sort, which we have not tried it in the laboratory, but we will plan to try again. And then other side, there is options. If you want to get, once you get the organoids or the tendon, there is another story that we need to put these organoids in place in the specific location that is actually in the body, in a live organism, especially in vivo, if you want to do in vivo experiments in the rat models or the or higher animal models. Then in that case, we have to make tendon injuries on those animals, similar to what actually the injuries that we get to the time of sports or accidents. And then we need to have some scaffoldings. We need to have some means by which we can incorporate these organoids which are developed in vitro into those scaffolds, and then place them in the exact location where the wrist tendon injury has happened because of the movement. And those regions, especially like the tendons, ligaments, they are like structures which allow the movement of the animal or the organism. So it's very hard to get them in place and then stay them in that location. So we need to kind of immobilize that limb or that area for quite some time, a couple of weeks or months. And then just like what we happened like in the case of fractures, and then let the organoids develop further, just like what they develop in vitro. They have to develop further in vivo conditions too. So that placement of that procedure, but that we are not doing, not planning to do in our institution, but we have got an overseas collaborator, and they have specific strategies for doing that, or taking our tendon organoids and then putting them in the animal models, especially in higher animal models, and then seeing whether those organoids can be able to develop and replace and generate the tissues for the tendon. So that would be like, if you are able to do that on both sides, like what is happened in India and something happening in overseas, then it would be like something like the tendon organoids will come into reality. And the good thing, especially if you're using this matrix, like the matrigel, which is very popular and very commonly used for generating this type of extracellular matrix, or this type of experiments. This matrix itself is coming from rat, and basically the rat sarcoma cells, and those contains, the matrix itself contains a lot of collagen 4, and that would itself be complementing for the stem cells or to differentiate into organoids, and organoids that can develop into tendon fibers, which is rich in collagen. So almost more than 90% or 99% of this tendon or the ligaments, it is basically collagen. So a lot of times, if you're using this extracellular matrix, we can of course get this extracellular matrix is already there, and then it will allow this kind of tweak the stem cells develop into organoids of tendon, and those organoids, once we put it into in vivo systems, they would be able to develop into full and to replace or regenerate the lost part of the tendon, and it will be like any kind of new thing, developing tendons, the organoids of tendons. So that's something which we are looking forward. Apart from that, apart from the extracellular matrix, apart from the growth factors, we have got some other more candidates, which I have told, like the photobiomodulation or the sonobiomodulation, which I'm going to talk to you in the next couple of slides. I'll quickly go through it because of the shortage of time. So everybody know about what is electromagnetic spectrum, and everybody know how does the light works. The light also has got effect on biological systems. And that's why sometimes we go and stay in sunlight, in the good sunlight in the morning time, to get some vitamin D. synthesized in our body. So the importance of light is so much. I mean, I don't want to explain this now, but at the same time, you also know that how does electromagnetic spectrum look like? There is like a very short visible area of wavelength of light that we can of course appreciate. But apart from that, there's a wide range of wavelength, starting from gamma rays, X-rays, ultraviolet, and infrared, microwaves, radio waves, which has got different or variants of variable wavelength. So wavelength starting from say this 10 raised to minus 11 meters, which is like this is like in nanometers. So this is if you come to this place, that's like 10 raised to minus 7, which becomes like 400 nanometers. And starting from 400 nanometers to 1000 nanometers, that is the light which we are already seeing. Otherwise, the rest of the lights, NASA used these radio waves to see the extracellular planets, but the X-rays, this used in the medical field to take X-ray. So otherwise, we are not aware of any of this rest of the light. So coming to visible light range itself, we will find the violet to red, and within the violet and red itself, blue or violet, there is some effect. This violet has got some effect on biological systems. Likewise, the red also has got some effect. So that effect of this light is actually because of the different receptors, the photoacceptors, not receptors, photoacceptors on our body. So what are the photoacceptors? They are basically chromophores. Some of this, they are the light absorbing molecules, the chromophores. So there are also light scattering elements, which are like the cells, organelles, fibers. So once we look at this electromagnetic spectrum, and then if you see the visible range of light is falling on our skin, it can be from different sources of different wavelength. Say under the skin, if it's coming on the skin, falls on the skin. A lot of things happens here. One is reflection, diffraction, absorption. And you know what is all of these. And absorption is something which is what is interesting for us. So the absorbing molecule is a chromophores, and the scattering molecules, of course, the light gets scattered and move around. So don't worry about that. I mean, the absorbing molecule is something which is more important. So apart from the light, there are light can be from different sources, as I told. The light can be from, it can be normal light, which is non-directional light, just like our LED light or incandescent bulb light. It can be it's non-coherent and polychromatic. What is non-coherence? What is coherence? I'll come to that. And again, there is light can be laser light, the light which is like unidirectional or single directional, and coherent and monochromatic. So in simple terms, monochromatic. What is monochromatic? It has got only single one wavelength going through. But it is polychromatic. There are multiple wavelengths, not just one wavelength, multiple wavelengths. So it's like a mix of wavelengths. That's what we get from the LED or the incandescent bulb in our home. But the laser is like monochromatic. It's coherent. So in that case, that's why we use the laser pointers for presentations. But these days we are doing all online presentations, and we don't need laser pointers. But the laser pointer is actually like basically it's a laser which is like the red light or the red light laser, or it is actually basically coherent, and it's directional, and it is monochromatic, single wavelength, and that's why it is very well focused. It just falls at single point. All right. So what is the effect of this light on biological systems? As I told, the laser the light affects the mitochondria, and mitochondria there is Krebs cycle, which produces ATP, and also it produces oxides. Oxides, the free radicals are called the tertiary messengers. The tertiary messengers, which actually kind of interact with different other signaling pathways, and it can actually, in fact, influence this protein kinases C, D especially, and coming from the adenylate cyclase, the enzyme on the cell membrane, and actually modify the gene expression. So this is like a very generalized picture. I will come to the details in the coming slides. And again, this laser or the light, it changes the biological system, the process called photobiomodulation, and also the chromophores. The chromophores are the light absorbing molecules, and the transducers. We'll come about some of the transducers in the next few slides. So when the cell is remaining like this, undisturbed, it is quite happy, but you see it's quite happy, but actually it is not. It's a lot of things going on, especially from the point of view of the free radical or the oxidative stress. The cell balances itself, oxidative stress, by producing a lot of antioxidants. And a lot of times these free radicals are generated from many of these organelles in the cells, starting from the nucleus, mitochondria, peroxisome, everything. And so I'm not going into details of each of this. It's quite a lot, and it's biology 101. So you know that this free radicals produced is quenched by antioxidants. at the same. So its cell always remains like in a balance between these oxidants and antioxidants, and that is what we refer to as the cellular redox, the reduction and oxidation status. And that is a normal state, what is called a cellulose hemostasis. Right. So once you come to the photobiomodulation, that's what we are talking about now. You will see once the laser or the light, low intensity laser irradiation, which is called Lily, once it falls on this mitochondria, the mitochondrial complexes 1, 2, 3, and 4 get affected. And there's this electron transfer. The speed of electron transfer increases. Why? Because light itself is photons. Photons is electrons. Electrons falling more electrons, stimulating more complexes, the respiratory complexes. Then coenzyme Q, cytochrome C, and then basically more electrons, the more energy, more oxides, more free radicals. The cells, the redox status get imbalanced. So if you get imbalanced, there is good things and bad things. Again, it's like a double-edged sword. The oxides are double-edged swords. They have got good effect and bad effect. If you harness the good effect, is beneficial for differentiating stem cells. That's the potential of this light on our biological system, and it's very crucial to know this. Different oxide radicals are produced, and they're produced in a variable amount, and that can be either beneficial or harmful oxides. That will come to that later, and that can be harnessed for differentiating the stem cells. All right, so I'm going quite fast. Sorry about that because of the time shortage. But I hope you understand. Again, this is biology 101. You know, once this regularly, if there is normal situation, if there is like there is electron transfer happening between the complexes 1, 2, 3, and 4, and the fourth one is the cytochrome C oxidase, and there is electron transfer occurring between, and there is a proton gradient that is increased hydrogen because of the Krebs cycle working over there, and it pumps a lot of hydrogen ions into the intermembrane space of the mitochondria, and where actually the complexes sit, and then there is electron transfer through these complexes, the subunits, especially the complex 4, cytochrome C oxidase, and there is like this copper A, copper B, and then the hem A and hem B, and hem A3. There's like copper A, copper B, 2 subunits, hem A and hem A3 is another 2 subunits. All right. So then technically what happens is that there's a normal amount of these electrons passing through this place. And in case if there is any increased, or if there is a light falling on it, if there is any stimulation of this cytochrome C oxidase, which is especially light, which is like photons, which produce electrons, which are actually electrons, photons are electrons, and electrons pass through this in a higher level, in a higher frequency, in a higher rate, which actually stimulates a lot of things, which actually increases the ATP production. And that ATP itself is something which is very interesting to know what actually is doing. More ATP means something doing more. Cells are tending to do more. All right. So that is what is being told almost a decade back. One Russian scientist, one of my mentor, she actually kind of plotted what is actually happening between the subunits once the light falls on this cytochrome C oxidase. How much oxidation reduction is happening. So when she plotted the wavelength of light, you can see the wavelength starting from 300, 600, 900, up to 900, which is a visible range of light. Then you will see how the blue light, then the red light, how the oxidation reduction takes place between the copper A and copper B. See the frequency, this changes. And so that tells him that the amount of light falling on it, on the cytochrome C oxidase, and that is sitting in the inner membrane of the mitochondria, has got some effect, some oxidation reduction happening, while if you are inducing light on biological or living cell. Okay. So that's, she's retired already. She was one of my mentor, and she has already plotted a lot of these things in 1988, changes in redox properties and acceleration of electron transfer. That is one thing, and then there's something of hypothesis which she established. This nitric oxide release from catalytic center of cytochrome C oxidase, that is nitric oxide hypothesis, superoxide generation, photodynamic action, changes in biochemical activity induced by local transient heating of chromophores. So the chromophores, again, this light absorbing molecules, and the effect of this in the redox changes on the redox status of the cell, the reactoxin species generation, as well as the heating. So there is variable properties on the biological systems has been addressed by changes in the free radical concentration. And that is what she has been published in 2007. And all of her research articles has been like compiled into a book. And it's a very beautiful book and one of the Bibles of the phototherapy. And so we actually were interested, actually, once once I was working with my mentors, I was interested to know what is the effect of this red light and the near infrared light. So as I told, this comparing the red light and near infrared light is something which I do. But of course, there is blue light also doing some other effect, affecting some of the vitamin synthesis on the cells. But I'm not interested in that. But I'm interested in the red light and the near infrared light because of the reason that they have got a better or higher wavelength. And they got a better therapeutic effect, which we'll come to see in the next few slides. So you see, if you put red light, there is not much change in the reactive oxygen species. It's almost down, much down. But interestingly, if you put this near infrared laser on a living cell, on a biological cell, not very high energy, but a moderate energy starting from 5, 10, 15, 25, you will see this is something like interesting which is going on here, which is the reactive oxygen species level, the ROS, the ROS level, increases at 5 joules, but drops down drastically at 10 joules, and then it increases further in 50 and they almost stay stable. And this has been further investigated by the team in the Harvard University, where actually they have looked at different concentrations starting from 1 joule, 2 joule, 3 joule, and 5 joule, and goes on until 10 joules or 15 joules to see, and they found a dose response. So basically, this actually goes like a dose response curve, where there is a reactive oxygen species production at 5 joules, and then it goes down to 10 joules, and then it goes up. And so there is like two things happening here. There is the cells cannot take beyond 10 joules, 15 joules of a near infrared laser. Because beyond that, the cell actually kickstarts, these caspases all start working and cell undergoes apoptosis. Because then it's a different pathophysiology happening for the cell. So this is something what we need to kind of look at. This is like the good part, is a beneficial part of the light on the biological systems, producing the reactive oxygen species or the free radicals. And once you go to this side, actually it is more cell-killing. It's more cell-killing, and that is something what we want to explore that in the cancer stem cells, where how can this light be kind of useful for killing the cancer stem cells? Because by increasing the light or increasing the energy level, the cancer stem cells, there is free radicals produced in the cancer stem cells, which actually can kill them, because cells will perish beyond this. After 15 joules, the cells will perish. So this is something which we kind of harness for differentiating the stem cells, especially differentiating this into different other cellular layers, like the same layer of mesoderm, or it can be different layer of mesoderm, like ectoderm or endoderm. So we explore that further. However, this good part of this double-edged sword, which is the reactive oxygen species or the free radicals, would be useful in differentiating the stem cells, which is quite interesting. But before that, I want to talk about that one thing. There is this phenomenon that you saw in the previous slide is something like what is called the Arndt-Schulz principle. This is something which is like almost half a century before. Scientists have found that if you increase the dose beyond the dose, there's dose response, and beyond a certain dose, then there's a drop down in the situation. So this is something beyond 5 joules, beyond 4 joules, there is like a drop down in the amount of free radicals. So the strong stimuli of the light would inhibit physiological responses, and that is something like this, the harmful part of the light, which can be harnessed for killing the cancer cells or the cancer stem cells. All right, and again, this one, there are different chromophores, and as I told in the light, blue light, red light region, there are different chromophores. The effect of the chromophores, like the oxyhemoglobin, deoxyhemoglobin, melanin, water. So this water actually is very less absorption of this light by water or water molecule, but it's also shown in the logarithmic scale, whereas others are on the arithmetic scale. So the blue light, it's, of course, there is higher polar extinction coefficient and it's higher level absorption, but more therapeutic effect. More therapeutic effect of the light is more seen towards like the 1000 nanometer. So once we put this into practice, I'm not going into so much detail of that because time constraints, but we'll come to that later if there is any questions. Then once you look at this bioenergy level, again, with the red light and the near infrared laser, again, as I told, the light produces energy. It not only energy, but also free radicals. So the reactive oxygen species. So once, of course, it's interesting to know what is the effect of the light, or especially the red light and the near infrared laser on a production of ATP. So once we look at the same thing, the same parameter, we also found that this ATP levels increases. See, I told in the previous slide that Beyond 15 joules, the cells will start dying. And that's what we see here. Beyond 15 joules of energy in the near infrared laser, if you put on any cell, any living cell, whether it is a fibroblast, this is done on fibroblast, not on stem cells. And so this actually, if you put on the fibroblast, you will find beyond 15 joules, the cells will start dying. So the same thing for the stem cells too. So the biological systems now take beyond 15 joules of light energy. As for the beneficial part of the light, which is like the free radicals and the higher ATP production, will happen within 15 joules. Beyond 15 joules, free radicals burst, and then it's like a total collapse, and then ATP levels goes down, the Krebs cycle falls down. And so once you compare and then put everything together, then compile, and then you see how does this affect the signaling pathways, then you'll find this the reactive oxygen species effects, especially red light and the near infrared light. The near infrared light is slightly darker, it's slightly invisible, and beyond the near infrared light is infrared light, and it's, of course we cannot see infrared light because it's beyond the visible region in the electromagnetic spectrum. So, and again, there is a water layer here. You can see how much the water layer would be like affected. Because water layer actually complement for the absorption. So if you're using near infrared layer, it of course there is increased absorption with the water layer, and also it's also complement for increased absorption of the near infrared laser and increased production of this ROS and bioenergy. So this near infrared laser is something which is very interesting to look at to see whether they can be used for producing a reactive oxygen species, and free radicals is double-edged sword. And at the same time, whether there is kind of instrumental for producing ATP, higher amount of energy in the cell. Right. So that's what one of my mentors in Johannesburg, she actually kind of helped me to put me into this research, where she was looking at the photomedicine and stem cells and shown us the Janus phase of photodynamic therapy to kill cancer stem cells, and photobiomodulation, which is used to stimulate the normal stem cells, which is used for differentiation of stem cells. So we worked together, and then we got around this model of how does this ROS affect this different other signaling pathways. It's too much detail, I cannot go through that now. Again, different inducers like growth factors, neurotropins, morphogens, how would that be useful in differentiating stem cells? On these are all been published, so these are reviews already gone through. So this talks a lot about how much these growth factors can be useful, and especially the role of the growth factors in the proliferative phase, especially these growth factors in this side of this slide, and neurotropins to some extent they actually proliferate stem cells, whereas the neurotropins with the morphogens towards this side, it would be like more differentiative. So if you're looking at a neuronal differentiation in vitro, and then getting stem cells differentiated into neurons or neuron-like cells, or with functional neurons, whichever, you want to induce injection with external or environmental cues, especially with growth factors or these neurotropins and morphogens. And you can see a lot of literature lingering around this area. They use these growth factors in order to stimulate differentiation of stem cells. And but in case if you use more of these, the receptors, the ligands for these receptors, then if you stimulate these receptors, then be more proliferative action of the stem cells. Okay, it's been all reviewed, not going into detail of that, it's too much. Again, if you put light, there are a lot of things happening. I'm going to summarize here things. Again, free radicals produced, it acts on the adenylate cyclase enzyme. Then further on, production of cyclic AMP, and then the cyclic AMP again, it affects this protein kinases, and protein kinases again get transferred, and then gene expression. And again, if there is increased reactive oxygen species of the free radicals, it stimulates the production of calcium, increased calcium entering into the nucleus. Again, so this signaling itself will change a lot of different, which can either proliferate or differentiate according to the presence of the growth factors in the media, which are growth factors. And also the production of calcium, and the calcium affecting different calcium affecting different other pathways. Also the calcium entering into the nucleus, and also the calcium effect of the calcium in the cytoskeleton will change the stem cells or make stem cells to differentiate in vitro into different other cellular cell types, or like these are differentiation or transdifferentiation. So previously it's been shown like how does this differentiate with the light and other growth factors into muscle cells, especially smooth muscle cells. These are some of the transcription factors which they found are upregulated by a microarray study. And then later on, there is also like, of course, this is like a mesodermal cells, and this is like ectodermal cell. So differentiating the stem cells into trans differentiation, this different layer of the gel, different germ layer, this trans differentiation, or differentiating mesodermal stem cells or mesenchymal stem cells or mesenchymal stromal cells into neuronal cells is higher level of task. Unlike differentiating them into the same germ layers, which is like the osteochondra or the osteocytes, chondrocytes, or muscle cells. All right, so whatever it is, if you get adipose-derived stem cells, which are of course the mesenchymal stem cells, we can work out a plan, because previously there were people who showed before, like how does the light at the 5 joules here and this 5 joules here, and it's like using the growth factors, and if you use a co-culture, how does that affect this differentiation in the same germ layer, mesodermal layer? So one of the tasks which I did there was kind of see if there is like this near infrared light, because this is a laser actually. This is actually a red light. So this is 636 red light, and this is 825 nanometer. It's a laser. Whether it would differentiate them into neurons. Okay, so then that's a big ball game, because that's differentiating the stem cells, which is mesodermal in origin, into ectodermal gem layer. All right, and then going through that, we put hypothesis, and we got the stem cells from the fat layers by the abdominoplasty from the human beings who undergone abdominoplasty or tummy tuck surgery. And then in hospitals, and then we brought them into a laboratory, and then we separated them by centrifugation, and then we also got immortalized from the ATCC as a control. And we found that getting them into spheroids, clusters with growth factors, and then provide laser irradiation. This is laser, is near infrared laser, and then getting them transformed to human neurons. And that is actually the summary of the methodology. So it's very, very broad. And then again, if you get the adipose stem cells, the adipose cells separate, and then stem cells, of course, characterization based upon surface markers, and then looking at the neuronal markers which is on the surface. So there is like ADSC, which is the stem cells, and then what's the differentiating neuronal precursor cells, or the NSCs or the NPCs, or the neuronal stem cells. They actually get changes in their gene expression as well as the protein expression, and further on they get into mature neurons and glial cells, and those ones they got variable different morphology, and again they get more differentiated, as you saw in the previous presenter has given a detail about how all this kind of neurogenesis happens in the in vivo situation, and she looked at the transcriptional factors and all those things. But in this case, actually, we're looking at this different genes and as well as these proteins, and how does this each of us, not just neurons alone, there is glial cells which forms 6 times more number than neurons, and also this different other glial cells, and also the neurons, which are like depending on neurotransmitters, how they can be classified. So it's quite a lot. So we are not reached or nowhere near that. We are just only able to differentiate the stem cells into neuron-like cells at the moment. And some of them have some of the researchers have shown the functionality of it. But in our case, with the light, especially with the light, we have found there is the 5 joules of light, especially when you look at the neurons. This is like an adherent culture model, but it's a suspension culture here. So 3D cell culture models, actually, there is a significant increase in the neuronal marker, one of the neuronal markers. This is one among several other neuronal markers is representative. More of them has been published already. Not yes. This is comparing between this adherent culture and suspension culture. So this talks about how does this light on a suspension culture, especially using this using the spheroids, how does this suspension culture would be useful in kind of stimulating? If you stimulate with the light, how does the neuronal markers, the level of neuronal markers, once you normalize, light itself will induce some level, almost 10% increase of this expression or differentiation of the stem cells in neurons. So that's indication that the photobiomodulation, how much it is doing. And this is quite a lot of things to say, but I'm just skipping a lot of things here. Everyone know about this Krebs cycle and this glycolysis. And of course, in stem cells, one of the things, if you look at the metabolic part of stem cells, you will find the glucose. And most of the time, the stem cells, once they proliferate, they always almost resemble like the cancer cells, because they show the Warburg effect. The Warburg effect is something like the glucose, lot of glucose, the glycolytic flux, and the flux actually just goes through, and a lot of lactates are produced. And that's because of this LDHA and LDHB. And in 2018, yes, some group in California, they have shown beautifully well, and their publication is in eLife, which shown how does this variable different enzymes level change in the stem cells, especially while they are differentiating. So during the time of neuronal differentiation, one of the important things that's happening here is that a lot of these inhibitions will go off, and then because inhibitions have gone off because the growth factor as well as the stimulation with the light. And then technically this amount of lactate produced is reduced, but amount of glucose getting diverted into this Krebs cycle is more. Krebs cycle will work faster, oxidative phosphorylation is more, and that indicates that there is a switch in the metabolism from the glycolytic cycle, from the glycolytic flux to the oxidative phosphorylation. That indicates that itself is a trigger. That itself is a preamble for differentiation. And so metabolic change happens, followed by first is transcriptional change, then metabolic change, and followed by the phenotypic change, and that is manifested as differentiation and change in the phenotypic markers or the cell surface markers and manifestation neurons. So we look at different real-time PCR and then look at this cells which are like adherent cells which are like in suspension, and then the differentiated neurons using the stem cells, and we found there is the amount of flux, the amount of fold change is so much in some cases. There is a definite huge change, like this is 100, this is 1,000. So there's a huge change in some of this. This is like actually these are like the GPX3, these antioxidants. There is some antioxidant fluctuations, the T-form and all those things. It's already been published. And so some of these antioxidants get higher level in suspension cell models compared to adherent cell models. And those then kind of try to correlate which of these enzymes, this LDHB, then PKM2, which of these antioxidants or antioxidant genes get upregulated or downregulated in the systems. All right. So then again, coming to summarize this, and that's one side of the story. As I told, the glycolytic flux here, and you can see this Warburg effect, just like in the cancer cells, and then it goes down, and then it goes down to, in this case of differentiating stem cells, there is oxidative phosphorylation here. This glycolytic flux, there's change, and then it becomes oxidative phosphorylation. Further on, that's not the only thing here. Again, further, sorry. So again, light onto the mitochondria. Again, there is inherent ROS which is produced because of the oxidative, because of the electron transport chain, that is ETC, but it's inhibited by NADH usually. So there is redox status balance. This redox status get disturbed with the light or it, and once redox status get disturbed, that itself is a trigger to change metabolism, and that is actually manifested and if you add along with that, so that if you add along the growth factors like the polyunsaturated fatty acids, so growth factors, polyunsaturated fatty acids like omega-3, omega-6, there's a lot of changes happening in the cell cyclings, the CDKs, and this growth factor levels, this cell signaling pathways, and some of them has been observed, and we can find that in some of the literature. People, some group in Israel, they were trying to use this omega-3 and omega-6 fatty acid, and then finding out whether that would actually increase or stimulate the differentiation of stem cells to neurons. So there is quite a lot of interest in this area at the moment to see whether, along with growth factors, with or without light, whether these polyunsaturated fatty acids can make a change on the differentiation of stem cells. I'm closing time, sorry. And then coming to this towards the last part, of course, this has been done, the autologous stem cell therapy, you know about that. Dr. Biju George has mentioned about this, what is autologous, what is allo, how does this works in the human beings, especially in the bone marrow transplant. So, but if you're in the same phase, in the same type, if you're taking lipoaspirate, you can get adipose-derived stem cells, and we can obtain the stem cells, the mesenchymal stem cells, or as we heard in the morning, which is like very small embryonic VSCLS, embryonic-like stem cells. So whichever it is you obtain, and then further on, then it's our job to enhance and to make use of this part of growth factors or the light or the fatty acids, and then differentiate them and put back into the patient. And this is kind of a summary of all the things. And apart from that, the last couple of slides of the talk, one of my mentors in the Boston, he was like, we all published some of this, and then some of these, in case, not apart from differentiating stem cells, in case this light also has got some effect. So transcranial neurogenesis using the photobiomodulation, because if you just get heat from your enemy, for whatever reason, sorry about that, and then you will find the neuron excitotoxicity, whereas these neurons are having a kind of damage, and then that damage will kill the neurons. And the bad part of the neurons is the neurons will undergo apoptosis, and once they undergo apoptosis, there is no neurons to replace it, because the neurons cannot be replaced. So basically, then the patient will undergo a paralysis, or it might end up in some disease conditions. And so not just this heat, again, it can be because of sports injuries, then it can be because of accidents, it can be because of old age, and whatever. So a lot of things have been worked out in that, and then it has been found in one of the recent papers show that the near-infrared transcranial intranasal photobiomodulation significantly modulates the neuronal oscillations. So I'm not going into detail of that because how does this work out is basically how does typically just like what I told here, the light stimulating the mitochondrial, the electron transport chain functions, then the cytochrome C oxidases, the respiratory complexes 1, 2, 3, and 4, and then increase ATP production here. And then again, light, of course, they actually can has, along with growth factors, increase neurogenesis, and it kind of suppress these cytokines, especially inflammatory cytokines. So one of my mentor and co-publisher of many of my couple of my articles, he already retired. He was in Boston. He actually found whether it's interesting to put a kind of helmet of light. Unfortunately, we cannot use laser. So he was saying, do you need a laser? Actually, can we do that? Can red light be doing this thing? Of course, because we cannot use this laser like near infrared laser because of safety reasons. And so it's always sometimes better to use a red light, how much it can be like useful. So how does this kind of bring about? But again, the problem is whether it will cross the skull. But anyhow, they have found he has published some reviews, beautiful reviews, and some data also, of course, how shining light on a head, how does photobiomodulation useful for brain disorders. Then especially this kind of Alzheimer's and Parkinson's and all those patients, you will find them having this kind of light helmet. And well, it's interesting to know how does this work, and I recommend you to go through these reviews. It's quite detailed to talk about this. Again, once you go to summarize the effect of the light, once you kind of put, then what happens is that it gets higher on a chromophores, electrons falls, the chromophores get into a higher and excited stage, and then goes to second excited stage, and then once it comes down, it emits fluorescence. And again, that fluorescence actually is manifested as like there is a lot of things going on here, like the fluorescence times emit light, and that also like a stimulation, like this photostimulation of the fluorescence because of the photosensitizers that you use, especially for the cancer cells, especially for treating or killing the cancer stem cells. So some of the things, aspects here is like directly connected to the effect of the light, which is harnessing the production of the reactive oxygen species for killing the cancer stem cells, which is the other side of the story. So I'm not going to talk about that in details, but I just want to mention here, not just the light, even the sound got that effect, because even sound produces has got some inertial effect, this cavitation effect, this cavitation inertial, non-inertial effect. And if you use sonosensitizers, again, it can produce these free radicals, and it can produce apoptosis, necrosis of these cancer stem cells or cancer cells. And then technically it will be like killing those cancers. And a lot of work has been done, a lot of publications, tons of publications you'll find starting from 1980s or even beyond that. Using the sound, ultrasound for killing the cancers or the cancers or reducing the cancer growth. So all works with the one single thing, which is the reactive oxygen species or the free radicals, which I told in the very beginning is a double-edged sword. So until previous slide, we have seen how much this reactive oxygen species is useful for differentiating stem cells. But now this slide you will see how much this reactive oxygen species is harmful for the cell. And it can be harmful, especially, and it can be good side of that if you harness that effect of the higher level of reactive oxygen species or higher production or higher generation of free radicals in the cells, it can be used for actually producing or affecting this or killing these cancer stem cells. Again, this is something is sonosensitization, which also we believe, just like what we have told before, sonosensitization also produce these reactive oxygen species or free radicals that would in turn would have an effect on NF-kappa signaling and AP1 signaling. These things are something which we are hypothesizing. There is no literature available so far whether the sonosensitization would affect the stem cell differentiation. I have looked left and right in the literature, but this is something quite interesting, whether this would be knowing. Of course, sonosensitization is used for killing the cancer cells and cancer stem cells, but never been used the good part of that for differentiating the stem cells, which is only for killing. So it's killing and leaving. So it's like two things happening here. So that's why it's called double-edged sword. Double-edged sword is a reactive oxygen species, and the reactive oxygen species and the reactive nitrogen species, they are actually like beneficial and either can be harmful. And we try to classify which are the beneficial reactive oxygen species or free radicals compared to the one which are like harmful here. All right. So that's actually, it remains a redox window. And there is ROS scavengers, which is like antioxidants here, and then the ROS generators here. And that is what is happening in the cancers. And we try different medications, and then we work around that. And then we get frustrated most of the time because we really don't know which of these free radicals produced are harmful, which of them are beneficial. So the harmful would be one which is killing the cancers or cancer stem cells. And a beneficial one would be like helping the normal cells to restore, to regenerate, or to differentiate. So that is something which we still, there is some, because these free radicals are ultra short lived molecules, and it's not easy to kind of harness them and look at only the after effect of the production of that. So that is one of the technical limitations that we are facing. We cannot even look at this very closely into what is happening in this reactive oxide species, nitrate species. In many of the stem cells and cancer stem cells, but at the same time, we are also not aware of whether this is actually, some of these free radicals are beneficial, some of them are really harmful to us. So that is actually the double-edged sword, or the two-phase, or the Janus phase. So in the Greek literature, you will find the Janus phase. There's a god called Janus. Janus has got two phases. It's a good phase, soft phase, nice phase, and then the bad phase will get angry. So that same thing you will find these days with the people within all walks of life, right? So anyway, right now, our Prime Minister got the Legion of Merit from President Trump. But it's again, this like the Janus phase of the Indo-US relationship. And that's what I need to talk today. And I'm done with that. Thank you so much. And I think I have shown my Janus phase. It's time for you to show your Janus phase. Thank you by asking me questions, please. Thank you. Bye.
Dr. Sargent, thank you for your enlightful talk. I have a simple question to you.
Okay.
Do any organ development using 3D model is successful as of now for organ therapies?
Yes, they are working on intestinal 3D models like the intestinal spheroids, these organoids, and whether they are seeing whether especially some of the labs in California, and especially some of the labs in Netherlands. Hans Clever's lab is very pioneering, and they are using the 3D model system, and they are basically using for drug screening and disease modeling, but potential of using that intestinal miniature organs for regenerative medicine is a little bit longer, wider goal. But at the same time, if you can get them into structures like tendon or ligaments, that will be much more easier than putting those 3D structures directly into like or replacement of internal organs, which is quite more complicated than tendons and ligaments.
Do we need to follow any ethical clearance to develop these organs?
Yeah, no, we don't have to know this, because the ethical clearance, not only that we need is to kind of get to know what actually, from what source actually we need to get these cells, whether we are getting the cells from human beings or whether we are getting the stem cells from other animals, laboratory animals or farm animals. That is ethical clearance that we need. Otherwise, for conducting this kind of differentiation assays in vitro, or conducting this kind of therapy, this kind of differentiation procedures, that we don't need any ethical clearance. And further, once we try to put this back into animal models or human beings, again, that side also we need ethical clearance for putting these organoids generated in the laboratory onto the animal models, because in that case, we are actually kind of sometimes removing the organs and putting the organs which are like generated by organoid development or 3D model systems in the laboratory, which would kind of another ethical violation there. So that also need to get the ethical clearance. So two sides: once the source of getting the stem cells, and then differentiating in the laboratory, making it organoids, it's not a problem. That's something which you need to work out experimentally. But then putting those organelles for replacement therapies back into organisms or living cells, or into living organisms or lab animals, human beings, then again, ethical issues. Yes.
Thank you, sir. Now I request Professor Suresh to moderate the question and answer session.
Thank you, Dr. Asha Devi, Madam. Dr. Sargent George,
thank you.
A lot of information, and you tried to summarize. Believe it or not, we have about 7 questions, Dr. Sargent George.
That's wonderful. Okay.
One after the other.
Yeah, sure. Yeah, the time allows. I'm sorry that I crossed the time, but I guess I can quickly answer some of these questions because it's quite important. because I understand a lot of students are there, and that's why there are a lot of I have put a lot of slides with a lot of animations to make them interesting and make them stay in the late part of this evening.
Yes.
All right. Yes.
The first question is from Munasa Rashid.
Okay, sir.
Why do tumor organoids develop from adult stem cells?
Okay, this developing tumor organoids is something for the simple reason we need to perform the drug screening and also to delineate the disease physiology. The cancer, especially tumor cells, will be a good tool if you want to perform a lot of biophysiological study and also to conduct a drug screening. That's the most important thing, because these days the drug screening conducted using these 3D model systems and the organ on a chip, they are more labeled, easy to publish compared to what they kind of get this drug screening or high throughput screening, which you have done on 384 well plate or high throughput or medium throughput screening. So one of the reasons is the science is moving towards developing 3D model systems, and that's why the engineering of 3D model systems is become so much important. And also that's why people are having a lot of interest in finding out how engineering this 3D model system is or how finding the scaffolds, extracellular membrane, extracellular matrix, or different biomaterials, especially. That's why there's a lot of interest in that direction, because that's where actually we need to explore further and find out whether we are able to mimic the organ in the laboratory. Using the patient-own stem cells, it would be great if it is possible. Can basically make those stem cells and then send the stem cells deceased and then try the disease, different gene therapies or drug therapies, whatever you like to do. Is that fine?
Yeah, I mean, you talked about matrix. There is one question from Mr. Surya S.
Yes, yes, sir.
Along the same lines.
Okay.
Considering decellularized adipose cell matrix, DACM, from donor's cadaver is the latest technology in tissue engineering and bone engineering now, with only one FDA product is approved right now. What do you think of DACM, that is decellularized adipose cell matrix, plus low-level laser wave therapy to address hypoxia?
Yes, that would be like something which I would, that's not my, because these areas of that decellularized, I mean, this is a bit complicated answer. It's a complicated question because to be honest, I'm not very well versed. It's at the moment, it is beyond my expertise to answer that question, but still I'm trying to answer that. The process of getting this decellularized cells from the cadaver and then putting it into into a material or biomaterial which can be used for therapeutic purpose and developing as a product for therapeutic purpose is something like a wide, it's like a long road. At the moment, its interest, what I'm talking about, especially in this presentation, is something like developing the model systems, which would help for tissue engineering, which would help for actually kind of putting those models into practice in the laboratory so that we can generate organoids and disease modeling or drug therapies. Getting them into a product and then using them in the human and animal species is a big problem, it's a big challenge, but I'm sure that that will happen one after another, each of the products in the due course. So again, this is just my opinion, and there will be more updates about it, which I'm not aware of it. I'm sorry about that.
Okay, okay. Next, Dr. Kavita Thirumurugam madam has 3 questions.
Oh, great. Okay.
One after the other.
Yes.
Question number 1.
Okay, madam. Yes. Sorry. Yeah.
What is the effect of near infrared light on aging senescent cells, human fibroblasts?
Yes, that's a beautiful question, and a lot of work has been done on that direction. The laboratory where I worked before, and some of them, a couple of those laboratories, they were exclusively working on that direction. And people also have worked about the effect of this photobiomodulation on the patients which are diabetic, and effect in those patients which have the difficulty in the tissue repair, wound healing, etc. And will this low-level laser irradiation or the photobiomodulation would help for the wound repair? Likewise, for aging or senescence, this is a very beautiful question, and there's a cosmetic reason. Cosmetic industries, they use lasers quite a bit, and you know about the reason. And most of those they don't know about the mechanisms. They actually work on a kind of hypothesis. They are misled. They actually try to use laser, and then they don't know how much the potential the laser, what's the good side and bad side, and because the double-edged sword, as I told. So people have found benefits. So it's just like a debate, just like using the mesenchymal stem cells for clinical trials, right? So people say mesenchymal stem cells for clinical trials, yeah, wonderful, it's beautiful, immunomodulation, a lot of things, and it's regeneration and rejuvenation and all those things. But at the same time, some people say, no, mesenchymal stem cell is doing nothing. It's just a waste of the people are cheating us. That's why people fly to South Africa, people fly to Thailand and spend their vacation there, get their stem cell therapies done because legislation will allow to do them. But at the same time, I think more than 50% of the cases they get failures. Some people, most of the people in North America, they go to Mexico to get this done because it's much cheaper in Mexico. And there is a lot of advertisement, people, agencies canvassing. And they all go with intention for the aging problem, senescence, and cosmetic reasons. And sometimes they get cheated quite a bit. So it's very tricky, depending upon the therapeutic effect, depending upon the patient, it's depending upon the individual. Sometimes, if it is done properly, appropriately, in the right way, in the right directions, right guidance, it works well. It works beautifully well, and that's what there is. I think I would redirect you some of this. He was my co-supervisor. He actually they have got this Wellman Center of Photomedicine in Massachusetts. So they are exclusively working in this direction. There's a lot of researchers. They actually work, and they have shown how much this light, especially near infrared light, has got benefit. And now they're trying to replace this with different other industry partners, which I'm trying to pull out that information from the web. Yes, in the meantime, I'm getting this web page. I think so. Yes, this one. So you will see this photobiology and photobiological science online. It's always there on my desktop. So a lot of things are about this photomedicine and this photophysics, a lot of things, and a lot of research has been done, and a lot of industries. You'll find, just Google it, you'll find the effect of photomedicine, photodynamic therapy, low-level mechanisms of low-level laser therapy. It's all there already, and people are working on it. And a lot of, in the athletics, in the sports, a lot of athletes, they use this low-level red light or near-infrared light, especially once they face some injuries as a part of physiotherapy. And if you go to physiotherapy places, so again, low-level laser therapies, you get less therapies, always, yes.
Thank you, Dr. Sarjan.
Yes.
Before we get to Dr. Kavita Tundukar Madam's second question, you mentioned about photo modulation.
Yes, sir.
Question about from Prarthana, how about complications associated with photobiomodulation?
Sorry, I didn't get the word before that. Sorry, sir.
How about the complications associated with photobiomodulation?
Yes, sir. Okay. Yeah, the complications. Practically, there is. If you are doing this, we are using the light, as I told, with the visible light. The red light, up to red light, absolutely no harm, absolutely no harm. So that's why people are using under red light. But once you cross the red light and once you go to the near infrared red light zone, that is actually what I'm trying to show in this electromagnetic spectrum here. Or give me a second, let me pull that out. Yes. So here you can find that, so under this violet or blue light to red light, absolutely no problem, no complications, nothing, no harm. But once you get into infrared light and getting into near infrared light comes here in this border, then it's quite a little bit harmful because you need to keep your eyes away from this near infrared because it can hurt, it can damage your retina. And if you go more and more further down here, this is actually not actually a dangerous zone, and radio waves, and especially when that's once you go to outer space, then always the radiations, like we are worried about the radiations. That's why these astronauts, they use a spacesuit. So this part is a lot of harm, and this part also is harm, but this is a safe zone, and this is what actually this biologically relevant. So there is no complications until if you use red light. That's why always in a lot of this, I did the comparison. I do the comparison, comparing all the time, red light, near infrared light. So red light, of course, there is no complications. There is no much reactive oxygen species produced also. So that's a sad part. But if you go to a little bit, if you take some risk, keep your eyes safe and then start working on near infrared zone, then you will get the benefits of this photobiomodulation. Otherwise, within the visible light, there is no complication. People try to stick, they don't go to this region, especially for photobiomodulation and phototherapy, because getting into this phase is dangerous. If you use an equipment in a hospital or a clinic, then there is safety issues. So that's why people stick only until red light. And a lot of times chromophores are the same, so it affects, but compared to near infrared light, red light is not that great.
Yeah, thank you, Dr. Sarjan. The second question from Dr.
Yes.
Kavita
Yes.
is how transient is the ROS production when near IR rays hit the cells? How long do the ROS live? The half-life of ROS?
Half-life of the ROS is 10 raised to minus 6 seconds. 10 raised to minus 6 seconds. So that's what they say, to 10 raised to minus 2 seconds. So what we are actually measuring, so I have seen some of these presentations even before. That was 1% ICE-TB. That is something which is conducted before the faculty development program. That is 14th and to 16th of December. So some of the presenters, they were like looking at the reactive oxygen species, and I asked specifically, how do you measure that reactive oxygen species? So actually, this is very well-known phenomenon. Actually, we cannot measure the free radicals because they're ultra short-lived. What we are looking at is after effect, the effect of the reactive oxygen species, which is done, which it makes on the other biomolecules. So, and that's what we're looking at the fluorescence. These assays are flow cytometry assays, and there is a lot of difficulty in getting this light, the cells irradiated, and then shifting those light without falling any other light. It should be done in dark, and putting that into processing the cells quickly, as quickly as possible, and putting them into flow cytometers. It's extremely, each of these assays are extremely lengthy, and each of these took almost a day. And they're starting from morning 6 o'clock to evening 6 o'clock, and so that's the sad part. I mean, looking at free radicals, ultra short-lived free radicals, it's not easy, it's not possible, but we are only looking at the effect of them on biomolecules, and that's the technical limitation which I tried to pull out in the very last slide. We are not even able to look at the reactive oxygen species and reactive nitrogen species, but we are able to look at only at the after effect, and that's a problem. We are not able to identify which is beneficial, which is harmful for us. So that's why we are sitting in the middle of the double-edged sword, and we are facing this Janus phase of this of the reactive oxygen species in everyday life because of the stress on us. Some of them get benefit, some of them are harmful to us. But we are living in that complex scenario.
Thank you, Dr. Sarjan. The third question is: ROS and oxidative stress are co-players.
Yes.
High ROS level produced by near IR or red light, what happens to oxidative stress?
Actually, this question is a little bit funny. ROS and oxidative stress both are same, I would say. It's not different because oxidative stress is a manifestation of the free radicals, which is increased ROS. So if there is an increase in ROS, reactive oxygen species, that is manifested as oxidative stress in the biological systems or living cells. So it's basically one and the same. One is a reflection, one is a cause, and the other one is effect.
Perhaps Madam meant the dose of ROS. At high ROS, what would be the response?
Yes, that's what I was talking about. That in the slide. Going to that again. So high ROS. That is actually a beautiful side of this. If you're looking at if you're using the red light, so that's why a lot of times people go and do this cosmetic for laser therapy, phototherapy. They get failed, frustrated. Nothing will work out. And some of the times they use the red light for treating the skin disease like psoriasis and some immune-related disorders. And a lot of times the patients undergo this phototherapy with red light. And then the psoriasis or whatever skin conditions or skin, like a lot of even starting from the speckles and this comedo and this all cosmetic things. A lot of times this does not work because people don't know the red light is not one which is useful for them. They have to use the near infrared laser, and they don't use the proper laser which is properly calibrated. So they use some different other wavelength, and then they take the money, and then people get frustrated. But if you're using the right laser and the right amount of time, like just like if you're using the lasers or red light or lasers instead of the red light in the brain injuries, then you will get the effect. You will find the beneficial rows and you will find harmful rows. So you are sitting in between this beneficial rows, which can do the stem cell differentiation, or it can complement for the growth and the properties of the living cells. But once you shift or once you go to higher end, if you don't use equipment properly, if you switch on the equipment for a long time, and if you don't do the proper irradiation intervals, proper time intervals, and then technically you are generating the harmful rows here. And harmful rows is actually doing more damages to your skin condition. So that is something which we need to know. I mean, once we go to some sort of clinics, sometimes the clinicians or sometimes the person who is doing this, they are very unaware of these things and they do more harm to us than doing anything good. So, but at least now onwards, we need to know what is a good rose and what is a bad rose. And this is not just happen because of the light. It happens because of drug therapies. It happens because of the sound, the sonosensitization. Sonosensitization also produces the similar amount of good and bad reactive oxygen species and push the cells in between the oxidative stress and the reductive stress. And that is a very beautiful part of this, especially once you come to look at the cancer stem cells. So we need to have all these things. Sometimes we work on, because we have seen so many lectures today. And one of the things Professor Krishnamurthy has told just before was that sometimes we are doing wrong things without knowing whether it is good or right. So if you don't do the good things in the proper fashion, you will get bad results. So this is a typical example. And that's the reflection of Janus phase of the reactive oxygen species or the free radicals. Thank you so much.
We have three more questions. One, one we asked directly.
Yes.
This question is from Prarthana Pradeep Mishra.
Yes.
Good evening, sir. Use of hydrogels may reduce the mechanical strength.
Okay.
What measures can be taken?
Use of hydrogels may reduce mechanical strength. Yeah, that's a good point. I mean, while you are using that, that's very genuine practical question. I understand that. This is what she's talking about. That here, if you're using the hydrogels, especially one of the examples is matrigel, suppose, and if you're trying to differentiate these stem cell clusters or stem cell aggregates in the matrigel, in the hydrogel, this probably does not stay like that, because just get dome shaped and then get flattened or the next day or next hour. So that's why this we need to look at this suspension method. For suspension method, we can get them into the transfer plates, where even if there is a proportion, like a 1:5 proportion. There is 1:5 proportion of adding matrigel to the number of cells, the volume of cells, which you kind of incorporate into this extracellular matrix. So if you are having like a 50 microliter of this cell aggregate, stem cell aggregate, which you want to differentiate into organoids or spheroids, and the spheroids into organoids, then you have to use around 250 microliter about this of the extracellular matrix, and then can be holding in kind of hanging drop or suspension method the transfer plate. So that would suffice and alleviate the problem of this loss of strength of this extracellular matrix while differentiating them. Yes.
Okay, before Dr. Debashis asks your question directly,
yes,
this question has been routed through Dr. Manjuwala Madam. I am not sure what technique this student had in mind, or this registrant had in mind.
Okay.
Question from Malar Kodi.
Yes.
What will be the major difficulty or inconvenience caused during practicing this technique? Do the cells degenerate? You talked about I think several techniques. I'm not sure which technique.
Yeah, which technique I'm not sure.
I think she's addressing the photomodulation.
Okay, photobiomodulation.
That can degenerate the cells.
Yes, differentiate the cells. Yeah, photobiomodulation. If you are differentiating cells, again there is again
degenerate the cells.
Photobiomodulation, degenerating the cells. Then actually you are using the photobiomodulation. That is actually what you need to look at. You need to read. This book is maybe expensive. It's not in our library. I'm not sure. This is the wonderful book. One of my mentor. So she actually both are my mentors. So they have published this in 2017. And then actually this book, you can see this one in the Google, at least the introductory pages, the first one chapter, I think. Then if you look at, then it says about the Janus phase or the photodynamic therapy to kill cancer stem cells and photobiomodulation, PBM, to stimulate normal stem cells. Okay, so what I'm talking about now is to stimulate the normal stem cells. Right. So if you want to degenerate or to kill the cancer stem cells, again, if you use this photodynamic, if you use this light especially the light is red light or this near infrared light, and especially if you use photosensitizers. So these days people are looking at these nanomolecules for nanomedicine. So there are a lot of photosensitizers, there are a lot of sonosensitizers. So those molecules which there are a lot of magnetosensitizers. So those molecules which are sometimes targeted, they have got molecules that they have got the structures on it which can direct them into the tumor cells or cancer stem cells, which are very directed, like for targeted killing of the cancer stem cells. Again, those molecules itself can be like photosensitizers. Lot of research, lot of reviews you will find in that direction. There is a lot of articles published. When I look at the 2005 review of sonodynamic therapy, like PDT, there is sonodynamic therapy, SDT. There is almost like 100 reviews itself in 2005 for using the sound for killing this cancer. At that time, the cancer stem cells were in the infancy, I think. And then now the interest of using the PDT and SDT is to degenerate, not cancer, looking at the cancer stem cells, specifically targeting the cancer stem cells, using nanomolecules, and then killing these cancer stem cells, targeting to the cancer stem cells, and then killing them in the presence of the light or the sound or the magnets. Okay, so this is one of the areas of research, and a lot of work has been done in that area.
Thank you, Dr. Sajan George. We'll take one last question from Dr. Debashish Mishra.
Yes, sir. Tell me, sir. Yeah.
Hello, sir.
Really, please tell me, how are you? Yes.
Really good to see you in this special cap.
Good to see you with this cap, right? Yeah, all right. Thank you so much. Yeah. Yes, please.
Merry Christmas
Thank you, sir.
to you in advance. It was really energy loaded, full of explicit images which can talk for itself also. But meanwhile, you were also explaining. There isn't any stone unturned. Just one simple basic query here.
Yes, sir.
I just want to know, as you have actively worked in PDT.
Yes, sir.
And that's the reason I'm asking here. See, we have two uses of IR waves as of yet. One is, of course, we do spectrophotometry or spectroscopic technique. We use, we see molecular vibrations, molecules react to it.
Yes.
With bit of molecular motion. That too, not the full molecule in motion. We have the parts of the molecules moving with respect to each other. That's one. Then there is one more application of IR we see here is in radars. We use IR in remotes and radars. The reason is IR don't interact or much with that of the material or matter. So meaning large obstacles are meaningless for IR. That's why it can go very far distances. Now here it is a conjecture we can understand. That means even if you give a good flash of IR, whatever photon which will actually interact with that of the molecule is actually very less, right? That is what the physics says. Now I just want to know that, is there any specific photo focusing technique you are using just to increase the number of photon hits to the cells? That is the first one.
Yes. That's a very very, very useful and very nice question. One of the limitations, I mean, actually you are directing me to one of the limitations of this, of the photodynamic therapy, the PDT. The major limitation of the photodynamic therapy is the ability or the inability, I would say, it's inability of the light to penetrate beyond the few dermal layers. It usually does not. So if you just for simple example, I'm just trying to say, if you put your laser pointer onto your finger, you can see it's like more your finger get reddish. It might go to a couple of centimeters down your skin, or you can see a couple of centimeters, I would say, but it won't just go into like different organs. the light cannot penetrate that depth below the skin layer. So if you are using the PDT, people say about using the PDT and then treating for cancer, especially if you want to use the colon cancer, for example, colon cancer or like pancreatic cancer. I don't know why people those are in vitro studies, and but once they put them those light or the photosensitizers into animal models, its light cannot go beyond a certain limit under the skin. That's important, the PDT, that's the inability of the PDT. And so some of the times we use targeted therapies and there's a targeted photosensitizers, but I would strongly recommend light therapies would be like limited to the skin diseases or just something happening under the skin. If you want to go into organs, which is like organs, internal organs, address some of these issues like cancer or kill cancer stem cells, then of course you need to go down and look at this, something which go under the skin and reach the organ and specific location where if you use the sonosensitizer, then the sonosensitizers can become stimulated with the sound instead of light, because sound can go much higher depth. It can go through and through, and that's why we use ultrasound scanning and all those things, right? These days replaced by MRI, but this is one of the reasons. So there is limitation for the PDT regardless, but there is a lot of literature on the PDT. Those are
no, it is working, I understand, but yes, these are some issues I thought I should clarify. Second thing, I just it is just an addition to what Kavita ma'am has had her query on the quantitation and things, the amount of loss.
Yes.
I think is there any clue that which is the particular molecule which is stimulated first? If it is the membrane or maybe the glycocalyx,
okay.
What is or maybe the ECM is stimulated? What is the particular molecule which initiates the process?
Yes. Yes.
If we're talking to stimulation, now there is 2 points. If you're simply saying that yes, water absorption, presence of water increases the absorption. That means water around is absorbing or simply absorbing the air, increasing its motion, meaning generating heat. That's clear. Okay. Molecular motion generating heat, and that heat is being transferred. So then there is no question altogether. But if it is the pure air which is doing the job, not via water, then I think a molecular aspect. You have shown a lot of pathways, probable pathways. That's really awesome. But it is not so. It starts always from a ROS or yes. So and ROS generation had many ways of doing it, right? So in cells.
Yes. Yeah. So that yes. So please proceed. Yeah.
No. My question is done. I just want a response from you. I mean, is it possible to get to a molecule? Are you interested to get to a molecule, or are there some person who is trying to do it?
Yeah, I mean that's quite interesting. There is of course there is a lot of because I worked in this area before, so I know people. So they are actually interested. I mean, they are continuing their interest in the photosensitizers and photosensitizers that get stimulated with light, especially, and then looking at that in the taking that from the in vitro models to in vivo models. But again, I keep telling them there is a limitation, which is the light. And then on the other side, which is like using sound. That's something I would say if you want to take this, see the effect of the ROS, because sound exclusively, because the light using the PDT and sound, using the sound, especially for cancer therapy, has been something like for around 4 or 5 decades before. So, but PDT has gone so far, SDT also has gone so far, but now PDT has been treated like photobiomodulation, which can be used for differentiating the stem cells and using the stem cells and then invigorating or rejuvenating or then differentiating into like other 3D models and all those things. But unfortunately, that is not happened for the sound. And because the sound is something like people treat exclusively for going beyond the skin layer, and it's exclusively been used for treating conditions and looking at this reaching the organs or internal organs with especially the sonosensitizers, which can give more documented, more promising effect compared to the light. Because people try to translate in vitro to in vivo and using photosensitizers, and then if you say the amount of light getting beyond the skin layer, if it is not that great, then again, the in vitro, converting in vitro models in vivo models or getting into therapeutic effect of the in vitro models is again a question. So I would definitely limit light, looking at the conditions which are right under the skin, or treating skin diseases, or using photosensitizers to reach organs to some extent below the skin, but not to the extent that it would kind of something like having a greater effect for like cancer stem cell killing and all those things. But people are working on it. I know them, and they are successful to some extent. And I don't know what will happen once these in vitro systems comes into the in vivo models that they are not aware of. They are not exclusively putting everything what they get in the in vitro system into the in vivo models and finding and stating in the publications, okay, so this comparing the results in vitro and in vivo models, but they limit their study within the in vitro systems. Suppose in cancer stem cells, they put it in vitro culture and then effect of the light photosensitizers on it, done, publication. It's not getting translated into in vivo models. But that will be more promising if you use that sound, because sound is able to get into the deeper layers, into organs, and it can be, if you're using sonosensitizers, especially targeted sonosensitizers, targeted nanoparticles, you'll be able to reach cancer stem cells, and that will be one of the promising effects. Just like another candidate is a magnets, magnetosomes, all those. So these are other molecules I would suggest. So that's, I think that, I hope I answered your question. Is that correct? Is that okay?
You have given me the full perspective, though, where the world is going on. That's really good.
Yes.
Thank you very much. It was really a brilliant presentation.
Thank you so much, sir.
Great vision.
I think we have run a little bit over. We were supposed to finish by about 6 o'clock. We're about 40 minutes.
Okay, sorry about that. Yeah.
We would like to chat on and on, but the other organizers and the registrants will have to get back and come back tomorrow.
Yeah, I understand. Yeah. Please direct other questions.
Dr. Sarjan, personally, I'll come and talk to you about a lot of topics, including some questions raised by my own student here, which I'll post.
That's great. That's great. Yes.
And also, personally, slide preparation, fantastic slide preparation.
Thank you so much, sir. Yes.
I like to get some tips from you.
I'll always be helping you, don't worry. Yeah, because my people say that I'm a bit creative, but as I said, it takes a little longer time. It's just not like copy paste anything, pictures from the articles. I try to make it more lively so that audience will listen to it, especially once it is students. Some of these slides I use for undergrad teaching, so that students don't get bored. So that's, I think it's all a matter of time, because most of the time we don't have time to work on the slides. But if you have time to work on the slides, anybody can generate beautiful and nice slides.
It is really fabulous, original arts. Yeah.
Thank you so much, sir. Thank you.
Over to Dr. Asha Devi, madam.
On behalf of convener and organizing secretary, I would like to thank for your talk in engineering 3D model systems for tissue engineering. The talk was simple and crystal clear, even a layman can understand, sir. Thank you very much.
Thank you, madam. Thank you so much. Thank you I'm sorry for taking it a little longer, but I hope you got benefited out of the talk.
Yeah, sure, sir, sure.
Thank you. The organizing secretaries and the HOD can stay back for a couple of minutes. Shall I make a quick announcement, professors?
Yes, sir. Yes, sir.
Tomorrow we are starting at 8:15. We have a lecture by Dr. Ashim Gupta on orthobiologics, tissue engineering, and regenerative medicine. Dr. Anish Patti, augmenting biology in bone and tissue repair. Are we closer to perfect repair? Dr. Debashish Das, notch signaling, dictating fate of corneal stem cells. And then Dr. Sanjay Kumar, human mesenchymal stromal cell-based regenerative approaches in mice models. And then in the afternoon, Dr. Shaji, red cell diseases, focus on stem cell-based therapy. And the last talk by faculty from VIT, Dr. Tamil Selvi, madam, who is also the organizing secretary. stem cells and acute pancreatitis. For the registrants, please be aware, you need to attend at least 5 sessions, including the inaugural, and also fill out the feedback form. There are a lot of queries about the feedback form. It will be sent to your your email ID via Gmail tomorrow. So only if you attend 5 sessions, including the inaugural, and fill out the feedback form, you will be eligible to receive the certificate. So I hope you enjoyed the first day. I found the participants were much higher in the morning session compared to the afternoon session. Hopefully you will compensate by, by participating tomorrow and coming out in full strength and learning more about stem cells. Thank you.
Sir, I formally take this opportunity to thank the chairperson, Dr. Asha Devi, who had also patience in handling the sessions. I widely thank Dr. Sarjan George for a wonderful talk, and all the speakers who have enlightened us with the depth of knowledge with the basics and applications of stem cells. So provided with this, I thank all the participants for attending this session, and make sure that you are there in all the sessions till the end of the program. Okay. So I think people are still asking about when we will get the feedback form. And I try to maintain that by the end of the session tomorrow, in between any time you will receive it. So it's not that fixed. You have to wait like suspense. Is it okay, sir? Sir, please unmute.
Yeah, fantastic, madam. I totally agree. Fantastic. Now, madam, you can actually stop the recording, madam.
Yes, sir. Sure, sir.
Ma'am, thank you, ma'am. It's my pleasure. Thank you.
Thank you. Thank you, ma'am. Sir, first I will stop with the YouTube.
Yes, madam, please do.